Unfortunately, I will not be able to take part for the whole time of the panel discussion. I write it, yeah, because currently I'm at home and must leave to my institute to take part in the dissertation council meeting. So just in the beginning, yeah, I could take part in the panel discussion.
Thank you. I am also actually traveling in the car.
Okay.
Good. So.
Welcome to all. It's 9 a.m. sharp IST Indian timings. Good morning. This is Dr. Sanil Kumar, working as assistant professor in the Department of Biotechnology, Vigyan Foundation for Science, Technology and Research. Welcome for all the dignitaries for InBix-21. This is an annual Indian Bioinformatics conference organized by BioClose. This time it was being hosted by CSIR NIST under the leadership of Dr. N. Narahar Shastri, Director CSIR NIST. In this conference, more than 300 participants have been registered across the globe from more than 12 countries. And prior to this conference, we have successfully completed pre-conference workshop during last 2 days. Now we will start the program with Saraswati Vandana by Dr. Sri Manjari. Manjari, please.
Yeah. नमो नमोऽम्ब देववाणि देवि कामवाणी कामपूरणि नमो नमोऽम्ब पाहि मां पुनीः कामरूपिणि नमो नमोऽम्ब देववाणि देवि कामपूरणि चारुशब्दसुन्दरास्य वल्गुलास्य मोहिनी व्यासवाल्मीकिकालिदासभासवासिनी भावभव्यवाग्विलासरङ्गमञ्चरूपिणी भावभव्यवाग्विलासरङ्गमञ्चरूपिणी रम्यगीतनृत्यनाट्यमेलनेन शोभिनी रम्यगीतनृत्यनाट्यमेलनेन शोभिनी नमो नमोऽम्ब देववाणि देवि कामपूरणि तर्कशब्दशास्त्रचित्रवाग्विनोदरञ्जनी ब्रह्मकर्मचिन्तयानि गूढतत्त्वदर्शिनि वेदलोकशब्दसम्पदूर्जिते मनोन्मणि वेदलोकशब्दसम्पदूर्जिते मनोन्मणि भारतीय संस्कृतेरुदारसारवाहिनी भारतीय संस्कृतेरुदारसारवाहिनी नमो नमोऽम्ब देववाणि देवि कामपूरणि नमो नमोऽम्ब पाहि मां पुनीः कामरूपिणी. Thank you.
Thank you, Dr. Srimanjari, for singing a wonderful Puja Saraswati song. Now I request Dr. Hriday Mahanta to give the welcome address. Hriday Mahanta, please. He is serving as Secretary InBix-21.
Thank you, Anil. A very warm good morning to all of you from Nice, Jorhat. Honorable Advisor-in-Chief BioClues, Professor Jayaraman Valadi, President AP Bionet, Dr. Asif M. Khan, Chief Executive Officer, International Society for Computational Biology, Ms. Diana Kovacs, invited speakers, guests, and all the participants. On behalf of the organizing committee, it is a pleasure to welcome you all to the 4th edition of Indian Conference on Bioinformatics, InBix-2021. The theme of InBix-2021 is strengthening computational drug discovery, which is also one of the dreams of our Honorable Prime Minister of India under our Bharat. Under this theme, we have received overwhelming support with more than 200 registered participants and over 92 abstracts. The speakers in the conference are uniquely placed to debate, discuss, and highlight the key points, trends, and reforms in the fields of drug discovery and allied areas for the audience. It will be great to hear from our keynote speakers on diverse areas like drug repositioning, the data era, and advanced genetics. We also have many eminent invited speakers from across the country and the globe who will be talking on the frontiers of biotechnology, bioinformatics, structural biology, machine learning, and translational omics. We have arranged an abstract and poster presentation opportunity for our participants to put forward their reforming thoughts in this vast field. InBix 2021 is being hosted by CSIR Northeast Institute of Science and Technology, Jorhat, Assam, with extended support from BioClues. CSIR NEAST is a constituent establishment of Council of Scientific and Industrial Research, CSIR, and the Ministry of Science and Technology, Government of India. The institute was established in the year 1961 as one of the multidisciplinary laboratories of the Council under its Chemical Science Group of Laboratories. Its major thrust areas are research and development activities, has developed indigenous technologies by utilizing the immense natural wealth of India. The year 2021 being marked as the Diamond Jubilee year for the Institute, we are extremely happy to host the InBix 2021 from CSI NIST. In this regard, I once again wholeheartedly welcome all the delegates across the country and globe for being a part of this conference. We are really honored to have all of you with us. I would like to thank President BioClues and Director CSI NIST, Dr. G. Narahari Sastry, for all his support, suggestion, and guidance in organizing this conference. I'd also like to thank the advisory committee, the organizing committee, the volunteers for giving such a platform to bring the eminent scientists, technologists, and young researchers together for discussing the achievements and newer directions of the cutting-edge research. After the whole day of speculation on the most demanding field of drug discovery, we have organized a cultural program to refresh the minds of all of you in the evening of the second day of the conference. I wish you all have a wonderful time today and in the next 2 days. Thank you all for your presence and participations. I declare the conference open for the further proceedings. Thank you so much.
Yeah, thank you, Hruday, for giving the welcome note. Now I request Jayaraman Voladi, Professor Jayaraman Voladi, Advisor-in-Chief BioClues, to give his concept note. Jayaraman, sir, please.
Good morning, all. Warm welcome to all dignitaries, invited speakers, and other participants. I'll just share my screen, just a minute. Are you able to see my screen?
Yes, sir. Please.
Okay. Yeah. The Indian InBix-21 is the Indian Bioinformatics Conference 2021. InBix is the flagship annual event of the Bioclues.org. Bioclues is an affiliate of the International Society of Computational Biology and Asia Pacific Bioinformatics Network. The debut InBix event, InBix-17, was organized in Jaipur, Rajasthan, followed by a series of successful events, including the InBix-19 at HMV Jalandhar, Punjab. The online InBix-20 was conducted at Northeast Hill University, Shillong, Meghalaya. This 2021 edition of the InBix is being inaugurated today at CSIR, NIST, Jorhat, Assam. BioClues has entered the 17th year of existence. As an advisor, I take pride in BioClues and her achievements. Our mission statement is built on 4 themes: mentoring, outreach, research through awareness, and entrepreneurship. We have some explicit intents, which includes creation and dissemination of consciousness of research in the field of bioinformatics and system biology through webinars, to identify practicable knowledge drivers through a comprehensive look for computational biologists, impart a complete understanding of the principle of bioinformatics algorithms and tools through interactive summaries, polls, and discussions, to provide an online project platform for mentoring graduate students while bridging the interaction of students in the field of computational biology, to cater to the needs of assimilating existing open source code and developing novel tools and web servers for research community. We also have a very active Women in Biology forum. Women bioinformaticians are more empowered than any time before, and Indian bioinformatics has a real balanced gender ratio. We have created avenues for mentor-mentee relationships to the future generation to bring them into the same boat. We have already started the Bioinformatics for School Children initiative. started by Prashant, the one and only Prashant, we have grown tremendously thanks to the leadership of Professor Narahari Sastry as president, Sugunakar Uri and Harpreet Singh as secretaries. One of the key components of the InBix series is the additional skill development workshops along with the main conference. I am happy to share that yesterday we have successfully concluded the pre-conference workshop on Galaxy community tools and workflows for chemoinformatics and NGS data. This InBix 2021 event is a 3-day event with 3 keynotes, 5 technical sessions as seen here in the slide, 14 invited talks, 2 sponsored talks, and oral presentations and poster talks. Yeah. In addition, a special session on bioinformatics for school children and 2 panel discussions on current research scenarios pertaining to COVID-19 and Galaxy India needs special mention. Our organizing committee has also planned a cultural event. Just a second, it's not moving. To give you an opportunity to showcase your talent in extracurricular aspects, which many of you may possess, and also to give some lighter, enjoyable moments during the 3-day intellectual fiesta. We have an active involvement in the conduct of the InBix-21 from CSIR NIST under the able stewardship of Professor Narahari Sastry. They have very excellent programs of international scientific excellence, which includes computational modeling and informatics. They also have a big program on open source computational drug discovery. And also they have several software suites, including the most famous molecular property diagnostic suite. On behalf of the BioClues organization, CSIR NIST, the entire organizing committee, ISCB, AP Bionet, and the BioClues community, I welcome all distinguished speakers, delegates in this August virtual conference. I wish you all take the conference to greater heights as envisioned by able leaders. InBix has been taking leaps and bounds with successes. Wishing you all success in the conference. Thank you.
Yeah. Thank you, Professor Jayaraman sir, for giving the brief concept note on BioClues. Thank you very much. Now, I will play the recorded version of Dr. Asif M. Khan, President AP Bionet Close. He will give the recorded note about AP Bionet address. I will share my screen. Are you able to see my screen? Sugunakar sir?
Yes, Anil, please go ahead.
Yeah, fine. Thank you.
Thank you, InBix and BioClues, for inviting me to say a few words for the welcome. Good morning, everyone. On behalf of the Asia Pacific Bioinformatics Network, I welcome you to the 4th Indian Conference on Bioinformatics, or InBix 2021 in short. Hello from Dhaka, Bangladesh, where I'm currently conducting a bioinformatics workshop for medical doctors, and today is the last day. As the timing of the workshop overlaps with the opening of InBix 2021, unfortunately, I'm not able to welcome you live and have to do through this pre-recorded message. My apologies for that. I want to firstly congratulate the organizers in making this event happen. We just concluded our very own flagship conference, InCop 2021, and we know how much work and coordination it requires. So a big sincere thank you to the people behind InBix-2021 for their tireless effort and contributions to present this platform to us, the community, for a wonderful discourse on bioinformatics and its impact on science. InBix is a flagship conference of BioClues, which is India's largest bioinformatics society, and it turned 17 this October 2021. Although this is the fourth year, BioClues has been organizing virtual conferences over the last 16 years. Sustaining one, and on top of that, growing a society, never easy, as it is a voluntary work that one has to do on top of their day job and caring for their personal life. So it is really something for BioClues to have come this far, 17 long years. I wish BioClues a long life, supported by motivated individuals who come together to advance the mission and vision of the society in its pursuit to promote and advance science through bioinformatics across India, the subcontinent, and beyond. I look forward to its 20th anniversary. The program committee of InBix has prepared a diverse, exciting, and thought-provoking program along the theme of notable aspects include the 3 luminary keynotes,
Anil, are you there?
Yes, sir, I'm there, sir.
We are not able to listen to the audio of Asif.
Audio. Is it playing?
Right now it is playing. Thank you.
May he rest in peace. I want to end by saying that BioClues is an important affiliate of Bionet and a key player contributing to the bioinformatics community effort in the region. Bionet will continue to support BioClues in its efforts wherever we can. We are currently working with BioClues in 2 main areas. Number 1, addressing bioinformatics grand challenges. And number 2, coming together to organize the first Bioinformatics Congress in 2024, where we hope for the first time to bring together the major bioinformatics communities in the region on a united platform. I recently recognized a hashtag that Prash likes to use and which I have come to like too, and it is so apt for today and our future together. So here it goes. Hashtag Together We Can. I wish you the best of InBix 2021 and have a great time. Bye for now. Thank you. Thank you, InBix and BioClues, for inviting me to say a few words for the welcome.
Thank you, Dr. Asif Khan, for sending the pre-recorded version of your AP Bionet address. Now, another pre-recorded version by Diana E. Covers will be played, and she will address the ISCB.
Welcome. I am Diane Kovacs, the CEO of the International Society for Computational Biology, lovingly shortened to ISCB. Thank you for letting me be a part of your event, even if only in this small way. We are delighted to have BioClues as one of the ISCB affiliated regional groups representing India. BioClues has been working towards the development of a mentor-mentee relationship in the area of bioinformatics, functional genomics, and systems biology. We've been closely watching their activities over the past years and the growth of the Indian Conference on Bioinformatics. We wish them all the success this year with InBix-2021 and you as a participant, your success to be inspired, encouraged, and connecting with your fellow colleagues.
Anil, are you there? Dr. Anil? Anil, are you there?
Welcome. I am Diane.
Thank you, Diana, for sending the pre-recorded version. Thank you once again. Thank you all. And now I request Dr. Sugnakar Ure, Secretary Bioclus, to give his vote of thanks. Sugnakar sir, please.
Yeah, thank you, Anil, for the warm welcome. Yeah. Now, I'm here to give my vote of thanks. I welcome all the guests once again for gracing this occasion. And as one of our secretary for InBix 2021, Hriday Mahanta has used a word saying like Atmanirbhar Bharat. It signifies the self-reliant India. That's where the India is moving towards developing the scientific temperament across the different facets. And as a part of that, strengthening open computational drug discovery has been the thematic area on which the InBix is focused on. So I extend my thankful regards to our beloved president, Dr. Narahar Shastri, our advisor-in-chief, Jairam Waladi, for his concept note. I extend a warm regards to AP Bionet President, Asif M. Khan, and ISAB CEO, Diana E. Kovacs. I would like to thank our beloved President, Bioclus and Director, CSR NEAST, Dr. G. Narahar Shastri, for all his support, suggestions and guidance throughout for the conduct of this event. I further extend to thank our founder, Bioclus, Dr. Prashant Suravarjala, Women in Biology Chair, who initiated, Dr. Renuka Suravarjala, and other executive team members who initiated Bioclus. There's a dire need of bioinformatics way back to 2005. That's where the visionary leaders like Prashant, Pritish has drived to start Bioclus India to enlighten the mentorment relationships in India. So I heartfully thank all the participants and the dignitaries over here. Thank you. Thank you so much. Now, yeah, thank you.
Thank you, Dr. Sugnakar Ure, for giving vote of thanks. Now the inaugural session comes to an end. We'll end with national anthem. I request everyone to stand up for the national anthem. Now I'll play the national anthem. Please stand for the national anthem.
पंजाब सिंध गुजरात मराठा गणगणमङ्गा विन्ध्य हिमाचल यमुना गङ्गा उच्चल जलधितरङ्गा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you, everyone. Now, the next session starts at 9:45. I thank Dr. Manjari, Dr. Sridhar for giving the welcome note, and Professor Jayaraman Valadi for the concept note, Dr. Asif M. Khan, President AP Bionet, for giving AP Bionet address, Dan E. Kovacs for ISCV address, and Dr. Sugnakar Ure for vote of the thanks. Thank you, one and all. Thank you. Sugnakar sir. Hello, Sugnakar sir.
Now we have a buffer time with networking. We'll start the session at sharp 9:45. Thank you one and all.
Thank you, everyone. Thank you.
Anil, you stop sharing the screen.
Okay, sir. I'll stop.
Yeah, Rohitash Yadav, could you please stop sharing your screen? Video. Rohitash Yadav.
He played. Anil played. Anil played, but this is coming on now. This fellow. It's fine, no problem.
Thank you very much, Professor Pareko, for waking up very early.
Thank you, Sastri. It's okay. Don't worry. Yeah.
We are very much honored to you. And I have to visit, to travel to a place called Mizoram. And I have to take a 100-kilometer drive and then take a flight. So I finished about 150 kilometers and then having some tea.
I have some tea with me. Yeah.
You up at 5 in the morning and then get ready and
Sure. Today it's early time for me, but usually I sleeping up very early. So don't worry, it's okay. Unfortunately, I could not come to the institute so early. So I presented from my home and then go to institute.
Okay, thank you.
Welcome.
So last 2 days we have this workshop on Galaxy.
Yeah.
which fairly well attended and good discussions have taken place. And today we are going to start this workshop for the next 3 days. And
That's
hopefully
important. Yeah.
these discussions help to strengthen the computational drug discovery in general in the world, but particularly in India, strong support from people like you. Because Professor Paraiko, for the benefit of all the participants, is one of the very, very strong supporters of Indian science. And he had a very long collaboration with Punjabi University, Patiala, with Dr. Rajesh Goyal. And for the last 7 years, we are having reasonably good interactions, published a few papers together and shared lots of thoughts. And definitely this interaction will be very useful for all the participants. And I really thank him for taking the time very early in the morning and be with us.
Oh, thank you.
Nagamani or Sugunath?
Yes, sir.
Yeah, please, sir.
We will mute, sir.
A small problem with this network connection. May I request you to introduce Professor Paraiko?
Yes, sure, sir.
Yes, sir. We'll coordinate, sir. Thank you so much, sir. 9:44 we will start. 9:45 will start.
I don't want to create a problem during the introduction if the bandwidth is not good.
Is it good?
Okay.
Sure, sir.
Yeah, sure, sir.
Yes, sir.
Namaskaram andi, Dr. Shishla.
Sir, Namaskaram.
Nice.
Good to see you. Albeit virtually.
So I'm sure that you will like Professor Paraiko's talk.
Yes. I tuned in just in time.
So Professor Holger Golke has joined.
No, sir.
Okay.
Sir, we are sending me a reminder, sir. We are sending a reminder to him now. Thank you, sir.
Okay, because it's really for him also, it's very early.
Yes, sir.
5
Yes,
o'clock or so.
completely agree, sir.
Dr. Sugnakar, am I audible to you?
Yes, Anil. Yeah, you're audible. Thank
I
you.
will start at sharp 9:45. Okay, wait a minute.
Yeah, thank you.
Dear all, welcome back. After the inaugural session, now we are moving to the scientific session. And now I request Dr. Narahari Shastri sir to chair this session. We'll have one keynote talk followed by panel discussions. Dr. Shastri sir, please take the session. Please chair the session.
Thank you very much. And it's a great pleasure for me to have Biomedical Research Moscow, who is one of the very, very effective researchers in bioinformatics and played an important role in trying to make these programs available and have a great value addition to this whole community. As I am traveling, I'm requesting Dr. Selvaram Nagamani to introduce Professor Pareko, and I thank him for taking his time despite his busy schedule, a little bit odd hours. And more importantly, at a personal level, I thank him for all his support and mentorship for my group as well as several groups in Indian science by generously collaborating with us and also interacting with us. Thank you, Vladimir, for everything that you have done in science, and we will continue to have your support for us. And I ask Nagamani to introduce Professor Pareko.
Thank you, sir. So, Professor Vladimir Poreko, he is currently principal researcher and head of department in bioinformatics and laboratory for structure function based drug design in the University of Moscow. And he has received various prestigious awards, and he is the member in several scientific bodies in Russia, and that includes member of Russian Academy of Sciences, member of Council for Grants of the President of the Russian Federation, the expert board of the Skloko Foundation, and expert council of the Cyrus University of Science. And his major area of research interest includes bioinformatics, chemoinformatics, USAR, molecular modeling, and also software development. And he is an editorial member in various prestigious journals, including ACS Pharmacology and Translational Sciences, ACS Omega, Journal of Chemical Information and Modeling. And he has more than 300 publications with a Hutch index of 48. And one of the remarkable contributions from his group is PASS online server, which is popularly known for computational drug repurposing and identification of biological properties of the small molecules. So with this small introduction, I would, on the behalf of BioClues team and CSIR NIS, I would request Professor Poreko to deliver his keynote address on drug repositioning, current lessons from the COVID-19 pandemic. Sir, please, sir.
Thank you, Dr. Sastry and Nagamani for introduction. And with this, I will try to share my screen. And
yes, sir.
yeah, let's see. Hope you see my screen.
Perfect, sir. Please go ahead.
Thank you so much. So once again, I would like to express my gratitude to the organizers and particularly to Dr. Garikapati Narahari Sastry for inviting me to present a keynote lecture at this very prestigious Indian conference. Actually, it's not surprise that at this time we are talking about current lessons from the COVID-19 pandemic. Because no doubt that this situation touch everybody in our community, in our countries. And first of all, I would like to express my acknowledgement to the people who are taking part in our joint studies. First of all, my team from the Institute of Biomedical Chemistry. Also, for our collaborators from Russian Medical Academy of Postgraduate Education and from Chumakov Federal Scientific Center for Research and Development of Immune and Biological Products of Russian Academy of Sciences. Of course, no study are possible without funding. Therefore, I would like to express my gratitude to the Russian Ministry of Science and Higher Education and Russian Foundation of Basic Research that have supported our efforts in this field. And also we are grateful for the academic subscription on the databases and software provided by Clarivate Analytics and Chemaxon. When we are looking to this very grim statistics, we see that both in India and in Russia, COVID-19 is becoming more and more important cause for death and for reducing the life expectancy. Therefore, of course, a lot of different information appeared in the internet and published sources. When you are trying to make the search using COVID-19 as query with Google, you can see about 4 billion different records, which actually correspond to very, very diverse fields of human life and research. When we see what we have due to this search in India, it's also about 4 billion records. And of course, when we are talking about drug repurposing, the number of records is almost the same. Actually, it reflects that the drug repurposing is one of the main routes in our possibilities to overcome the common challenges. Unfortunately, COVID-19 is not just pandemic, it is accompanied by infodemics. Because a lot of different posts in the internet, in the public media are just opinions of people and are not grounded on the strong scientific basis. Therefore, it's very difficult to filter out some fake news from the whole number of informations, news which are coming in each day. When we are doing similar search in Web of Science and core collection of Web of Science, we see over 185,000 different publications. And a little bit surprising that the first publications are accounted for 2019 and even 2018. Because we know about these pandemics just from 2020. Therefore, let's see what is the first publication published in 2018. It's actually research creation to the community, pedagogy, feminist maker cultures, and the critical work on making face mask in the time of COVID-19. Therefore, it is probably just some delayed publication. So maybe this published source was published later, but it's actually accounted for 2018. Because in 2018, the term COVID-19 was not known at all. The next year is announced by UNESCO by the International Year of Basic Sciences for Sustainable Development. And at the web page of this Year of Basic Science, you can see that our possibilities to answer on any biogenic threat like COVID-19 are based on the results achieved in basic sciences. What we know now, it's actually possibility to measure temperature very quickly by infrared thermometer, to actually provide some possibility to watch about movement of people using cell phone technology, to prevent some COVID-19 complications using dexamethasone, to provide some treatment of COVID-19 patients with remdesivir, to analyze the genetic sequences of the virus, which allowed us to get some vaccines and diagnostics, etc., etc., etc. So the basic sciences and results achieved in the framework of these sciences is very important for the survival of mankind at all. The decode of SARS-CoV-2 genomes, which are currently more than 5 million in the GIS-AID resource, provides the prerequisites for the development of diagnostics and vaccines. Here you can see the major vaccines from Russia, Sputnik V and GAM-COVID-Vac, which are, of course, used widely for vaccination of Russian population. Unfortunately, not so many people are still vaccinated. The latest news, latest estimates was about 50%, but maybe just the first dose of vaccine. Having any information about genomes of SARS-CoV-2 and some other relevant viruses, we can use this information also trying to find some similarity in genetic and protein sequences, which actually allowed us to think if the drugs developed against other viruses could be applied for treatment of SARS-CoV-2 infection. And we know that in 2012 we have MERS-CoV infection, and in 2002 SARS-CoV infection. Unfortunately, the drugs that were started to be developed against these viruses were not approved for use in medical practice. Therefore, they are not immediately ready to be applied for treatment of SARS-CoV-2. I believe that you have some temporary guidelines for treatment, for prevention, diagnostics of new coronavirus infection in India. We also have such guidelines in Russia, and first version actually was in January 2020. And you may see that at that time there were no etiologic therapy for treatment. Therefore, it was proposed to use ribavirin in combination with lopinavir and ritonavir, interferon beta, aprotinin, corticoids, flavonoids, vitamins, etc. Fortunately, at this time, we have some drugs which are claimed to be a tiotropic therapy, including favipiravir, remdesivir, umifenavir, and interferon alpha. However, these drugs are still in clinical trials. Therefore, they are temporarily approved for use in acute emergency situation. The number of clinical trials that are currently carried out in the world is about 3,000. And of course, most of them are associated with drug repositioning. For instance, remdesivir, which is one of the first drugs claimed to be a tiotropic therapy, initially was developed for treatment of some other viruses. And when we see the information about remdesivir in PubMed, you can see about 1,800 records. And first records are very, very low. And now we see the way how it was repositioned to development for COVID-19 therapy. First, it was tried against Ebola virus in 2015. And then, very quickly, it was repositioned in different countries as the tiotropic therapy treatment for COVID-19. But it's still in clinical trials to prove or disprove its efficiency in treatment of this disease. And of course, remdesivir has some pitfalls. First of all, it could not be taken orally. It's only available for intravenous injections. Moreover, it produced some severe side effects, which also restrict the possibility of its efficient use. Fortunately, just a week ago, new oral drug, molnupiravir, actually was approved by UK regulatory agency for patients with COVID-19 based on the successful results of clinical trials. I don't know what is the situation with this drug in India, but in Russia, it's not approved yet, and forecast is that it may be approved just in 1 year after this time. To see the overview of COVID-19 antiviral therapy, you can see this good review, and some drug candidates are discussed here in detail, so I will not talk about them. Let's compare the traditional way of research and development with the drug repositioning. As you know, traditional way has many stages, from basic studies to clinical trials, through experiments in vivo and in vitro, and then, in successful case, to drug registration. Drug repositioning allowed immediately switch to the phase 2 clinical trials. Of course, if the drug is applied in the same dosage and the same route of administration. And of course, drug repositioning significantly reduce the time, expenses need to get the registration of the drug, and also increase the success significantly. When we look into computational approaches for drug repurposing in Google Scholar, you can see that there are about 18,000 different records. Therefore, computational approaches are very widely applied for drug repurposing. Also, very early, this approach for discovery of new drugs against COVID-19 were based on in vitro studies, because SARS-CoV-2 virus is replicated in cell cultures. For instance, HCV virus is not replicated, which actually a very complicated finding of new effective therapies. But in case of SARS-CoV-2, it's possible to see which pharmaceutical agents could inhibit the replication of virus in the cell culture. And maybe first attempt published in March 26, 2020 was possibility to reposition dipyridamole for patients with COVID-19. And this is very attractive opportunity, because in this case it was shown that dipyridamole inhibited the entry of virus into the cells. And since it's also prevented thrombosis, it could be considered as multi-targeted drugs. But of course, the studies of clinical efficacy is still ongoing for this compound. Also, you can see some other approaches in cell assays published in April. You can see that they tested more than 1,500 compounds and found 13 actives with certain threshold of activity. And in another study, they studied more than 5,000 compounds with 64 actives. But unfortunately, no one compound from those studies coincided with each other. That actually means that each particular assays are not standard and don't correspond to each other. We performed some comparison of experimental studies for anti-SARS-CoV-2 with the purpose of drug repurposing. And you can see that different cell lines are used and different number of compounds was tested, and just a few compounds were coincided with the results in different studies, but for some of them, not enough clinical efficacy was already shown. Therefore, in vitro studies in cell assays, still not enough to conclude that compound will be efficacy used for treatment of COVID-19. It was our surprise to find that there is already some web service in March 2020, which allowed to predict anti-coronavirus activity. This web service was developed by Chinese colleagues, which is not surprised because they started very early to fight against COVID-19. But when we created the independent test set, we showed that balanced accuracy is not very good, and results predicted by different methods don't coincide with each other. We also have found some other issues with this web service. We sent email to Chinese colleagues, and partially they actually fixed these issues. In this slide, you can see an overview of the current methods that are used for finding anti-SARS-CoV-2 agents. They are from in silico through in vitro and then in human agents. Of course, design de novo, synthesis de novo requires the next synthesis, and screening in silico, confirmation in vitro, and is associated with long-term research and development. Ligand-based drug design, target-based drug design actually requires libraries of the synthesized and synthesizable compounds, and also requires a lot of time for confirmation in vitro and in vivo. When you start by biochemical screening and cellular screening, it takes less time. And of course, the most quick way is drug repositioning. You have drug substances in different libraries, like Prestwick chemical libraries, and it's possible to introduce the successful candidates to the medical use very quickly. Of course, all this approach are combined very often. In this slide, you can see the process of interaction of SARS-CoV-2 virus with cells and the induced pathological processes. Therefore, the main targets which are currently studied are ACE2 receptor, serine protease, which actually take part in the processing of virus, main protease, papain protease, RNA-dependent RNA polymerase, and some other proteins of virus and human. Network pharmacology approach is widely applied for analysis of interaction of virus with human and to help to identify more and more proteins that are involved in the pathological process. In this slide, I try to show that in silico approach could be applied for very many chemical compounds. The next is in vitro, the next is in vitro cellular assays, and then in vivo and clinical. Of course, the number of compounds that could be studied in different studies significantly reduced from the lower part to the up part of this triangle. And when we see the percent of different studies, it's not surprise that a lot of studies are associated with in silico screening, then in vitro, then in vivo, and then clinical. A little bit surprising that the number of clinical studies is more than the number of in vivo studies, which actually reflects the drug repurposing. When you are trying some drugs in clinics without doing in vivo studies. The most popular targets are 3CL-Pro, PL-Pro, spike protein, ACE2, RDRP, serine protease, etc. But the problem is that there is very low correlation between the in silico and in vitro methods. That actually reflects the incompleteness of information which are used for ligand-based approach to finding some anti-SARS-CoV-2 agents, and low quality of 3D structures that are used for molecular modeling, trying to find some anti-coronavirus agents. However, it is necessary to stress that low correlation is also observed for different in vitro studies. In these papers, Savosina and Slati published in 2021, it was shown on many statistical data. And the reason for such low correlation is the lack of standard assays and the lack of proven reference compound, which is the cause for low agreement of test results with each other. For instance, when we are looking on the in vitro assays. They are very diverse, even for the most popular targets, main protease of SARS-CoV-2. Many different materials, wild-type isolates, laboratory strains, some different cells, etc., are used for studies. Many different methods of activity detection, chemiluminescence, dye-assay, fluorescent assay, RNA assay, etc. So it's not surprising that the results of separate studies very often do not correspond each to the others. For instance, for this drug, you can see the results of docking, which actually generates the hypothesis that it could be the potent inhibitor of this main protease of the virus. And docking into Mpro SARS-CoV-2 of nirmatrelvir also showed that it could be the candidate for repurposing. And in literature, it's shown in vitro that nirmatrelvir really inhibiting SARS-CoV-2 Mpro with micromolar, 10 micromolar IC50 value. But it's still in clinical trials and no results are available yet about its clinical efficacy. Another candidate is disulfiram that was docked by our colleagues from the Zelinsky Institute of Organics and Chemistry, and they confirmed this activity in vitro with reasonable micromolar IC50 value. And I think that the number of such studies is very, very big. In the past year, it was announced joint European Disruptive Initiative, Grand Challenge Against COVID-19. It was announced in May, when not so many information about SARS-CoV-2 virus, about pharmaceutical agents active against it was known. But the European JEDI challenge actually invited many international teams to try to find some promising hits among the billions of molecules which could be actually used against COVID-19. And the libraries which should be screened must include the approved drugs. Therefore, also in these studies, some drug repurposings were considered. And goal of this challenge was to find compounds that inhibit SARS-CoV-2 with IC50 at this threshold. And the people should select at least 3 from the list of 6 targets. Virtual screening must be provided by 3 independent computational methods among more than 1 billion available compounds, including the launched drugs. Our team actually selected 4 targets, 3 targets of viral protein and 1 is human protein. And we actually tried to develop the classifier of different biological activities of pharmaceutical agents active against SARS-CoV-2, and then created the training set using also homology in sequences with some related viruses, and then actually applied machine learning methods and some other methods to find the potential hits. The general results of our study for more than 1 billion molecules were based on the application of chemical similarity assessment, machine learning methods predictions, and then visualization of the results using some molecular modeling methods. We selected the requested number of hits and submitted it to the organizers of Jedi Challenge. And the main overview of these results, in which many teams took part in the past year, were on this slide. You can see that totally more than 54 billion molecules were screened with a lot of hours of high-performance computers. And more than 130 teams took part in this challenge. And of course, the molecules which were selected are synthesized, but first the organizers tried to provide the expert assessment of the submitted results. And our team passed to the top 20 of the 130 participating groups. And at the moment, 38 molecules that we selected were synthesized. We actually don't know which one. Organizers don't provide us with this information. Yeah, but results of the testing is still not announced. And probably due to some inside information, I know that the results are not so spectacular that the organizers expected. It is not a surprise because, as I mentioned, in May and June past year, we still have very small information about SARS-CoV-2 and pharmaceutical agents which are active or maybe not active due to the contradictions in the different assays. Recently, Alexei Zakharov, who is currently worked in NCATS, but formerly he was PhD student in our institute, proposed two-stage approach for finding SARS-CoV-2 inhibitors that help to overcome the issues of inconsistency of the published information. Therefore, they actually test some compounds in their cell assays and then get the second round using the results obtained in the same cell assays as the training set, which allowed them to increase hit rates to 16%, identify 3 promising chemotypes with sub-micromolar activities, which are also active against the mutant viruses. And of course, they developed some in silico predictive model, which is available at the NCATS website. In the framework of grant of Russian Foundation of Basic Research, we have developed the informational computational system anti-COVID-19. And it's accumulate data from publicly available and commercially available resources about disease, about biological processes and pathological processes, about potential targets, and about some potential pharmaceutical agent. Then this information is used to create some predictive models based on the integration of the available data. And then, of course, we are able to make some predictions. This is general interface of anti-COVID-19 portal. And for instance, as I told you, we have here informational model about disease, biological processes, targets, and pharmaceuticals. And of course, we have some predictive models which are used on the past prediction and some other methods for similarity assessment developed by our team. Here you can see the prediction for remdesivir. Input is just structural formula, and when you clicked on predict anti-coronavirus activities, you can see the results. And you can see that for remdesivir, anti-coronavirus inhibition activity is predicted with very high probability PA and low PI. But the main target, which is RDRP, is not predicted for this drug. It's also not surprise because remdesivir is actually pro-drug. It's phosphorylated in the organism and is bind to the RDRP, RNA-dependent RNA polymerase, in the phosphorylated form. Another example is molnupiravir, which could be used as a cure to find some structural analogs in the database, included more than 4,000 of worldwide approved drugs. And you can see some candidates with not very high level of similarity, but anyway, they could be tested by some other complementary computational methods and then in vitro for the appropriate activity. In the framework of the project supported by Russian Ministry of Science and Higher Education, we are currently analyzing the opportunities for repositioning of drugs against main protease of SARS-CoV-2. We collected information about different studies in cell-based or biochemical assays, information about registered pharmaceuticals by Russian regulator authorities, about clinical trials, and try to identify the most promising candidates for study in vitro and in vivo to confirm or to disprove their possibility to be used against SARS-CoV-2. And I should stress that this is very, very complicated pathology. Therefore, just recently, World Health Organization published definition of post-COVID-19 syndrome, and you can see that it creates some depressive and anxiety disorders. Actually, its influence on many different organs and tissues in the organism. And the hypothesis is that in each particular patient, its influence on the tissue or organ, which is not so good in this particular patient. Therefore, of course, depression is associated with general situation with COVID-19 pandemics and some bad expectation of the people. Therefore, it's very important to see that, for instance, fluvoxamine, widely used antidepressant, actually improve the status of COVID-19 patients. It's shown in very, very wide clinical trials. The mechanism of antiviral action, if it exists, is still unknown, but actually it's maybe associated not just with direct antiviral actions, but also with improvement of general patient's status. Of course, despite the very promising way of drug repositioning, there are some pitfalls. And in particular, so many publications for so many drugs, actually you can see that according to the Cardite database, more than 600 pharmaceuticals are suggested for repositioning. Therefore, it may actually restrict the patentability of the appropriate findings, which actually reduce the interest of big companies to the drug repurposing. But I think this is a situation where we should actually overcome the problem of patenting, because pandemics is worldwide. Therefore, we should not restrict the possibilities for use of drugs against this disease. Therefore, we are also trying to permanently improve the training sets, to combine the in silico, in vitro, and in vivo studies. And then the promising candidates could be going to the clinical trials. About 30 different drugs are currently in clinical trials in Russia against COVID-19 and associated complications. Jointly with colleagues from Russian Medical Academy of Postgraduate Education, we developed the portal which provides a consultation for clinical pharmacologists which are working in the red zones, providing the support to the physicians which are working in the red zones. And also, of course, the different complications like thrombosis, cytokine, inflammation, etc. should be taken into account. Recently, jointly with our colleagues from different countries, by initiative of Alex Tropsha and Artyom Cherkasov, we published a critical overview of computational approaches employed for COVID-19 drug discovery. And we posit that truly impactful computational tools must deliver actionable, experimentally testable hypotheses, enabling the discovery of novel drugs and drug combinations, and that open science and rapid sharing of research results are critical to accelerate the development of novel, much-needed therapeutics for COVID-19. Therefore, I would like to summarize the lecture, and most home-taken lessons are that drug repurposing is one of the current trends in discovery of more safety and effective pharmaceutical agents. It is the only way for immediate response on COVID-19 pandemics and other possible biogenic threats. And of course, cooperation between the researchers working in different fields of organic and medicinal chemistry, pharmacology, virology, etc., is necessary to combat this challenge. And of course, the main problem here is the assays, because to have possibility to test the live virus, it's necessary to have laboratories of very high biological safety level, like BSL-3 or BSL-4, which actually we have very, very limited number in Russia, and I'm sure worldwide as well. And another point is that COVID-19 is a complex pathological process. Its curation requires application of several drugs with different mechanism of action, taking into account the peculiarities of the patients. And of course, multi-targeted drugs are welcome in this field. The further fundamental biomedical research is needed to fill the lack of knowledge about the biology of this virus, which will increase the effectiveness of countering these and new biogenic threats in the future. With this, I would like to thank you for your kind attention, and I'm open for your question and discussion. Thank you.
Thanks a lot, Vladimir, for a fantastic lecture, which is highly contemporary and also we are extremely happy to know the activity that is led by you in the European Union about this COVID activities. There are plenty of questions that I could see, and I request Nagamani and the team at NICE to coordinate the question answer. And your talk was also streamlined in YouTube, and so there may be questions to be collected from that. We have about 11 minutes for the question answers. Nagamani, can you please go ahead?
Yes, sir. Sir, thank you, sir. Once again, thank you for your wonderful presentation. And there are few questions in the chat box in the MS Teams. So the first question is from Shivam Pandit. So he is asking, there are many repurposing studies are carried out against SARS-CoV-2 by computational studies and even showing promising results. But however, these compounds are failing in clinical trials. So whether any wrong in the strategy while repurposing studies or whether we are missing something through computational studies. So he asked this doubt.
Okay. Actually, as I stressed in my lecture, the bad correspondence between different results and low quality of the 3D structures which are already available in PDB reduce the efficacy of in silico studies. Moreover, from my point of view, at this moment, we have not congruence between different test systems in silico, biochemical assays, and cellular assays. And also another point is that it's very difficult to perform the clinical trials to select the appropriate group of patients, because most of repurposing is directed to the direct antiviral action. But such compounds are effective only in first days after the infection. So when the people are in the hospital and we already have 3 or 5 days after infection, direct antiviral agents are not very effective. In more detail regarding the issues and promises, I address all of you to our review published in chemical, sorry.
That chemical review.
Yes, chemical reviews, because it's available, freely available, full text, and you can see more detail which is considered in this review.
Sir, in connection to the same question, there are multiple promising potential targets are available. So can we look after combination methods like a synergistic effect?
Yes, you can. Actually, a lot of studies with application of network pharmacology approaches are oriented to this goal, to see the synergistic effect in the complex networks of pathological processes in organism. I did not touch this in my lecture in detail, but you can see a lot of publication regarding this. But as I say, the most limited problem here is the low quality of information on which all these in silico studies are based. So just in a few years, probably we will have enough quality of information which allowed us to get more conclusive suggestions.
Sure. Thank you, sir. Sir, there is another question. So when we are doing this computational, in silico studies, so when we are trying that in silico outcome through in vitro or in vivo. So we are facing very much a different ambiguity. So how we can optimize and troubleshoot when we are having the contradictory results from in silico to in vitro or in silico to in vivo?
As I said, actually, in this case, the only approach is to select the results which are confirmed by different approach. So when you have 1,000 molecules, for instance, which are candidates or hits for the anti-SARS-CoV-2 activity, you should provide different computational methods based on machine learning, artificial intelligence, and molecular modeling, in vitro biochemical assays, in vivo biochemical assays using the cell lines, and then select only small number of candidates for which you have the results which are positive in all these independent testing. That was recommended by Artyom Cherkasov in the paper of Mslati et al. In Journal of Chemical Information and Compute and Modeling. Yeah, and you can also get this paper from this journal and use this recommendation.
Thank you, sir. And another question is, you have identified some of the molecules for computational repurposing. So these molecules, when you are giving this molecule as a input in the PAS online server, so whether you got the same predicted activity through the server. So in case if you didn't get that, what can be the reason?
Excuse me, I don't get the question. What is the question?
Actually, sir, that one student, he asked the performance of the PAS online server. For example,
Yes.
like you have explored so many molecules in silico and in vitro also, which is identified as a anti-COVID molecule. So whether you got the same result when when you're given in this PAS online server.
Actually, in PAS, when you have results, it means that it's only prediction, which should be tested in the experiment. When you have no result in PAS, it means either that molecule does not suit it to the criteria which restrict the structures that could be sent to this web service, or that nothing was predicted. It has nothing similar to the training set which are used in PAS.
Okay, sir. And one more question, like whether you have come across a single molecule which can be targeted for the multi-targets as far as this SARS-CoV-2 concerned. Single drug for multiple targets.
It's okay. If you have such result, yeah, it's possible that you have multiple mechanism of action. Yeah, but maybe not. Maybe it's just false predictions. It's also appeared sometimes.
Yes, sir. And I would like to take the last question from one more student that he is asking, like considering this immediate need to design drug for COVID-19. So how long it may take for identifying a drug as a good candidate through through drug repurposing studies?
For computational part, it may take a few minutes, but unfortunately, in silico prediction must be confirmed in different experiments and then finally in clinical studies. And as I told you, there are many different inconsistencies between these studies. Therefore, even if you have compound active in some cellular assays, or even in some experimental animals, like for instance, Syrian hamsters. Actually, you are not sure that the compound will be efficacious in the clinical trials. But in silica, actually, it's the easiest and the very quick way to get some hits, which should be confirmed in the experiments.
Thank you so much, sir.
Thank you. Dr. Urmi, you have a question? Okay. There are no questions. I would like to thank Professor Poreko for his excellent talk. Not only about the topics that he has covered on COVID-19, but also the reflection on the knowledge-based approaches and how we can look at polypharmacology, network pharmacology, and try to take a stock on the current molecular modeling and data science and other approaches of molecular modeling in the COVID discovery. So with this, we will be very happy and honored to have an excellent keynote to start the lecture. And I request all of you have a big round of applause for the excellent talk for Professor Poreko. And I give back to the audience.
Thank you so much.
Sir, I request everyone to please switch on your camera. Thank you.
To take a photo.
Yeah, to take a photo, please, everyone.
So that we can have a group photo. Please, all of you switch on your cameras.
Yeah, right. Screen.
So,
Yeah, thank you, Anand. Anil, please, now you can take over.
Yeah, thank you. Professor Vladimir Poreko has given and wonderful insights into drug repositioning, how it helps in identifying new drugs for COVID-19 pandemic. And once again, I thank Professor G. Naraj Shastri sir for chairing the session and also Dr. S. Nagamani. Now we move to the next round, that is panel discussion. It's a panel discussion one. Where are we heading as researchers post-COVID times? With the panelists, Professor Vladimir Poreko, G. Naraj Shastri sir, Professor P.K. Gupta, Holger, Jayaraman Valadi, Andre, Urmila Kulkarni-Kale, Vibha Tandon, Anshu Bhardwaj, Himakshi Sharma, Esther Jamir, Aparna Banerjee, with rapporteurs Prashant Surojala and Sugna Karure. Please start the panel discussion one. Thank you.
Yeah, thank you. Thank you very much, Anil. I hope I am audible and my screen is visible. Yeah.
Yes, Prashant. Yeah, thank you, Anil.
Yeah. So thank you so much. It was a wonderful talk by Professor Poreko. That has set a kind of a wonderful precedent. And we just thought, we usually have a panel discussion after every keynote. And I hope, the wonderful keynote has already set a kind of a precedent. So Sugunakar and I would like to take this panel discussion on something called where are we heading as researchers post-COVID times. So I would just like to confirm if all the panelists are here, I presume. I'll just have a quick introduction. I hope you would appreciate, or if you don't mind, if I simply call by first names. So these are all the lead guests. So Vladimir Poreko is needs no mention. He was just a keynote speaker that was wonderfully introduced by Narayana Shastri. Narayana Shastri is the CSR DG and also the president of BioClues. PK Gupta is and my reverse mentor and the mentor. He is a professor emeritus at Padri Charan Singh University, Meerut. He needs no mention. Also, Golpe is our another invited speaker. He'll be speaking today later. We have Jayaraman Baladi, who is also a professor emeritus at Flame University, Pune. He is also an adjunct professor and emeritus at Savitribai Phule University. We have Andra Guinness, who is another speaker. I think is going to give the talk later. And Urmila Kulkarni, who is an information scientist at SB Pune University, and also one of the other advisors like Jayaraman Baladi. We have Vibha Tandon, who is going to give her scintillating talk maybe tomorrow. We have Anshu Bhardwaj, who is a principal scientist at MTech Chandigarh, who has given a wonderful talk day before yesterday as a part of a pre-conference workshop. We have Imakshi Sharma, who is a scientist at CSR NIST, and also the same is with Esther Jameer. And we have Apna Banerjee, who is a joint secretary of BioClues and also an assistant professor based in Chile. And Sugnata Gure is an assistant professor and group leader at Lovely Professional University. He's a cancer geneticist. And myself, I'm the founder of BioClues and the principal scientist at Amrita University. So the very first few things I just thought, we'll give you a kind of a gist. I apologize, some of these particular slides were shared through our other talks earlier. So in fact, COVID-19 has had its environmental impact. In fact, not only taking in cognizance of how we have led the lives of the last 20 months or so, but the point is also going, more organic. It was sometime at some point of time believed that, we are trying to come back to normalcy, taking away the pollution and taking up, the organic chemistry into consideration to the environment. Staying at home, probably, we might have guzzled, wonderful freedom, spending time with the family and all those kind of things. In a country like India, where we have, more than 1 billion population, looking into the streets, looking into the kind of noise levels, probably, we might have, cleaned a lot of, stuff with that. But there has been a growing concern, with rapid growth of this pandemic. And one of the major concerns that I could really narrate down is the kind of the big data challenge that we are currently looking into. When this COVID-19 pandemic came into picture, Van Bevel et al. In 2020, as early as March 2020, published a wonderful article in Nature that are we at a confluence of leadership and social context? Are we really taking up a kind of a threat as a kind of a perception? Are we really emotionally driven with a lot of other things about our jobs, about our family condition, about the derailing income, about the kind of what people might really think, the kind of stress factors, the kind of mental conditions, and the kind of how we really coming up with the coping approaches, and also the kind of infodemic kind of things that Vladimir has wonderfully inserted. So there are a lot of things that Van Bevel et al. As one itself. So having said this, many of us, including me, have been, guzzling between these two syndromes. One is what we call it as an imposture syndrome, and the other one is what we call it as, Dunning-Kruger syndrome. So for those who are named with these two syndromes, I'm sure many of you have witnessed this. An imposter syndrome in a scientific career largely implies that we doubt about our own capabilities. Wherewithal we stand. Are we really concerned about are we really, delaying our science? Are we really, trying to, transform our science, to a kind of a reality? Are we really putting our research into a kind of a translational practice? So this is a kind of a syndrome, that everyone would really get into, when the world, stops with, such kind of pandemics. The other way around with Dunning-Kruger syndrome is something called, we become overconfident at some point of time. Maybe, if you're publishing too much, or maybe, if you think, that someone praises us, that, that we are trying to do, some kind of a good things, we probably think, that we know everything. So this is what, it is wonderfully exploited. The very first things that have already been predicted. So in 2020 April, when this pandemic came into picture, we were at some point of time, Mount Stupid. This is where, the Dunning-Kruger syndrome, we felt that maybe, by the summer, that creeps in, by May or June, the virus, would fade away. But that has not really happened. And if you see in 2021 March, again, which is sometime a kind of a peak summer in some parts of India, we have seen a lot of fatalities now in Indian conditions. So I believe, or we believe, that by December 2021, hopefully, if we think that, we have gone through this Dunning-Kruger syndrome and imposter syndrome during the last 2 years, maybe by December 2021, we will, come back to normalcy, looking into these kind of, conditions. And this was a slide, that also came into picture wonderfully as early as May 2020 by Malish et al. I don't know how many of you want to get a kind of a good take-home message here, but a wild guess, would be here is about an absolute, gender imbalance. As you could see, this poor lady is taking the entire burden on her shoulders, where the men are really taking forward the small viroids or whatever that you may call so. But this lady is really helpless. She's really left behind. So during this panel discussion, we also would like to take this gender balance issue as well, and then I think we have proposed earlier that the 3 C's play a very important role: collaboration, convergence, and consistency. And that's where we can probably keep our human activity intact. So this is where together we try to propose. So we have all the distinguished panelists here. We have 4 emeritus professors, 3 elite scientists who have published a lot on COVID. 2 early career researchers and 3 mid-career researchers and 2 senior scientists who have really published a lot on COVID. And I'm sure the next 40 minutes or so we'll have this wonderful panel discussion. So the very first question probably I would like to request Professors Vladimir and P.K. Gupta is, are we really saying that that COVID is a kind of a blessing in disguise? Have we really learned those kind of lessons? Can we say that COVID has been a kind of a blessing in disguise for scientists sitting at home, working together, and probably publishing some kind of good science, or has it been a kind of a bane? So please, sirs, to start with, maybe Professor P.K. Gupta, please, sir. Yeah. Can you hear me? Subbu Anil? Yes, sir?
Yeah, Prash, you're audible. Yeah, it's clear.
Yeah, is Professor P.K. Gupta on? Yeah.
Sir, please, P.K. Gupta, sir, please unmute yourself. Guruji? Yeah, please unmute yourself. Guruji, on the right side corner, you have a, yeah. Meanwhile, Vladimir?
Yeah.
Okay.
Yeah. Vladimir Proikov, sir, please, yeah. Do you have any quick say on this? Yeah.
There may be some technical issues. Yeah, can we call on Andrew to answer and Urmila ma'am?
Sure. Yeah, I think it's sort of a mixed situation. So, and you did mention it as part of the presentation, that you did. We do see very different types of effects depending on what stage of your career you're in. And I saw, for example, very negative effects for several early career researchers or students that were doing their PhD and were affected. For other types of researchers, time away from university and from endless meetings and having a lighter load from an admin perspective was actually quite useful to have the time, allocate the time and the piece to actually do some writing, write some interesting papers and get published. But we do see very different types of realities. And of course, there's an extra burden for women in general and for other types of researchers that were heavily affected, that had small kids and had to stay at home and take care of them, for example. So it's very different types of realities, and I'm not really sure how we're going to handle it in the future. How are we going to differentiate people that were affected differently?
Right. Yeah, wonderfully inserted, Andrea. Yeah, thank you so much. Urmila ma'am, are you around? Please, if you can have a quick say. Yeah.
Yeah. So hello everyone. Please bear with me for a bad throat because of some allergy. So COVID-19, have had multiple impacts on various walks of life, as we have been discussing. Being an optimist, I would first like to concentrate on some of the positive, impacts, and then move on to the challenges that COVID-19 posed in front of us. So first and foremost, I feel the biggest impact is that the COVID-19 brought the scientific community, regardless of geographic boundaries, together. It also brought some of the competing industries together to address some of the pressing issues. Um, COVID-19 emphasized enough, and everyone, including the policymakers, now understand that research and development needs to be supported, needs to be funded, and all basic applied, translational, computational, all walks of studies. They need to be supported as part of the as part of dealing with the existing pandemic, but also as part of the pandemic preparedness in general. COVID, I feel one of the important effect is also that it fueled interdisciplinary research. Virus, virology, of course, but and all basic and applied sciences included. I feel computational biology, chemoinformatics, virus bioinformatics played a role in facilitating global collaboration. Today we have, resources like GISAID, which shared the genomic data runtime. And in fact, yesterday, GISAID announced that they crossed 50 million genome sequences of SARS-CoV-2 isolates in the public domain. And it's a humongous task which needs to be applauded, appreciated, and it has also helped many of us, to work on different aspects of SARS-CoV-2 biology to translation. For the first time, it happened that COVID-19 papers were made available free, regardless of APC by all the publishers, which I feel is also a remarkable and interesting fallout of COVID-19. COVID-19 established new normals, with respect to how we teach, how we learn, how we collaborate, how do we work. And it has impacted all walks of life. There is a huge impact on mental health. I recollect and remember having conversation with some of our students in the remote areas of India, where they missed classes, online classes, because of lack of network, lack of electricity, and then we had to come up with different measures to have the lectures recorded, share them, allow them to learn at their own pace. I feel the there has been a huge transition with respect to online teaching and learning in India because of COVID-19, and which I feel again is a step forward. We need to strengthen this infrastructure and take it ahead. Of course, some of us, especially the women scientists with young children, managing home, managing young children, and yet to sort of carry out research was a big challenge for many of us. And Prash, I recollect a wonderful session that BioClues had organized with respect to challenges faced by women, that you had taken initiative to organize this. So there are many aspects, and we can continue to discuss these. So maybe I would like to also hear views of other panelists, and maybe come back for a quick one or two comments. Thank you, and over to you, Pranav.
Wonderfully insighted. Thank you so much, ma'am, amid your sore throat. We wish you a speedy recovery. We'll get back to you on that.
Thank you.
Subbu, would you mind, taking the next point on infodemic? Maybe, Jayaraman sir as a machine learner and Narahari Shastri sir as the director of CSAR, he can probably give those wonderful points. Yeah.
Yeah, sure. Brush, first of all, I would like to welcome Vibhatanan ma'am to coming over here. Yeah, thank you so much. And as we are in the era of COVID-19, we would like to ask something based on Infomedic, an excessive amount of information about a problem typically with unreliable, it spreads a lot of data, and it may to make a solution also to become very difficult at some times. How did you cope up with this? That is one thing I would like to ask first with Jayaraman sir, then Narahari sir, and Vibhatanan ma'am. A quick 30 to 45 seconds each, please. Thank you. I think they are not able to reach out. Vibhatanan ma'am, yeah, please. Can you unmute your microphone? And then
Yeah. Thank you for involving me for this panel. And sorry, I joined later because I had a class from 10 to 11. And I was thinking to speak on something else. But as you have raised a question about the data, and that how are we handling the excessive data which has come? I think this was your question.
Yes.
That how are we handling the excessive data which has come? That is what you want an answer from me?
Yes, ma'am, please.
Okay, so fine. So say as far as it concerned that I myself earlier don't used to work on the data, but in terms of are you talking about the data from COVID, or you're talking about the general COVID data?
With respect to COVID, ma'am, very specific to
Okay.
COVID.
So in respect to the COVID, say there are a few things which I would like to mention to the students if they are also a participant over here. That there is one good thing happened, that if you go on the website of the WHO, and there are several other say website, that is ICMR, that is our Indian website, that they have made a lot of data available to us, and those data, we should fetch the data from those authorized website, because there are so many publications have come in last 1 year, or you can say 1.5 years, and everybody is trying to do something in COVID. And sometimes those kind of those data are misleading also. So my first of all, I would like to say that please do not, because I have seen personally that there are so much data has come into the which has not been properly reviewed. So until unless it is a peer reviewed data, please don't go through that data. And specifically because I work on drug discovery and drug development, I would like to say that each and every molecule, everybody is saying that it can be repositioned. So please look into that kind of a data before repositioning it, or before going ahead, because wet lab people are looking at the computational data and trying to go ahead with the wet lab strategies, and trying to develop the wet lab strategies. So please look into the data until unless it is a target specific data, and you have to see that there is no cross reaction with these molecule. If these molecules are really if they are against inflammation, if they are against the similar kind of homologous protein and acting on the homologous proteins in the different say a different organism, or maybe different microbiome, say microorganism, maybe bacteria. So then only you should try to look into that. And at least when I started working in one of the project in COVID of synthesizing the nucleoside analogs, we have done a lot of research, and what we did actually, that whatever was the computational data available to us, we just recycled it at our end also. We try to do it, recheck it. Recycle means I did my own computational analysis, whatever knowledge I have, or I made my own research questions first and gave it to some of my chem informatics and bioinformatics friends and collaborators, and then asked them to like for example, I was given 500 molecules. So I just gone through those 500 molecules on my own, and then looked into that, are they really fulfilling the medicinal chemistry requirement, because there is a drug likeliness properties are already identified. So please look into that data, and then I reached from 500 to 21 molecule, and out of those 21 molecule, only say 3 have worked in my hand. So one has to be very say one has to look into this, because this 1.5 year have along with the anxiety, we have been faced with several data which are not up to the mark. So please pass through it several parameters before using it. Thank you very much. I'll come back if there will be time for anything.
Yeah, thank you so much, ma'am. Thank you. That's wonderful explanation that most of the young students would have really understood, and the young scientists here. Yeah, I would like to ask Jayaraman sir, if you're around here, like what was like a machine learning approach?
Yeah. Yes, sir. Are you able to hear me now? Yeah.
Yeah, it's clear, sir. Please.
Okay. So before coming to the data, which is my main bread and butter, I must first of all tell the impact on the research due to COVID-19 has been very dramatically painful. And this impact is going to remain long term, especially in experimental research. The pandemic has curtailed the experimental research most and have forced them to modify their research plans and goals. Project scope, aim, and procedures and fundings have been affected. So long-term project goals and aspirations have been shelved, and plan A to plan Z have been thought of and formulated. Under these conditions, a scientist needs good support from institution, departments, and essentially a successful scientist under today's condition has good mental stability and knows the tricks of the trade. He also knows which data is good, which data is bad. The serious scientist who is never aware of his surroundings, disordered in every possible ways, needs to change, gear up his communication mechanisms, and he must know how to separate garbage from good data. And what I would like to say is, see, this gives an opportunity to the experimental scientists who always fight with their data scientists or theoretician to strengthen their theoretical knowledge and then become algorithmic and have to add their skills and algorithms and for a change to be friendly with the theoreticians. And they must be able to better interpret the current available data in a very optimal and way, and they must know how to plan to remove what we call it as noise from data. So as a data scientist perspective, I would like to help all scientists who are around and who want me to help them to differentiate garbage from data. Thanks. Yeah.
Thank you, sir. Subbu, Andrew has raised his hand, but before which, P.K. Gupta sir has a comment. Yeah, I guess. Yeah.
Yeah. Guruji, please. Guruji, please unmute your mic.
Yes, are you asking me to?
Yes, yes, sir. Yeah, please, sir.
Hey, Prashant, thanks. Thank you very much for inviting me to this panel discussion. You see, most of the people who are attending this conference and the panel discussion may not be knowing me. I am not a computational biologist, and computational biology or bioinformatics has been a subject which is a sort of secondary interest of mine. And during the last 5 to 10 years, and due to my association with Prashant, we have published a series of papers in the area of bioinformatics, which particularly included identification of novel genes in wheat in various areas of our interest. For example, we have been working in wheat on the biotic and abiotic stresses. Therefore, a number of gene families have been studied by us during this period. So that is, of course, before COVID-19 started. But COVID-19, if I speak my personal experience, it has been a blessing in disguise, as Prashant has mentioned as one of the first. It has been a blessing in disguise because it gave us more time to study at home and write. So my writing ability, not really ability, but output. My writing output has increased during this COVID-19. I had regular meetings with my students, online meetings, and that increased our productivity, and we published a series of papers. I would have not published so much if the COVID-19 were not there. But one of the problems this COVID-19 we had was experimental activity. You see, the wet lab experiments, which we always follow, the bioinformatics analysis, those wet lab experiments had some difficulties. Some of the students could go to the molecular biology lab and conduct wet lab experiments also to confirm the findings of the bioinformatics. But by and large, the wet lab experiments definitely suffered during this period. And also field activities also suffered because the students and the teachers, they were afraid of going to the field. And I remember last year, the students and the teachers were not even prepared to harvest the crop. But then I took special permission from the Vice Chancellor and the other important people in the university to allow at least 1 or 2 students to go and harvest the crop. So we could therefore harvest the crop. Otherwise, it would have been irreparable damage to our research activities. And then, of course, we could design wet lab experiments during this period, which we would like to undertake now. That is one important outcome of this period, because the wet lab experiments which we could not undertake during this period, we can undertake them now, because almost all students have started going to the lab and conduct wet lab experiments now. Coming to the publications on on COVID-19 appeared. Almost every day there were a few papers on COVID-19. And after initial interest, I stopped reading these papers because it was almost impossible to read the literature which was appearing at such a fast rate. Some of my students wrote articles on COVID-19. For example, Manoj Prasad in Delhi, he wrote 5 or 6 articles on COVID-19 and he kept on sending those to me but I also wanted to write an article on COVID-19, then I gave up because so much was being written and it was almost impossible to be update and write something which will be really up to date and will be useful for the readers. So I did not write anything on COVID-19, although I am very fond of writing popular articles on the emerging subjects.
Yeah, thank you, sir. Thank you so much.
Another area of COVID-19 which interested me, but I did not write about it, is the plant-derived products which could be used during COVID-19. There are a number of products which have been identified during this period which could be used for development of vaccines and drugs for COVID-19. That is one area which interested me, but I did not write anything about it. Another area, Prashant, do you have time? Another 1 or 2 minutes?
Yeah, please. 1 minute, sir, please. Yeah. Thank you.
So, the teaching activities also during COVID-19 was continued. I took classes, taught a course to my MSc and MPhil and PhD students during this period. There are too many webinars and you see, it was almost impossible to attend all of them. You see, all academies, Indian Academy of Sciences and National Academy of Sciences, all the academies arranged lectures, which it was impossible to cover all of them. So I had to control myself. Otherwise, I am very fond of attending all these webinars, but it was almost impossible to attend all of them. Too many of them, because it did not cost anything. There were so many conferences, webinars, anything. So everybody was organizing conferences and webinar lectures during this period. That was something which sometimes me, because too many of them are there and you feel like attending many of them, but because of shortage of time, you cannot. I think that's all I would like to say.
Thank you, sir. Thank you so much. That's indeed wonderful. We'll definitely get back to you, sir, on this. Moving slightly further, I mean, from infodemic that Subhu has wonderfully raised. I was just wondering if Aparna and Himakshi has a say on slightly a different topic, which is something on whether or not we learned any lessons in combating pandemics from home to lab. I mean, as early to mid-career scientists from 2 different continents, how did you guzzle this right from gender balance point of view? So Aparna and Himakshi, please. Yeah.
Thank you, Prash. Thank you for including me to the panel discussion. I was listening to all. For me personally, COVID has become a blessing in disguise. Initially, when the COVID started, I was really shaken up a little. And living from like 17,000 kilometers away from home, I had a lot of doubts. Should I continue being here?
Hello, sir. Prash, sir. Hello, sir. Himakshi here. Actually, there was some connection problem. So I'm joining from Nagamani's laptop.
Yeah, please, fine. Thank you.
Okay. Can I start or after Aparna ma'am I can speak? No problem, sir.
Yeah, Himakshi, like right after Aparna. Thank you.
Sure, sir. Sure.
Okay. So yes, I really had doubts. Should I continue being here or not? But then, as Prash was asking the question, how I manage this thing from home to lab. That was really I was reading an article actually all this time. One article that was showing that COVID-19 has actually increased the humanity, solidarity, and resilience in all of us. Being scientific community, we are like helping one another more. I personally have felt it has been quite easy to be in touch with the scientific community during the COVID-19 pandemic than it was earlier. So personally, I have been in touch with many of the scientists around the world that I personally did not put that much effort before COVID-19? Probably there was a lot of reason. Probably COVID-19 make us really think from home, how we can do extra things being from home. So initial 2 to 3 months, I was taking a gap, but then I had a project and the project need to continue. We cannot stop the science for one big pandemic. The science must continue. So I took special permission. I was vaccinated and I started continuing my project. And that time I really understood that my students, the project assistant, all really supported my idea. So that time was, I really had doubt if people will support my idea of continuing the lab work or not. But I have taken a big decision. That time there was in Chile, all the projects government make a continuation of 1 more year. So I had the opportunity to make my project 3 year to 4th year for COVID-19, but I choose not opting that because I worked during that year also of COVID-19. But personally, I felt that I had more resilience during that time. Earlier, I used to feel like, oh, the Fridays always used to be tiring, probably for all of us. But in COVID-19, probably we enjoy going to the lab because we understand how important it is, the normal lifestyle, when we really lose it. So personally, for me, it was initially it was challenging.
I have to stop you here. I'm really sorry for this, since we have got so many other points. Yes. Thank you so much, Aparna, for those wonderful points. Esther, do you have any quick say, please? We have other hands raised. Esther and Imakshi, please. Yeah.
Yes, sir. Yes, sir. Yes, sir.
Largely, towards the gender balance, point that we would like you, to comment upon, please, as early career scientists. Yeah, please.
Sir, actually, thank you, sir. Thank you so much. Thank you, BioClues, InBix, and Professor Shastri for giving me opportunity to speak in such a big platform. Sir, I would like to share my personal experience. I think you have mentioned in your 4th point about the students' mental state and the future pandemics. Yeah. So we know that this pandemics has disrupted so many things, but I feel that it is also a kind of blessing, which has created a lot of motivations, innovations, and creativity among the researchers, those who are also already doing research, and also the budding researchers like me, who I have never thought that I will do any kind of research in viral informatics. And also what I feel that when during this pandemic, I got a lot of phone calls from my juniors, the school juniors, those who are doing BSc dissertation and MSc dissertation. Those days, they got stuck in the home and they called me and they were asking me, like, you are a bioinformatician. By sitting in the home, how we can do research? So I was very happy that this pandemic has created a lot of curiosity among the students to do viral research. So I'm very happy, and this pandemic in our lab specifically, due to the motivation of Shastri sir, we utilized our laptops in a best possible way, because we started learning. We have collected a lot of literature, and we also started doing some research on COVID. Currently, we have prepared the manuscript 2-3 manuscripts which are under preparation, and yeah. So I feel the student community is got a lot of curiosity to learn more about viral informatics and virology. Thank you, sir.
Thank you. Thank you so much, Esther. So Himakshi, do you have any quick points? Yeah.
It was Himakshi, sir.
Oh, sorry,
yes.
Himakshi. Sir, do you have any quick points, please? Please stick to only 30 seconds. I'm sorry for this. Yeah.
Hello, sir.
Yeah, please. Yeah.
Okay, sir. Okay, thank you so much for giving me this opportunity. Now, as a very young research scholar, it was witnessing such a major shift in the scientific community during the pandemic. It was really overwhelming for all the research scholars. And then we can definitely reflect on all the opportunities and challenges that the COVID-19 has brought for us. It was definitely not the same situation for all the students, because we were all in different situation in terms of facilities, in terms of involvement and accessibility. And as mentioned by Professor J. Raman, it was also seen that various fundings and projects were on hold. Nevertheless, almost all the researchers were continued their research work during the pandemic. And so we we should use this opportunity given to us, not only just for publishing papers also, but we should put a focus on contributing our knowledge for the betterment of the community, or bringing up or coming up with a solution in developing vaccines, and also this vaccines. And also we, as a student from a computational background, we can definitely work on analyzing or screening for potential leads that can be used for treatment of COVID. But as mentioned by Vibadantan ma'am, we should also be very careful in using the available data that is online. We should always go for authorized data, like mentioned for in ICMR or WHO database. So as research scholars, we should definitely continue to work, and this can also be achieved by collaboration with among one another, both nationally and internationally. So we as a research scholar in both experimental and computational field, I believe that we have important role to play, and in understanding the mechanism of the disease, and also in identifying a treatment for the COVID-19. Thank you very much.
Yeah. Thank you so much, Esther. Yeah, moving further, I'm not sure if Anshu Bhardwaj is here. We missed her. So I think the very final point before we come to the Q&A session with the participants as well, the mental condition has been has really turned into some kind of serious force. We have had post-COVID syndromes. We have had learned from infodemic to infobesity, which means we are too much over a lot of information that was really going awry as well sometime. And we have had post-COVID syndromes where we are trying to cope up with our health conditions. And there is a new word that has really come up towards something called long COVID sequelae. That's because of the non-adherence to the seroprevalence conditions in those patients who have been immunized. So maybe with starting with probably with Urmila ma'am. So since you also raised your hands, and maybe Andre, can you slightly discuss on this particular, the last point on the post-COVID syndrome and the long COVID sequelae, and how we could come back with?
Okay, thank you. Before I address that, I raised my hand before. I just wanted to add something to the infodemic aspect. And it's something that I think is really important that we miss as part of the discussion, which is that the current pandemic revealed that there's a serious issue with lack of scientific literacy. This is something that we see with the general public, but also with the media. And I think it's very important for us as researchers to try and bridge that gap and make sure that media and journalists are able to understand what the scientific process is like, what peer review is all about, because we see this continuous misreporting of results and information that is passed across and doesn't really match expectations. So it just misleads people. And I think that's an important aspect, an important point to make, and it's something for us to keep in mind. Regarding the mental condition of students and future pandemics, I think that students are a lot more resilient nowadays, and staff members also. I think that it was a steep learning curve how to adapt to the current situation. We will have some issues in the future. Luckily enough, in my specific case, I'm working in Macau, so the part of connected to mainland China. The situation in Macau was very different from the rest of the world. We had very limited number of cases, so we're more or less off the hook there. So I can consider myself quite lucky not to be facing this type of issues over here in this part of the world, which is yet another important aspect, how the realities across the globe can be very, very different, and the effects locally can be very different.
Yeah, so moving further with Urmila ma'am. Yeah, well said, Andre. Thank you so much.
Yeah, ma'am, please. Yeah, please unmute yourself, ma'am.
Very well said, Andre. And I also raised my hand to touch upon some of the aspects of infodemic due to this pandemic. A lot of misinformation regarding the virus, its origin, emergence, floated in social media. There were references to past mentions of coronavirus, the movies like Contagion, and so many other aspects. And there was a lot of confusion amongst the mind of people with respect to treatment options, vaccines, availability of oxygen, and so many other things. And that really impacted the society at large. Still there are anti-vaxxers who are talking about, and then there were so many releases from pseudo-science preachers to confuse the public opinion at large. And therefore, I feel that we as a community of scientists and BioClues as an organization need to step forward to combat infodemic in a systematic and chartered fashion. And I think overall there is a need and onus on us to sort of work towards make conscious efforts towards improving the science quotient of the society, so that we can effectively deal with any challenges pertaining to infectious disease viruses or other diseases. So thank you, Prash, and thank you all the panelists for valuable inputs. And before I really the other aspect of infodemic, the aspect that I mentioned was a negative.
If you don't mind, please restrict to 30 seconds. Yeah, please.
Sure.
Sorry.
Sure. No problem. I understand.
No, please. No, you can say, ma'am. You can say up to 30 seconds here, please.
Okay. So I thought you wanted me to stop. Okay, so I mentioned negative aspect of infodemic, as the connotation, but information and how to deal with information and data to translate into knowledge and wisdom is a very different aspect that this pandemic has put forward, and Dr. Jayaraman and few others have very well mentioned that there is a need for all of us to understand, methods for curation before we use the data and learn data analytics methods, use them with responsibility. This is with respect to, the students applying these methods for the discoveries. And apart from this, I would just like to mention that we have been working on coronavirus, and one of our recent papers this week, when we curated data, we realized that India sequenced about 0.14% of SARS-CoV-2 isolates from the confirmed cases, and India contributed about 1.58% of complete genome sequence globally. We have tried to put some infographic and some evolutionary analysis together in a manuscript in the journal Viruses. So I would like you to have a look at it. Thank you.
Thank you. Thank you so much, ma'am. Sibu, would you like to take this question to others, please? Yeah. Quickly. Yeah. 30 seconds.
I would like to add like Anshu Bharadwaj, any quick comments? No.
Yeah, hi everyone. Thanks for having me here. I completely agree with what the other panelists have raised, the concerns about infodemic and concerns about communication of science to a wider audience. I think that can play a major role in addressing future pandemics. That's a very, very critical point. One thing that we did as a very novel way of collaboration is we were trying to address the infodemic with respect to drugs or inhibitors proposed for SARS-CoV-2. And as a very distributed team, we could curate resources and structure of individual compounds for the benefit of the wider scientific community, so that they can understand what kind of chemical space is being probed. And you can look at this resource as Savior, which is available online. But one thing I must say, that the researchers and the young students we work with are very resilient. We were doing COVID testing initially last year, right after the lockdown, where vaccine was not in sight. nobody really understood the impact of the infections, but our students and our faculty members were receiving these samples from hospital on a daily basis. I think that shows the curiosity and the unselfish nature of the research community per se of our healthcare workers, which was very, people do recognize that. There, as some of the students highlighted, that there was an interest in data sciences, which again is a very positive thing to take forward. But we need to make sure that not just as a research community, we need to look at the impact of COVID-19 throughout all the spectrum. And if you look at the McKinsey's recent report, they are clearly highlighting that India is going to have a problem in job transition in next 10 years, which means that we should prepare ourselves for doing high-end skill development as we move forward, if we really need to make sure that the impact of this and the future pandemics can be mitigated. So yeah, that's all from my side.
Yeah, there is one question, like a personal question, like apart from the questions asked in the list. As we're seeing like COVID-19 impact on research, a lot of lessons has been learned on COVID-19 research. There is something which is pressing now, like the implications of doing pediatric research. So that might be one of the important thing. So I would like to ask any of the panel member a quick 30 to 40 seconds so that you can talk something on the implications of pediatric research, which might play an important role in the coming days to come. So with permission of Prash, I would like to ask all the panel members 30 seconds each, please.
Yeah, please. Please, of course.
Yeah, Andre, Vibha Tandon, Urmila ma'am, Aparna, Jayaraman sir, Anshu Bharadwaj. Yeah, anyone please.
Actually, Prashant, I didn't understand what you said. Can you please tell me again? Implications of?
Ma'am, implications of pediatric research. Pediatric research.
Pediatric research. Okay. So on children, you're talking about that?
Yes, ma'am. Yes, ma'am. Yeah, thank you.
Okay, okay. So I had a, so in terms of the implications on pediatric research on COVID, basically, I think you're asking this. Related to the COVID?
Yes, ma'am. Yes, ma'am. Yeah, thank you.
Okay. So the most important thing I would like to tell you that I think in terms of not only research, in general also kids were largely got affected by COVID, and while we were suffering, we just I would say that we have forgotten about the about the kids. And in terms of the pediatric research on COVID, like I had a collaboration with the Safdarjung Hospital about the COVID conditions of the COVID infected kids. So I would like to tell you that there was a separate research call was given by the ICMR on the how to deal with the children with the pediatric infection, with the COVID infection. And there was a it has been found that the most of the kids which were infected with the COVID, they were having very mild infection, and they were getting themselves being being cured, and or maybe, but there was a hospitalization, and hospitalization in Delhi was almost 5 to 10% of the rest of. And in terms of the research, they have found that there were certain, just like the adults, there were certain comorbid conditions where also the similar kind of conditions were found in the, in the kids, that they have been found, but the cycle of the COVID was little different. So there was a research occurred, and there was a whole funding was created by the ICMR, which I cannot just explain in 30 seconds over here, but there was a whole call by the COVID call was given by the ICMR, and in which they have shown that that it was the whole say, I would say, the cycle of the virus in the kids were little different as it was found in the adults, and as we say that after 5 days there was a kind of a cytokine inflammation was there, but in here they have found that it was not after 5 days, it was around, say, after certain more days. So even the treatment modalities were also different in the kids as compared to the adult people.
Sugu sir, can I come in?
Yeah, please, sir. Thank you.
Yeah.
Jairam sir, please.
See, the mental conditions of these kids have drastically been affected. There's no doubt about it. I myself have seen their traits have changed dynamically from openness to neuroticism. More than research, I think the parents and teachers need to understand and give them additional space. Yeah. Thanks.
Thank you, sir. Andre, any comment on this?
Well, I don't really work with clinical aspects, so I'd like to focus on something slightly different. So deviate a little bit from your question. I think that one of the things that COVID has highlighted is the importance of fundamental science and fundamental research. And it's important to notice that all of this issue with the current coronavirus pandemic started with an area of research that was more or less obscure and wasn't particularly well funded. And so I do have my concerns because on one side it has highlighted that fields that are currently not necessarily particularly relevant or particularly highlighted could be very important in the future. So that's one of the things. Well, the other thing that I worry about is the economic crisis that we're anticipating as a result of the pandemic. How will it translate into research funding? Because the normal thing that you see is cuts will tend to affect fundamental sciences, which is exactly the place where we shouldn't be cutting.
Yeah, thank you. That's wonderful to know. Yeah, Urmila ma'am, any quick comments?
Yeah. So per se, I don't work with clinical research and therefore unable to comment on the question pertaining to pediatric research. But I feel pandemic has impacted the whole social fabric in more than one way, and the solutions would come from the scientists from different walks of life, from social science to biological sciences to educationists. Everybody needs to come together in order to mitigate the challenges and the impact the COVID-19 have had on different strata and different sectors of society, and children are included. It's a huge problem that I mean, even to articulate in 30 seconds is very, very difficult.
Yeah, thank you.
Yeah, thank you.
Anshu?
Yeah, so I personally don't work with pediatric research in COVID, but I can tell you from experience in tuberculosis, there has been challenges in terms of devising methods which are not very child friendly in terms of either prescription of drugs or the way we do diagnosis. So I think if we really want to make it easier for them, we need to follow up with the diagnosis and the treatment options. They should be made, the formulations and the devices should be made in a fashion that they are more kid friendly and not something which is as hard as anti-TB drugs is. That's the only exposure I have in terms of clinical research or the fag end of what happens with kids when you are giving them vaccines or treating them with different drug regimens.
Yeah, thank you, Anshu. Yeah, Prash, last call. Yeah, you can take over, please.
Thank you so much, everyone. This has been a wonderful thing. I know, with so many panelists, sticking to the time is, very crucial as well. But I'll just try to summarize, the kind of wonderful points that you all have raised. I think, COVID is certainly kind of a blessing in disguise, no doubt about that. From the student-centric point of view, yes, there are certain hard lessons learned. Likewise, from the faculty point of view as well, there are certain lessons, but I think the kind of freedom that the faculty might have really guzzled sitting at home, from the students' point of view, the kind of impostures and the kind of Dunning-Kruger's that they have undergone, I'm sure that it would set a kind of a precedent in the near future. Say cometh COVID-23 or cometh COVID-24, even in the near future, even if we have some kind of COVID-like situation, I'm sure we are going to we need not really face the brunt, but we'll definitely be prepared for that. But as researchers, one of the striking forces behind COVID is, of course, the kind of publications. I think many have really published, but there is actually a very sad point to this wonderful story as well. The kind of hypotheses and perspectives that researchers have published over the last 20 months might really be a farce. Just keeping into consideration, because many of these hypotheses is really going to be false or farce, because the so-called seroprevalence story post-vaccination that we were talking about is no longer the story that we were talking at some point of time. When the COVID pandemic came into picture, we were talking about the herd immunity, where 60% of the people might really get into the curve of that kind of herd immunization. Even above 75 or 80% today, they're no longer talking about the herd immunity. And the third and the most important point that we have here is what happens to the kind of hypothesis and perspective articles that we have been publishing. Do we have room or enough time and energy to convert that kind of hypothesis or theory or perspectives and put it into practice and put it into the lab? Are we really prepared? That's kind of challenge that we really need to look into this amid all the kind of imposture and Dunning-Kruger syndrome that we need to talk about. And I think the onus is on the mid-career scientists, like probably many of us, who needs to ensure that the baton is put up very straight onto the young student community. I know the kind of problems that the students really face. I've been really privileged to have about 6 or 7 PhD fellows with me currently. Their conditions is absolutely pretty bad. They have been piggybacking between these two syndromes, and of course I was also going through that. But yes, the best thing that we can combat is have a healthy diet. If I were you, I think it might sound pretty stupid. Have one fruit before a healthy meal. That will keep your biological clock intact. Please don't have a fruit after your meal. That will really make your things probably pretty upside down. And of course have yoga. And the third and most important thing is keep smiling. We have wonderful this thing. Why not keep smiling? And now when we say keep smiling, Subbu and I would like to take you to a quick round of all smiling faces. So it's time you give a thumping round of applause to all the panelists and the participants here, please. Thank you so much. So I don't know. I mean, in MS Teams, I don't know. We cannot have photograph in one picture. I don't know. Maybe Nagamani or Subbu can help. All of you, please switch on your camera, please. It's a wonderful opportunity for all of us, to have a photograph with all these wonderful panelists. Yeah. Viday, Nagamani, and Imakshi, can we take in one window all the photographs in MS Teams?
Yeah, Prash, there is an option called as Together Mode. Then there is scenic beauty that comes with all the members. Yeah, I think I am doing that part. Yeah, please.
You want to post it on your LinkedIn profile or something?
Yeah, I mean, not only that, to cherish, this wonderful thing, we seldom get an opportunity to get a photograph with this.
Yeah. I understand.
Yeah, please.
Yeah, please.
So I'll restrict myself only in smiling. So we will take the photograph. Yeah.
Yeah, I've taken one.
So you have to count 3, 2, 1. So go, please. Thank you.
Yeah, please. Yeah, thank you so much. Just give me 1 second. Yeah, one more quick 30 seconds. Yeah, please. Yeah, please smile, please. Thank you. Yeah, we are in auditorium sitting together. So that's how.
Thank you so much, Sigoo.
Thank you so much.
Very much. You're all revered speakers, for obliging to our request and giving your opinion. We'll stay in touch. Thank you. Yeah, thank you so much.
I just sent some information about the pediatric research, so you people can utilize it. And there is a wonderful work has been done which I could not explain, so I just pasted it, so you can put it in your report.
Ma'am, I have already taken note of it. We will keep in the report, certainly. Thank you so much, ma'am. Thank you. So kind of you.
Yeah, thank you. Yeah, thank you so much.
Thank you.
Yeah, Anil, please take over now. It's like, yeah. Thank you, Anil. Thank you, Prash. Thank you all the panelists once again. Thank you so much.
Thank you, sir.
Anil, you are not audible. I think you need to unmute yourself.
Yeah. Can you hear me?
It's clear, Anil, now.
Can you hear me?
Yeah, it's
fine.
clear.
Thank you. Thank you for all the panelists. We had a wonderful panel discussion. Thanks for giving your opinions. Thank you, one and all. And once again, I thank the rapporteurs, Prashant and Sugnakar, for taking care of this. We will meet again at 12 o'clock sharp. Thank you. Thank you, one and all.
Yeah, maybe I would like to request Uday and Nagamani over there to play light music, maybe SMS music, so that the participants can stay intact. Yeah, thank you so much.
Welcome back. Welcome back to everyone. After good keynote one session and panel one discussion, now we are moving to session one, and the topic is advances in biotechnology. This session would be chaired by Professor Jayaraman sir and Dr. Harpreet Singh. Jayaraman sir, please move ahead with Niaz Ahmed sir, because Holger sir is not in the group. Once he joins, we'll move. Please take the session. Jayaraman sir, can you hear me? Harpreet sir, can you hear me?
Yes.
Yeah. Welcome.
Yes, I can hear you.
Yeah, please chair
Welcome
this
to the.
session. Thank you.
Welcome to the first session, and I am Jayaraman Valadi, and my co-chair is Harpreet Singh. So I will ask Harpreet to start the session, and then I will take over after that. Yeah.
Thank you, Dr. Jayaraman, and a very good morning. And it's a transition between morning and noon, so a very good noon to all of you. And we have a wonderful morning, and with the wonderful keynote talks and a very exciting panel discussion. So after a short break, I think all will be feeling refreshed. And now let's start with the second part of the today's the day one of the InBix-21. So we are actually beginning with the technical sessions from now onwards, and the first technical session is on advances in biotechnology. And I, with my co-chair and very senior and respected Professor Jayaraman Valadi, will be chairing this session. So just to make you summarize that in this session, we are having three invited talks by Professor Holger Gokhale, Niaz Ahmed, and Andre Gomeres. And we also are having three oral talks. So I request all the speakers to be ready as per their turn. So there is a little change in the schedule. Instead of the first talk by Professor Holger Gokhale, he is on the way and he will join us very shortly. So we'll begin our talk with Dr. Niaz Ahmed. Professor Jayaraman, shall we begin?
Yeah.
Thank you. So before I invite Professor Niaz Ahmed to the desk, let me introduce him. So it's my actually privilege to introduce Professor Niaz Ahmed, who is actually a leader in the area of molecular infection epidemiology and public health microbiology. He is an experienced senior director of laboratories with a demonstrated history of working at the interface of epidemiology, genomics, and laboratory science. I think he's an ideal person for this conference because he's working as an interface of so many, technologies which are gearing up the world now. Skilled in innovation and management and capacity building of young scientists, Professor Ahmed is currently serving as a full professor at the University of Hyderabad. Earlier, he served as a chairperson of the Department of Biotechnology and Bioinformatics at University of Hyderabad. Professor Ahmed was also affiliated with the University of Malaya, Kuala Lumpur, Malaysia as a visiting professor of molecular biosciences at the Institute of Biological Sciences. He also served as an adjunct professor of the Academy of Scientific and Innovative Research, India. Professor Ahmed's main research interest is about how bacteria interact with humans and the environment in order to evolve fitness advantages such as multitude drug resistance and strain host specific virulence. Professor Ahmed's research work embodied in about 160 international publications remains highly cited and recognized, and you can see that he has an H index of 46. Consequently, Professor Ahmed was elected to the fellowships of the National Academy of Sciences of India, the Royal Society of Biology UK, the Royal Society of Chemistry UK, and the American Academy of Microbiology. Professor Ahmed has been awarded with many prestigious awards, but a notable one is the prestigious Shanti Saboo Bhadraga Prize. It's India's highest science award in 2016. National Bioscience Award for Career Development in 2011 and University of Hyderabad Chancellor's Award 2015 are two another awards which goes to his credit. I think it's really a wonderful opportunity for all of us to listen to a speaker who is highly awarded, highly accorded, and who has such an experience on interface of many different sciences which are ruling the world. So with these words, and on behalf of the entire organizing committee, on the behalf of my co-chair, Professor Jayaraman Valadi, I request Professor Niaz Ahmed to enlighten us through his talk. Thank you.
I would like to welcome you all for my presentation on the pathways of antimicrobial resistance through data sciences. Antimicrobial resistance is basically seen as a fitness advantage, an attribute of the virulence of pathogenic bacteria. And virulence in pathogenic bacteria has been evolving as a function of genome evolution. The genome evolution, for example, in the two dominant classes of pathogenic bacteria, which also cause acute or chronic infections, as the case may be. If we see mycobacteria as a model organism or as a model pathogen, they evolve through vertical genome reduction over a period of time, thereby leading to the emergence of specialist lineages. These lineages are destined to a particular host species and have a particular spectrum of virulence. The enteric bacteria, on the other hand, they evolve through lateral genome acquisition. That means they aggregate their genome over a period of time by resorting to horizontal gene transfer. And this horizontal gene transfer mediated aggregation of the genome imparts them a flexibility of functional diversity. So they, for example, over a period of time become more diverse because of this ability of genome aggregation. Whereas in mycobacteria, the genomes become more and more clonal. So mycobacterial genomes are clonal, meaning that they are clonally evolving and have a closed pan genome. Whereas the members of Enterobacteriaceae, for example, Helicobacter pylori, E. coli, Salmonella, Shigella, Campylobacter, these organisms have been evolving through horizontal gene transfer mechanisms, and their genomes are becoming more and more diverse. And as a result, their pan genome or their gene pool is open. So what happens when you have a closed pan genome of a given pathogen, such as Mycobacterium, then how to type the lineages of mycobacteria? So the closed pan genome doesn't mean that they do not have any pockets of diversity still left. They have a lot of diversity still available for the purposes of classification or individualization of the certain lineages of mycobacteria. For example, in the context of drug resistance, here what I show is that some of the lineages have more predilection for such as the Beijing genotype, for example, to acquire multiple drug resistance than other lineages such as the ancestral Indian type of lineages of mycobacteria. Whereas in case of the open pan gene pool, open pan genome, the enteric bacteria, as they evolve, the meaning that they have an open acumen for acquisition of novel genes. That means every new addition of a gene, of a genome in their present gene pool would impart availability of novel genes. So if you can see here, if you follow the pointer, the E. coli genomes, for example, each new genome is supposed to give new genes. These number of genes could be estimated by probability. So an estimate could be that each new genome in Enterobacteria family could give 40 new genes, for example. And genes impart them the flexibility to adopt. For example, here in Helicobacter pylori, you will see that the core has consolidated to 1266 genes, and all other flexible gene pool of this bacterium actually is related to strain-specific genes. So these strain-specific genes could impart these bacteria the survival advantages, for example, to cope up in the new host environment and the changing niches of the host, for example, and in order to fight the activated immune system, or in order to activate or in order to interact with the human genes. Some of these genes could be the genes that are the drivers of antimicrobial resistance, those genes which may have the regulatory capabilities, or some of these genes could be the methyl transferases, for example, as it has been worked out in this paper, that these methyl transferases could be capable of even regulating their host genome or the expression of the host genes. So it is possible for them to aggregate new lifestyle functions because of the flexibility or because of the affordability to aggregate new genes through their genome evolution. And these new genes could also add to their resistome, for example. They could be very well assisting these bacteria in evolving their resistant phenotypes. So the AMR, the evolutionary dynamics of antimicrobial resistance in bacteria is actually two-way driven. So it is bacterial genetic driven and environmentally driven. And that means that the MDR clones or superbugs, they evolve at a multiple interface, whereby the human activity as well as bacterial genetic activity and the activity in the environmental niches of these bacteria are actually the drivers of the shaping of a multiple drug resistance phenotype. Nevertheless, the AMR analysis, as it appears to be the data science related lecture, and if we talk about their evolutionary paradigms, their interaction with the human habitats and the environment, their own bacterial gene pools, all these contribute to a lot of functional data through the analysis of functional molecular infection epidemiology at the interfaces of the pathogen and host genomics and variation of the host pathogen responses. All these data plus the laboratory information management systems, the quality assurance systems, the biosafety and biosecurity assurance systems, AMR laboratories, molecular epidemiology laboratories, genome sequencing laboratories. So all these laboratories and facilities and projects and modules are supposed to contribute a diverse data that is possible to be integrated for the analysis of AMR over a period of time in respect of different organisms in different settings. For example, the global AMR dynamics, as it is apparent from the data from the global analysis of the sewage systems here, as it has been laid down, that it is found that there are systematic differences in the abundance and diversity of the AMR genes between the European and American continents, for example, the Asian continents. So there are visible and systematic differences that have been observed. So AMR gene abundances here, as you can see, the gene abundances very strongly correlating with the socio-economic and health and environmental factors, and the AMR classes and the AMR gene diversity and abundance varied by region, and that improving sanitation and health could potentially limit the global burden of AMR. So these days, the surface water, the agricultural runoff, the sewage water, all these things are supposed to be the surrogate markers of looking at the AMR class divergence or AMR gene divergences or abundances, and whether or not they correlate with the human activity, whether or not they correlate from the source or with the source of these waters that are flowing. And you know, it has been that in many of the developing countries of Asia, these activities actually originate as a part of the human activity. And human activity originates as a consequence of the climatological activities. In the Bay of Bengal, for example, when the Bangladesh and India and the other adjoining regions are there, because of the rising sea levels, the agricultural land is becoming barren. And because of that, a large number of populations are forced to migrate. And when they migrate and come to the cities like Dhaka, for example, here in Bangladesh, they tend to form large slums. And when these human activities in these slums, because of the poor handling of the human waste and excreta and domestic waste and also the polluted water and other things, there is a possibility that most of the superbugs or the antimicrobial drug resistance bacteria could actually be shaping their repertoires of the antimicrobial resistance genes. As you can see here, the cover page of the Nature magazine, where it has been aptly labeled and captioned that superbugs are rising. And then they in the background, you can see that the compact human living, in the cognate human living, could actually be the driver of antimicrobial resistance that have been rising over a period of time globally. But then here it appears that agricultural and animal husbandry activities and other human clinical settings, as well as the human practices over a period of time, such as open defecation and uncleanliness and not practicing properly the water sanitation and hygiene guidelines, could actually serve as the multiple drivers of the shaping up of AMR functions. And these drivers could very well be identified if we do a diverse data analysis with the help of whole genome sequencing, the 16S ribosomal RNA typing, the metagenomic analysis of the bacteria that are found in water, for example, as a surrogate, or the sewage. You can see that these genes have been over a period of time evolving, aggregating. We are presenting here the resistance pattern of different antibiotics among all infectious bacteria, isolated and cultured in ICDDRB laboratories from 2005 to 2017. So this is a 12-year-old data. I mean, 12-year period. It's a very voluminous and bulky data, and so it was found that the antibiotic resistance over a period of time has been rising, and so much so that most of the Klebsiella pneumoniae and E. coli have become 100% resistant to the all available antibiotics. So these data are nonetheless very important, also to give an indication which of the drugs could be important over a period of time for use. For example, chloramphenicol and cotrimoxazole here have shown a declining trend of antimicrobial resistance. But also in the field, for example, the 113 wastewater samples that were analyzed from the vicinity of the tertiary care hospitals and the community areas, the prevalence of NDM1 positive organisms like these are the superbugs type of organisms, the New Delhi metallo-beta-lactamase. So beta-lactamase positive organisms were higher in the vicinity of the tertiary care facilities as compared to the community areas. Also in the surface water in Bangladesh from the rural and urban areas, it was found that the urban surface waters were particularly rich in antibiotic resistance genes, with a higher number of them associated with plasmids, as it has been analyzed in an interesting paper here in M Systems, meaning that they are more likely to spread horizontally. So human waste appears to be the main driver of AMR in water in some of the developing countries of South Asia, such as Bangladesh. This has also been seen, nevertheless, in the refugee setting. The data from the population here, from the Rohingya camp in Bangladesh, you can see that the tap water or the hand pump water was found to be contaminated with the fecal coliforms and E. coli, and most of these organisms were, up to 60% of these organisms were resistant, and we could also found the ESBL positive E. coli in the drinking water from these camps. So what does that mean? That urban settings, the clinical settings, the rural settings, as well as the refugee settings, all these settings are the problem of antimicrobial resistance, and much of the resistance is driven by the plasmids. So the diversity of plasmids also is an important tool, or the databases of the plasmid diversity. As you can see here, this is an unpublished data. We are still hoping to publish this, that it will be possible that different classes of the plasmids, based on their gene content sharing, it will be possible to map the available plasmid data with the available AMR phenotypic data, and whether it will be possible to predict AMR based on the plasmid diversity or based on the plasmid gene content data. So we are working on these lines currently. But nevertheless, for example, it will be also possible to see whether the Helicobacter pylori status, as it has been given here from an important publication, from the Chen et al. In eBiomedicine, that the Helicobacter status, positive or negative, could have a large bearing on the diversification of different taxa in the human gut microbiota. And we are interested to see if we can reanalyze and check if there could be abundance of antimicrobial resistance genes among the microbiota of the humans, and also the corresponding microbiota in the human waste and habitats, and whether the Helicobacter pylori colonization status will be able to offer any insights with respect to the evolution of AMR. AMR has also been reported in the chickens, for example, the Helicobacter pylori that has been analyzed by us and our group in 2017. It was found that the antimicrobial resistance in chicken isolates as well as the human isolates were comparable. And the broiler chicken isolates and the free-range chicken isolates were also analyzed side by side, and it was found that the multiple drug resistance is quite rampant in chicken, and through chicken it is possible that it might get into human food. Helicobacter pylori, as it has been evolving, as I told you that this is a highly evolving organism, some of the fundamental pathways in metabolism such as the lipid biosynthesis could be exploited. As you can see here, the Helicobacter pylori 421 has been, when subjected to knockout, the cell wall structure was compromised, and the bacteria usually when they are spiral, they become cocoid, and when the gene was replaced back, they became again the spiral. Similarly, the Helicobacter pylori, the resistance phenotype was also changed because of the knockout that was generated. That the knockout has made them more sensitive to antibiotics, and knockout was replaced. Then they became resistant. The organism otherwise very much resistant. I would like to conclude that AMR is an open-ended multi-sectoral issue, and the data integration and analysis is important in the study of global landscape of AMR, and much of the human activity drives the abundance of AMR classes and genes in sewage and waters, and human hygiene and other sanitary practices could curb AMR in the developing countries, and the discovery efforts are important, as important as the wash efforts. Thank you so much for your kind attention.
Thank you very much, Professor Ahmed.
Yes.
Yeah, I would like to take a few questions from the audience, 2 or 3 questions. We have 5 minutes of time for questions. Yeah.
Yes, please.
Yeah.
Professor Jayaraman, can we take some questions, or we have to take questions at the end?
I'm not able to see. We will take questions from the chat if they are not raising the hands. Yeah. If anybody has any questions, please put it in the
actually
chat.
to save time for questions and answers, that's how this was finished in 15 minutes, and so that in anticipation that we'll have more.
We have time. Definitely, we have time for 5 minutes, sir. So we'll just wait for somebody to ask questions here.
So we'll wait for the question. Let me quickly check the chat box if we have some questions.
Yes, sir. There is one question.
Yes.
Can gut microbiome and antimicrobial resistance have a role in tumorigenesis? Yeah.
In the context of methyl transferases of microbial origin, as I told that they are capable of even modifying the promoters of their host genes. So it is quite possible, some of the microbial organisms could drive the gene expression in their favor or otherwise. And it is very much seen in case of Helicobacter pylori as an organism, which is now the principal cause of, gastroduodenal carcinoma. So it is quite possible that there could be associations, of some of the methyl transferases or modifying genes, which may eventually render, some of the, pathways going corrupted because of their selective modification of the promoters. But this has been modeled, as we shown in case of the nucleic acid research paper. But then there could be a wet lab evidence, or using so many samples, from human tumors and otherwise, whether or not they are able to, upregulate or downregulate the expression of certain genes and subset of genes in their favor or otherwise. Okay, so unless we map the patterns of some of the genes that are expressed in the gastroduodenal cancers, and whether there is an association of the promoter as well as the methyl transferases of microbial origin
those associations, if could be precisely mapped and proven through the data, it will be certainly possible to show that there could be association with tumorigenesis. Any other questions?
Sir, if the participant have no other questions, so I would like to ask a question.
Yes.
So
please.
the path from epidemiology to discovery, it's it's not simple. You might have gone through many up and downs.
Yes.
Updating your strategies always to match what is happening in the recent time. So can you tell me how you consider this strategy is important in the future, and what is in the near future is going to support this strategy, which is going from epidemiology to discovery, in particularly in concern to pathways to antimicrobial resistance, and particularly to omics approaches.
No, this is an interesting question. Actually, there is definitely a nice pathway. For example, I would like to share with you a single diagram that gives the entire thing. I don't know if I can share a slide that gives you a glimpse of everything together. What we are looking from epidemiology to discovery. Are you able to see this, by the way?
Yes, sir. I think your screen is just beginning to share. Yeah, now we are able to.
Huh. So this was for some time back. Yes, please.
Request others to please mute your mic, please. Others, please mute your mic.
Okay, so if you can see that the data flow, how molecular epidemiological data could feed back into the discovery efforts. So what you have asked, chairman sir, is exactly summarizing this particular slide. But
just a minute.
There's a lot of background discussion and laughing.
Somebody, Sanika, I think somebody has unmuted the mic and causing a lot of disturbance. Please mute your mic. Yeah.
Yeah. So am I audible now? So you see that genomics, bioinformatics, microbiome data, the health systems data, the biomonitoring data, the trials data, behavioral data can all get into the discovery process. And then we have the enteric microbiome, microbiota analysis, bioinformatics, next generation diagnostics, big data translation goals, and discovery. So discovery basically is limited to biomarkers, vaccine candidates, and drug targets. There could be, many other approaches, but primarily these approaches based on the epidemiological data. For example, if we are looking for the development of a novel antibiotic, we must also have a drug resistance data, or whether or not the targets for that particular antibiotic are actually spread out in the universe. For example, antimicrobial resistance could also be a naturally occurring trait in the soil microbiota, but it could be an acquired trait in the pathogens. So those kind of data are very important for example, in order to precisely target. That is what is the meaning. And overall, the discovery has to be used in the field, So unless we have the data coming from the field, the discovery may not be bolstered properly.
Yeah, thank you, Professor Niaz, for answering the question. And this slide made the things, very much clear. And I think we have a hope that in the future, your pathway will lead to some very useful discoveries and making use of many real aspects of this particular process. Thank you very much, sir.
Thank you so much. And I thank one and all for listening to my talk.
Professor Jayaraman, we may start with the next talk.
start the next talk. Yeah.
And I just want to confirm whether Professor Gokhale has able to join.
Professor Holger is, are you there, sir? No. Okay. So, okay, we'll proceed to the invited talk number 3. Yeah. We will, this talk will be given by Professor Andre Lemos Antunes on microbial ecology, biotechnology, and education, showcasing the versatility of bioinformatics applied to microbiology. I will now give a very brief overview of his research. He is an associate professor at the State Key Laboratory of Lunar and Planetary Sciences at Macau University of Science and Technology. There he is the leader of the astrobiology research group and the coordinator of the recently launched astrobiology and cosmochemistry experimental platforms. He is an expert in microbiology of extreme environments, having specialized in deep sea and high salinity locations. He has done his undergraduate study in the biology at the University of Coimbra, Portugal, one of the oldest universities in Europe. He has worked at several countries across the globe in Europe as well as Africa and Middle East. Before his current appointment, he has had previous faculty positions at Cape Verde and in the UK. His current research focus revolves around biodiversity, bioprospection of unexplored extreme environments, adaptation to environmental extremes and environmental gradients, geomicrobiology and astrobiology. Professor Dr. Antunes has extensive experience with international collaborations, having participated as a PI and team member of several research projects across the globe and having participated and led several research expeditions. He has also acted as a consultant and invited committee member for NASA, ESA, and the European Science Foundation in several different projects linked with astrobiology and planetary protection. Professor Dr. Antunes is an elected member of the Communications Committee of the Microbiology Society and editorial member for a variety of leading journals in his field. He is also a regular contributor to the newspaper Ponto Final with an opinion column, Microcosmos, and of the radio show Eureka by Radio Macau, both dedicated to science and technology. Professor Antunes, please, yeah.
Okay, thank you very much. I believe that you can see my screen, yes?
Yes, sir, we can.
Okay, so thank you very much for the nice introduction and for the presentation to attend this, this meeting. It's an honor to be telling you a little bit about my research here. And so I'm preaching to the choir here, but I'm sure that you all know we live in this big data world and we're basically drowning on oceans of data. We have this amazing amount of data that is constantly being generated and that we struggle with. Luckily for us, bioinformatics is helping us to deal with all this information, to analyze and get some results out of it. It's been particularly important in biosciences in general and specifically in microbiology also. And so I wanted to highlight a couple of examples on how versatile biotechnology can be, and now we can apply to several different aspects of microbiology. So first example is on microbial ecology, and it's quite fitting that I was discussing oceans of data because the first example actually relates to the sea. It relates to the Red Sea specifically. And so you all heard about the Red Sea before, and what you probably don't know is that the Red Sea is opening up, and not in a biblical way like you had with Moses when he crossed with his people fleeing from the Egyptians. We have it from a more of a geological aspect. So we have the Arabian and the African tectonic plates are currently splitting, and they're resulting in the widening of the Red Sea. At the same time, this creates new isolated topographical depressions that are placed in the central axis of the Red Sea. And because the whole of the Red Sea has a deeper geological layer underneath it, this is constituted basically by concentrated salt. What this allows, what the formation of these depressions and this tectonic movement allows, is that part of the salt is leached by regular seawater. So regular red seawater becomes highly enriched in salt and is converted into a brine. Because brines are so dense, they will be trapped in these isolated deeps that I mentioned. And so we currently know around 25 different deep sea brines that occur in the Red Sea area. And these are some really interesting environments in some of the most extreme places on our planet. And so despite the fact that they're formed the same way, they can be quite different. And you have two extreme examples. You have Kebret Deep, which is further north, and Atlantis 2, which is one of the southernmost ones. They have very different dimensions. So Kebret Deep is around 2.5 square kilometers, and Atlantis 2 is around 35 square kilometers, and also differences in temperature. Kebret Deep has a temperature of around 23 degrees Celsius, and Atlantis 2 has extensive hydrothermal input and currently has a temperature of around 70 degrees Celsius. At the same time, differences in local geological setting contribute to the creation or the establishment of a unique physical chemical fingerprint for each one of these brines. So they're all different. They all have slightly different conditions, which makes them really interesting environments to study. The additional aspect to take into consideration here is that brines, because they are so dense and they're trapped in the deeps, they don't really mix with seawater that much. And so they form this very characteristic sharp brine seawater interface, where you see drastic transition from regular deep seawater into brine conditions within very few meters. Also, because the brines are so dense, any type of particles that would normally sink throughout the water column all the way to the bottom will actually be trapped. They will be caught at this density barrier. They will not be dense enough to break through, and so they will accumulate at the brine seawater interface, which is really interesting from a microbiological perspective also. And so at the brine seawater interface, we have transition from low temperature to high temperature, low salinity to extremely high salinity, transition from oxid to anoxic. So there's a very drastic shift in conditions. Here I have a short video. I hope that this will work. I just wanted to show you a little bit of a video. This is video footage from an ROV, so a submarine that we took into the brines. And you see this sort of hazy texture that you see at the bottom side of the screen. This is actually the high density of particles that you see over there. These are the particles that accumulate the brine seawater interface. And so this is the very first video footage from brine seawater interface. And so these environments are really interesting from microbial perspective. Curiously enough, the first microbial studies that were done in the brines date from the late 1960s. And what they found was this: they found absolutely nothing. They were unable to grow any microbes. There are actually papers from back then that say these brines are sterile. There's no way that any sort of life form could exist down there, could survive extreme conditions, because it's deep sea, so it's high pressure, high salinity, high temperature, high concentration of heavy metals, no oxygen. It's very, very harsh. And so there, for a long period of time, people just thought that nothing would be able to grow down there. Luckily for us, this this changed considerably with the advent of molecular biology and the new perspectives that it opened in study of microbial diversity. And so we now know that the reality is quite different. So these locations are thriving. They have thriving microbial communities. They have very diverse types of microbes. And not only that, they are a source. They have very high number of deeply branching new phylogenetic lineages that aren't really closely related to anything else on the planet. Although we do see certain groups that are present across every single brine pool, for example. Studies that were based on 16S only and then later on metagenomics also were able to identify clear stratification in microbial community structure. So if you're sampling deep sea water, or if you're sampling brine seawater interface in different parts of the brine seawater interface or the brine itself, you do find very different types of microbes in there, which makes sense because conditions are very different. From an isolation perspective and regarding new microbes that were found there, we also have some really interesting finds, and it's not the focus of the talk today. Just wanted to highlight this one in particular. This is Haloplasma contractile. It represented a new order and likely a new phylum within the bacteria, and it's a very unusual microbe that has unique morphology and unique cellular contraction cycle, which is really interesting and really unusual. And so taking us to the example that I wanted to highlight here is I wanted to focus a little bit on the viral communities. So as I was telling you, the last couple of years have brought a lot of studies focusing on different brine pools of the Red Sea. And we do see an accumulation of molecular-based data, namely from metagenomics. And the issue that we have with this type of studies is that normally they tend to be very narrow focused. People sequence because they're looking for a very specific, very narrow thing, and they're only interested in that. And so the idea that we had was to do a sort of a parallel thing, actually analyze data that was already available, already publicly available and existed, and try to extract additional information out of it. And so we wanted to look specifically into the viral communities because nobody knew anything about those. And so we were able to analyze data from Atlantis-2, from Discovery Deep, and from Kebrit. And in Kebrit, we had data from upper and lower interface. And so we were able to analyze it and get the first insights into the viral communities. So we're able to see that in general, there was a dominance of double-stranded DNA viruses and some similarities between the different brine pools. Atlantis-2 was clearly the one that was most different. And within double-stranded DNA viruses, a predominance of caudovirales, again, with Atlantis-2 being the most different one. And when you look specifically within the caudovirales, again, Atlantis-2 as a big source of unclassified caudovirales. And this makes sense because Atlantis-2 is the most extreme brine pool in the Red Sea. It's the one that has the highest temperature, so it has additional environmental factors at play there. On the other side, we were able to show that viral communities were also stratified. So in this table, you can separate between KL and KU. So that's Kebrit upper and lower interface. And so we could see that the viruses that were in the upper interface were more closely related with viruses that are typical in marine environments versus lower interface, where the viruses were more closely related with typical viruses that you find in salterns and other high salinity environments, which made total sense, but hadn't been previously reported. So we got a really interesting paper out of this, and we were able to get the very first insights into the viral communities in these brines and identify some future targets for interesting research. Second example that I wanted to highlight revolves around biotechnology. So microbiology is increasingly seen, and microbes are increasingly seen as the big solution to society's big issues, like how to feed the world, how to fuel the world, and how to heal the world. And so microbes offer a very wide range of potential applications. One in particular that I wanted to highlight here is one that is not very well known or is not as visible as some of the others, like production of bioplastics or biofuels or new drugs. This is something completely different. This revolves around production of biominerals. This is what you see here. These are pictures of biogenic calcium carbonate on top of colonies. And so in general, biomineralization is a relatively well-known phenomenon. So we know that it's more or less widespread. There are a lot of organisms that are able to do this. From a biotechnological perspective, the main focus has been on microbially induced calcite precipitation. And most commonly, when it's associated with ureolysis, the degradation of urea as a way of producing this. There's still a lot of unknowns regarding MICP. So there's a lot of things that we don't know. But it's a very interesting topic. On one side, I think it's really cool that it bridges the gap between two very different fields of science, bioscience and geoscience, which I think is a plus. And on the other side, it's captured significant attention and has been is currently already widely used for a variety of different things, most importantly in civil engineering and for producing things like self-healing concrete. So when concrete develops cracks, it will be able to repair itself. It's also used for restoration of cultural artifacts, things like statues and facades of historical buildings. It's also used to improve the agricultural quality of soils, for example. And there's some really interesting pilot projects that look into this as a potential way of capturing CO2 and controlling climate change, which is something that is a really interesting application. Just some highlights about concrete. So there's some stuff that I found. There's so much concrete in the world that soon it will outweigh all living matter, and something that we're not necessarily immediately aware of. It's the single biggest category of anthropogenic material, and it's one of the largest CO2 emission sources. And we rarely hear about this. So there's a clear drive towards improving concrete and finding new ways to make use of microbes to improve it. On the other side, an additional interesting application that is relatively new is the potential to use MICP and calcium carbonate production as a way of adding value to resources found on other planets. And so there's ongoing talks of making use of this approach to consolidate lunar dust, for example, and get construction materials out of it as a way of cutting costs to build a lunar base in the near future, for example. And so when we're discussing biogenic calcium carbonate, this is the guy that we're normally referring to. This is Bacillus. This is a photo of a species of the genus Bacillus. Bacillus is very widely used for most of the biotechnological applications that I mentioned here. And so part of the slides that I'm going to show next reflect the work done by Philippa Loftus, a former master's student of mine in the UK. And so the idea that we had is for her to explore a little bit, specifically this genus, and look at calcium carbonate production, analyzing the bioinformatics approach together with wet lab experiments. The big advantages that we had here with using Bacillus is that we have access to huge data sets. There's a lot of different species. Many of them have been sequenced, so we have whole genome sequence data readily available in public databases. It grows relatively well under standardized conditions. It's really easy to work with this microbe. Plus, it's the one that is most relevant for biotechnology anyway. First thing that she did was to identify the genes that were involved in calcium carbonate production. So this was part of the literature review that she was working on and trying to get the list of all the genes that had been previously identified as being involved with this. She then did a list of all the species that had been described, all of the genomes for all of these species, all of the ones that were publicly available. So we had 379 different strains. We had 114 genomes that were available, and she analyzed all of them. This is a lot of information. So I'm only going to show some of the results. I'm going to highlight some of the stuff that she found. So regarding visual presentation, I'm going to show results of 52 genome analysis that she's done. And so she based this on this phylogenetic tree, which is sort of the most complete that she could find. And underlined in red are species that were previously untested for biomineral production. Underlined in green are known calcium carbonate producers. And so the idea here was to produce this sort of genomic fingerprint for calcium carbonate production and try to see if there was similarities between species that were well-known producers versus species that had never been tested before, and also look specifically at species that are already very widely used for biotech applications. So based on this, she grew a selection of species that were already known, already applied for biotechnology, and compare them in the lab with species that had never been tested before. She used similar conditions, standardized. She grew it under very well-controlled conditions and control it throughout time to see how the biomineral production evolved with time. And so what we saw is an increase in number of crystals, an increase in their size with longer incubation time, which makes sense. So it's sort of sensible to be that way. Some things that we weren't really expecting is, on one side, that she analyzed in more detail 2 different subclusters within Bacillus, and she was able to see that there were some apparent differences in mineral morphology that seemed to be linked with species being in one cluster versus the other. And so this is something that is really interesting and that we want to look into with more detail. The other thing that she looked into was the quantity of biomineral that was produced. So what was the biomineral yield? And so as part of some of the new species that had never been tested before, she found some microbes that had some species that produced a lot of biomineral, comparable levels to what is to some of the species that are used for biotechnology. So they're good suitable candidates as alternatives. So as a sort of a general overview, and this is just a very summarized version of what she achieved, we were able to show that genomic-based analysis is a very powerful tool to find new biomineral producers. We have identified so far 10 new carbonate species, carbonate biomineral producing species that nobody knew that they produce these minerals. And we also see some interesting trends that we want to look into further. And this is part of ongoing work, so stay tuned. The third and final aspect that I wanted to highlight here, because we don't really have much time to go into detail, is education. And here the story is quite different. So this story starts with me being challenged by having to teach a new module at my former university. And the module was on genomics and bioinformatics. And my institution, my former institution, placed a lot of emphasis on different types of assessments, not necessarily just exams, and confronting students with situations that were more sort of like real life experience sort of thing. And so the idea here was to make use of the experience that I had with the brines of the Red Sea and the data sets that were readily available there. We had a variety of genomic data sets and metagenomic data sets. And the idea was to confront the students with specific tasks. So one of the things that students needed to do was they needed to register for online use of RAST and MGRAST, 2 basic tools for data analysis of genomic and metagenomic data sets from microbiology. And on the other side, this needed to be complemented with teaching them a little bit about tools for data search, because bioinformatics is pretty empty if it's just looking at data. You need to be able to put it in context, interpret, understand, and explain some of the trends that you observe there. So these were final year students. They already had some prior experience, but they clearly benefited from a more thorough, in-depth take on how to use PubMed, the advantages of using things like ResearchGate or Google Scholar to find relevant information for their work. So where should they start? Suggestions for students was to collect data from papers on the specific environments and the microbes that they were going to look at, list of genes, enzymes, and pathways that were involved in this, and to keep their sanity, to make it clear that they were not asked to be 100% thorough. They needed to focus on a couple of examples in their report that were interesting and meaningful, and they needed to contextualize and explain it properly. I divided the class into 4 groups of 4 to 5 students, and they had different topics, either osmotic stress adaptation or heavy metal resistance. And depending on the topic that they had to interrogate specific genomes and specific metagenomes based on the topic that they were allocated. And I'd worked with these students before, and they've done group work before. And the common criticism that we normally got is that students really enjoyed doing different types of activities, but they really struggle with group work. And the common issue that they had is that they felt that there were always 1 or 2 team members that didn't really contribute as much, and that was a persistent issue that we had and we needed to deal with, because that was affecting student satisfaction and whether they enjoy the class, whether they enjoy the assignment or not. And so as part of it, I designed this peer assessment report, and I incorporated it as part of the assessment. So all group members needed to grade their fellow students based on their contribution to the group work. And so they needed to fill this form. And this was part of the assessment guideline. Of course, well, the genomics and bioinformatics project was worth 60% of the final grade. Project report was worth 85% of the project, and there were different assessment criteria for different sections of it. But then the peer review report was worth 15% of it, and it's something that students really appreciated and really enjoyed. As an additional aspect here, I also incorporated a draft submission option. So students were able to submit a draft of the report for me to have a quick look and just receive some general feedback and see if they were on track or if there were certain things that they were not incorporating that they should. The results that we got were really, really good. Students really enjoyed being part of this, doing part of this work. And part of it was done in class. We were in computer lab, and I would hop from group to group and provide assistance and see whether they were struggling and help them in interpreting the data also. And that worked really well. We had a really good environment in class. And so they were able to produce these comparative tables that were relatively simple on one side to more complex stuff, like looking at genomic context and trying to interpret the presence of specific genes and the existence of entire pathways or partial pathways and how it related to other microbes that were either nearby or a little bit further away. They even did some they also did some comparison at the microbial community level that was really interesting and had some really interesting insights. So some of the stuff was really publication level type of work, which is something that I really wasn't expecting. So they did really, really good work. This type of assessment was very interesting and was even highlighted by the Royal Society of Biology. So we had our degree accreditation by the Royal Society of Biology. I was teaching back then in the UK, and they really liked how innovative our assessments were. And this was one of the assessments that they highlighted as part of their report as part of the accreditation process. And so as final conclusions, because I'm really running out of time, there is an increasingly large amount of idle data with different levels of unextracted and useful information. So on one side, this is a challenge, but also an opportunity that can be used for a variety of different things. Bioinformatics is increasingly relevant and permeating further into different areas. I mentioned planetary sciences today, for example, which is something that you might have never thought of, but it's another aspect that is really important. And for the foreseeable future, bioinformatics will continue to have this deep impact and revolutionize the fields of microbiology, biosciences, and beyond with wide-reaching implications. Thank you very much for your time. If you want to get in touch with me, feel free to send me an email, contact me via Twitter, look me up on Google Scholar, ResearchGate, or Twitter. And thank you very much.
Thank you very much, Professor Antunes, for an exciting talk. Now he will be answering a few questions if you have any. Please ask the questions in the chat. Yeah, there is one question from Hriday Mahanta. He is asking, are the data available in the open access for further research? Yeah.
Yes, they are. So the data that I use, and so the examples that I provided today, for example, for the things that I did with the students, they were publicly available. They were open access. And I picked a specific example and I incorporated part of the stuff that made sense to me based on my prior research. This is something that is easily adaptable to different types of settings. If you're teaching in India, if you're teaching in other parts of the globe, if you're struggling with what type of activities can you incorporate as part of your classes with COVID limitations and students not being able to do stuff in the lab, this is something that is very easily adaptable, and it's something that is very rewarding both for faculty and for students as projects.
Are there any other questions?
Yeah, Professor Andre, I think I'm really very excited to know how you connected the research with education. That's really very, very difficult. Sometimes people do more of research, and sometimes people they educate in isolation, and they are two different worlds. And and particularly for the students. And the way you have connected the student research and education, and you have nicely designed some evaluation forms, I really, very nice to see. And because I'm also I'm also like you. I always try to make a bridge between education and research for my students also. I will request to you to share your evaluation forms so that I can get some idea to develop my own from them. And second thing, apart from the limitations, like there are few people which are not performing in a group, you devise a strategy. Any other limitation you find in terms of group based activity for such a wide experiments?
Okay, thank you very much for your comments. I'd be happy to share information, experience. Feel free to contact me afterwards. I'd be more than happy to continue the discussion in other platforms. Regarding other issues that we have with students, with this specific module, I think that we were very, very lucky. On one side, this was an optional module, okay? So on one side, this was an optional module, so it wasn't compulsory for all students that we had in the department. It was compulsory only for the students that were doing the genetics degree on one side. It worked really nicely with the fact that students were in their final year, so they were a little bit more mature and more focused. And the fact that I had them working in small groups and we had a relatively small class and me going from group to group and working together with them, I think that worked really, really nicely. I was also lucky that my institution had a very flexible perspective on curricula, like what you're supposed to teach. So I know that some institutions don't have that type of flexibility, and they hire you or they assign a specific module and they want you to teach content, deliver content, and they have this long list that you need to deliver. At the previous university where I was working on and teaching, that was something that was seen as not secondary, but as something that was flexible and you could adapt. And the focus was more on different types of assessment and improving the skills of our students rather than just being focused on purely theoretical based, lecture-based teaching. So that was something that was very positive.
Thank you. Are there any questions? If there are
Thank
no
you,
further
Professor.
questions, I would like to ask one question, Professor.
Yes.
You have worked in different extreme environments. Could you please classify the extreme environments you worked with and also your experiences and what type of work you have done on different extreme environments? Yeah.
Okay, that's a very interesting question. Very complex answer though. So the majority of most of the research that I've done has been focused on high salinity environments, particularly under deep sea conditions. So it's a combination of high salinity, which is more sort of traditional, and there I work mostly with archaea, but also with bacteria. As I've done a lot of work in the Red Sea area, it's more accurate to classify it as polyextremophilic because you have multiple extremes. So you have high pressure also. I work with high pressure cultivation, high salinity, anoxic conditions. So I work with a variety of different settings, but most of the focus has been on high salinity.
Thank you, Professor. Yeah. If there are no further questions, let us thank Professor by putting your hands together and doing a big clap. Yeah.
Professor Jaraman,
yeah.
there's one question. Let me ask the question. In the biomineralization work with the bacillus, what were the samples from which they were isolated? So there's a question from Priya.
Okay, Priya. Thank you very much for the question. We work with type species of bacillus, and bacillus can be isolated from a very wide range of environments. So it's something that we didn't take into consideration. Our focus was working with a wide range of bacillus. So they come from any type of environment, from very diverse type of environments. Okay. Something else that I wanted to add that I didn't mention at the end of my talk. So I work in Macau at the State Key Laboratory of Lunar and Planetary Sciences. Even though I'm a bioscientist, I work with planetary sciences, which is really interesting, and it's something that I didn't really focus that much on. I'm assuming that several of our attendants might be students and early career researchers. We're regularly hiring people and recruiting new students. So if you're interested in some of the topics that I mentioned today, or you're curious, just look me up. If you're interested in coming and studying to Macau, just send me an email and stay in touch, okay?
Thank you, Professor Andre. And I have a question, a relevant question with the different things you are offering. You mentioned about some radio talks and other things, I think, in your introduction by Professor Jayaraman. And can you elaborate on what type of these platforms you are using for science?
Sorry, I didn't understand the question.
I think in your introduction, I think Professor Jayaraman mentioned about some radio talks or radio programs. So can you elaborate
yes.
what type of?
Yes, I understand now. Yeah, so it's something that started during the pandemic, and it's something that, well, we had the panel discussion before. And so as part of the setting during the pandemic, it's something that worried me to see that people were so disconnected from science, and there was this huge gap between scientists and general population and media in general. So it's something that I actively engage with, trying to look for closer ties with local media. So as part of it, I started writing a regular column for the local newspaper, and I discussed different aspects of science, new things that came out related either with biosciences or planetary sciences. And the radio show came shortly after. So the local radio decided to start a weekly show. It's relatively short, and they invite a scientist to come over and discuss science. And it's 15-minute based. I go once a month, and it's something that it's been really interesting. I think it's very important for us as scientists and as educators to make sure that we build these bridges to media and to regular people, to the bigger audience, rather than just staying in the lab, staying in the university. It's important to build these bridges.
Excellently, excellently answered. I was excited to know about that. Thank you, Andre. Professor Jayaraman, I think we don't have any other questions.
Yeah. So thank you, Professor. I think it was a very excellent and exciting talk, and let us all show our appreciation by putting our hands together and giving a big thank you, sir.
Thank you very much for having me once again. It was a pleasure.
Yeah. Before moving on to the other talks, I would like to again ask whether Professor Golke is here. We'll wait for a second. Yeah. No. So okay. So let us move on to the oral talks. The first oral talk is going to be by Danya and team, and it is going to be on an in silico approach towards unraveling common blood-based differentially expressed gene and interlinked pathways in neurodegenerative disorders. Danya and team, please. Yeah.
Yes, sir. I'll just share my screen.
Yeah.
Just want to check if I'm audible and my screen is visible.
Yes. Yeah.
All right. So before I start, I just want to thank the organizers of InBix-2021 for providing us with the opportunity to present our work on this platform. My name is Dhanya Pradeep, and I'm a final year undergraduate student at the Department of Biotechnology at BMS College of Engineering, Bangalore. And along with Disha Bangalore, Renuka Prasad, and my professor, Dr. Savitri Bhatt, we worked on a project titled An In Silico Approach Towards Unraveling Common Blood-Based Differentially Expressed Genes and Interlinked Pathways in Neurodegenerative Disorders. So with the global increase in lifespan, the burden and prevalence of neurodegenerative disorders is also rapidly increasing. According to the Global Burden of Disease study in 2016, approximately 3.74 million people in South Asia, including 2.93 million people from India, were experiencing neurological related disorders, and this number is only expected to increase in the coming decades. So, since we're aware that neurodegenerative disorders begin to present their symptoms approximately 2 decades prior to the presentation of severe clinical symptoms, there's a need to identify other therapeutic strategies to monitor these disorders. So some of the current diagnostics include neuropathology, which is considered to be the gold standard. However, it's based on an autopsy that's carried out after the death of a patient. So one of the most powerful molecular diagnostics is the application of biomarkers that are considered to be promising in aiding in early diagnosis of neurodegenerative disorders. So on the right-hand side, you can see a few of the genetic and biochemical diagnostic markers specific to Alzheimer's, Parkinson's, and Huntington's disease. So for our analysis, we hypothesized that there's a statistically significant difference in gene expression between the two sample groups, and the sample groups being the disease state and the healthy controls. So we aim to identify common differentially expressed genes between Alzheimer's, Parkinson's, and Huntington's disease using peripheral blood mononuclear cell microarray data, since one of our major goals was to identify potential prognostic and diagnostic blood-based biomarkers. We also focused on identifying potential links among the disorders in terms of their protein-protein interactions. So coming to the workflow, we first obtained data sets from Gene Expression Omnibus, and then we analyzed the data sets using GEO2R and the Bioconductor project in R. We then filtered the data according to the log FC and P values, which enabled us to segregate the genes into up and down regulated genes. On construction of the Venn diagrams, we were able to identify the common differentially expressed genes, which we input to string to obtain the protein-protein interaction network. We also input all the differentially expressed genes into Enricher to carry out the pathway enrichment analysis. And finally, from the results that we obtained from string, we fed the data to Cytoscape to obtain a list of the hub genes. So here are the data sets that we used for our analysis, and you can see the threshold values that we took into consideration while filtering our data into up and down regulated genes. And all the genes with a p-value of less than 0.05 were considered to be statistically significant. So moving on to our results, on construction of the Venn diagram, we were able to identify two common genes, namely SMARCA1 and DZIP3. Both these genes have previously been implicated in neurodegenerative disorders, but their specific functions, particularly in the context of Alzheimer's, Parkinson's, and Huntington's, is still unclear. To visualize our data, we also constructed volcano plots. So red indicates the upregulated genes, blue indicates the downregulated genes, and black represents the statistically insignificant genes. We also annotated the most statistically significant genes. Further, we constructed heat maps to visualize the level of gene expression of the top 250 differentially expressed genes that we screened using the Limma package in R. So moving on to the next section, we constructed the protein-protein interaction network based on confidence using string, and we also carried out gene ontology and KEGG pathway analysis to identify the pathways in which the differentially expressed genes that we identified were significantly enriched. And finally, we identified the top 10 hub genes using Cytoscape. These are arranged in order of their node degree. So all these genes here mentioned here have previously been implicated in neurodegenerative disorders, particularly in Alzheimer's disease, but their specific roles when it comes to Parkinson's and Huntington's is still unclear. So in conclusion, we were able to identify a number of potential blood-based biomarkers that can aid in monitoring the progression of these neurodegenerative disorders. With any bioinformatics analysis, it's important to note that further in vitro or in vivo research would have to be conducted to validate our findings and also to understand the specific roles that these common genes and hub genes play in the progression of these disorders. Further research can also be conducted by taking into account the differences between genders in the sample data. And finally, research can also be extended to include a wide array of neurodegenerative disorders and identify common differentially expressed genes among them to find a sort of convergence between the disorders. So I'd like to thank the Department of Biotechnology, Government of India for their funds and support, as well as VMSC management for their support through our project. So here are the list of a couple of the references, and that concludes my talk. Thank you.
Thank you very much, Dania, for your excellent talk.
Thank you.
Audience, if you have any questions, please post it in the chat. Yeah.
Yeah, sir, I've raised.
I think Prash has raised hand. Prash, do you want to ask any question?
Yeah. Thank you, Harpreet. Yeah, thank you, sir. Dania, it's an excellent talk, for an undergraduate level. Congratulations.
Thank you, sir.
I was just wondering, did you find any non-coding RNAs among those differential expressed genes?
There were a couple. We haven't specifically focused on those. We were but just looking at the common genes that we identified between the 3 data sets. But it's definitely something we will look into since non-coding RNAs do.
You should, probably, consider that, because I'm sure you would definitely find a lot of, long non-coding RNAs and PV and microRNAs as well. Second thing is, there are a good number of studies. I think, I mean, over the last 3-4 years, ever since or even before COVID has come, there were plethora of studies, done on neurodegenerative disorders. Taking the GO2R, the simplest NCBI-based things, even the RNA-seq, data sets, that are publicly available. So, did you find this SMARC1 gene, among the commonmost genes with those previous studies that people have analyzed?
We did identify it in one of the studies that focused on schizophrenia spectrum disorders. Apart from that, we didn't really identify it in the other differentially expressed gene studies. I think it's mainly because our microarray data sets were completely different from what they had used.
Right. That's it. Huh? Congratulations. Yeah. Thank you.
Thank you, sir.
Yeah, great. Thank you, sir. Yeah.
Thank you. Thank you, Prash, and Dhanya. May I also ask a question, because you have taken different data sets and you have tried to combine the genes between the database to find the common genes. So different database may be raised with different test condition or experimental conditions. I just wanted to ask, have you taken care of the variations? How have you normalized the data so that you can compare them at a one platform?
So we essentially use the lemma package in the bioconductor project in R. So it ultimately normalizes the data and it gives us a p-adjusted value. So all the genes were screened based on that p-adjusted value. So all of them with a p-value of adjusted p-value of less than 0.05 were considered to be statistically significant. So yes, the data was normalized.
Thank you, Dhanya. Thank you. It's really an excellent talk.
There is one more question from Shivam. Have you found any genes which are shown interactions and responsible for switching? A minute. Yeah. Just a minute. Yeah. Switching on or off of the other genes. Yeah.
Specifically with all the differentially expressed genes, we didn't go in depth in identifying the specific roles, but it's definitely something we will look at.
Okay, thank you. Yeah.
Thank you.
We will now move on to the next talk. Harpreet, can you please announce? Yeah.
Yeah. Thank you very much, sir. Thank you, Professor Jayaraman. Thank you, Dhanya, for the nice talk. And thank you, Prash, and others to ask questions. And I will encourage everybody to ask questions, relevant questions, basically, so that we can make this session more interactive. So we move on to the next talk by Asif Adil. Asif Adil and team, they will be talking on, Spark-based handling and normalization framework for single-cell transcriptomic data. So I hope Asif, Adil and team is ready.
Hi, good afternoon, everyone. Thank you.
Yeah. Will you please share your screen? Yeah, thank you.
Yeah, sure. Yeah, is it visible? So
yes,
is my screen visible?
Your screen is visible. Are we able to visualize the screen? Yes, now it's visible to me. There may be some network delay, so it's visible to me
Yeah,
now.
actually, thank you. A very good afternoon to everybody. I'm Asif Adil from Department of Computer Sciences, Baba Ghulam Shah Bacha University, Rajouri, and it's a quite far away located location university, and there might be some internet issues over here. Sorry for that. So today I'll be talking about the project which are me and my supervisor Professor Muhammad Asghar is currently working on. It's Apache Spark based handling and normalization framework for single cell transcriptomic data. Starting from the introduction, earlier the cells were sequenced in a bulk. The cells were sequenced in a bulk fashion, and the expression measurements were calculated considering the average of each cell across. But there are cellular level heterogeneity which was found in 1957, and it can be depicted from the fact that at the early stage of development the cell is able to decide its own fate. So that led to the question that if are the cells at this individual level expressing some genes differently? Yes. So the single cell sequencing started, the scientists started the single cell sequencing. So this is the current, this is the typical single cell RNA workflow. It starts with the dissociation of the cells, isolation of the cells, and then a single cell is being extracted and the RNA is extracted from it, followed by complementary DNA synthesis, amplification, sequencing, and the expression matrix is generated, which is followed by the downstream analysis. This is how our data looks like. It's almost sparse, considering that the sequencing starting materials are pretty low, as much as 6 picograms. So the data generated from the single cell RNA sequencing is very large. It's a big data that has been proved by the recent research, and the big data is actually having the characteristics of 3 V's: the volume, velocity, and variety, and all of them are followed by the single cell data. And these are like, there is actually a study from 2017 which is having 1.3 million transcriptomes of mouse brain tissue, and the droplet based techniques have enabled us to process tens of thousands of cells in merely an hour's time. And this is how the PubMed articles talking about single cell RNA-seq are also talking about data and data sets, which we can infer that the data sets are quite frequently used, and we need to analyze them. This is the PubMed article analysis. We use the search queries for big data along with the single cell RNA-seq. These are the data sizes on human cell atlas. Human cell atlas is basically a consortium project which will be analyzing, which will be actually sequencing the each and every cell of an individual of a human. And currently there are the largest data set right now on human cell atlas is of 1.5 million cells. Currently, there are I think number of tools are there, but we have selected a couple of tools. It's SURAT. SURAT is given by Satija et al. It's an R-based analytical pipeline providing methods for a variety of downstream analysis. The contribution from this is it addresses the downstream analysis of single cell and provides command-based interface for analysis. But there are certain limitations, and they are that it's not scalable, and there are limited methods for different analysis, and it bears a cell load of only 1 million cells. Another tool for single cell analysis is Falco. It's the first Hadoop-based tool for single cell analysis, but it works only on upstream. That's feature quantification and alignment. And then there's ASAP. which is an R-based and web-based portal for single-cell transcriptomic data. But the limitations continue to be there is no scalability, and large-scale data handling is still an issue. These were addressed by ScanPy, but the ScanPy also has the issue of using the HDF5 for data storage, which has the limited functionality. And then there is ISE, an R-based software package, and it still has the issue of taking the cell load of only 50,000. So that led us to a question that what can be done for handling such a big amount of data. So we use Apache Spark for that. Now, what is Apache Spark? Apache Spark is an analytical engine made for handling big data, which is being currently used by the social media giants like Facebook and Twitter. And it provides an essential parallel processing platform for large data sets, and it has the it works on a resilient distributed data set, which are read-only collections of objects, and it leverages the RAM. That is, it's an in-memory computational framework which brings all the data into the RAM and processes it from the RAM. This is the typical workflow of the Apache program. It starts from the master, and there are the cores or the nodes we use as the executors. So for this study, we used the brain cells of a mouse having 6 subtissues, and the data set was having the size of 3,400 cross 24,000 cells. That we used an Ubuntu OS 20.04 and 16 GB RAM, i7 processor. These are the hardware, and this is actually the framework we have actually been able to develop for the large-scale data handling. So we start with the input and we melt the data. So actually for data sets, there are 2 types of data which we consider, the tall and the wide. The wide is having larger columns, multiple, more than 1 million columns. We melt it and we just put the data in a single column. So that the reason for that is we actually have the Spark. The Spark actually works better with the tall format of data. We then generate a cell and gene quality matrix, and we filter the matrix and use Parquet. Now, this is for the first time anybody is using the Parquet. Parquet is in Apache open source storage format, which actually uses the shredded shredded analysis algorithm for storage. And then we normalize the data on the basis of 2 normalizing methods, global and quantile, and again save it with Parquet storage format. Then goes for the further downstream analysis. That is, we reduce dimension and visualize the data. So this is how the workflow is of our pipeline. I think I'm losing the time, and yes, do I have the time, sir?
Go ahead. Yeah, you can go ahead. Yeah.
You have taken more time, but please finish. It's interesting things. Please finish it.
Okay, so we actually we start with the transposition of currently our tool supports a cell cross gene matrix. Now that can be resolved with transposed data. If the data is in cell cross gene format, then we need to transpose that. And if it is tall or wide, it can be decided by the user, and we can make it tall and then pre-process our data. That is the removal of the unwanted characters. Like Spark does not understand the dots and dollar signs of the data set. We just use this opportunistic type of character, that's underscore, for the pre-processing, and then we generate the quality matrix. It's based on 24 measurements. These are the dropout percentage, external spike in percentage, mitochondrial percentage, and other variety of measurements. It then moves to the stage 2, where we normalize the data. And for normalization, we are using 2 types of normalization. We actually followed 2 types of normalization methods which are being currently used. That's global normalization. It was used by both ScanPy and SURAT. So we thought it would be better for the user to use the simplest normalization method, and the other one is quantile normalization. And then the pipeline goes for the second stage. It starts with the decision of making the filtering parameters for providing a filtering threshold for the filtering. We use a couple of decisions here, that if we are using mitochondrial filtering or external spike in filtering method, we decide the threshold and we give to the our pipeline, and we do this filtering. After that, it goes to the stage 3, which is which we are currently working on. We have to develop for the dimensionality reduction and other DG analysis, etc. So these are the results which we did on which we actually got after building our pipeline. This is the PCA plot, and this is the t-SNP plot, giving us a good amount of visualization here of the cells. These are again the results. These are actually on based on the default filtering method. We just use the dropout rate of greater than 90%, and normalization was used quantile, and this is the first run result of UMAP, and then the second run is like this. Again, this is the third run. Since UMAP is a neighborhood-based algorithm, we actually rerun the tool and try to find there might be a possibility of getting better results. But these are the results. And this is the comparative analysis of our tool with other three which I have mentioned. And this is DR Spark. It's actually showing a good improvement in the time, provided the resources are good. So for the conclusion, we have developed a big data enabled tool for biologists, which has fastest computation, fault tolerance, and highly scalable, embarrassingly parallel. Yes, these are some couple of references. Thank you. I'm open to questions.
Thank you very much, Asif, for this presentation. I think it's really a wonderful work you have done, and you have tried your best to present it in a short period of time. And so it's a time for questions, and I will invite the participant to ask any questions if you have. So till the time other participant can respond, Asif, you have used Apache-based applications. So are there any alternative to using Apache-based applications?
Yes, actually.
Yeah,
that's a good question. Actually, there are tools are being developed other than Apache Spark as well. Like as I've shown, there are R-based pipelines, there are Python-based pipelines, but there's a problem. The problem with single-cell RNA-seq data is that it is quite huge amount of data, and right now we are able to sequence on multiple omics layers. So it would be a better option for us to use Hadoop-based technologies, like which are already proven their metal in social media, handling the social media data. So that's why we use Spark only. That's for now. For the big data, there are no alternatives for other than using Spark. Or now there is a new technology called Apache Flink. Maybe in future there might be a possibility that people will be developing for tools for single-cell RNA-seq using Apache Flink. For now, this is the only solution. Thank you.
Thank you, Asif. I think we don't have any more questions. So thanks.
Yeah, I have a question.
Major John has a question. Please go ahead.
Yeah, so Adil, what was the size of your data set, and how did this Spark implementation made a considerable change in the terms of either the search or any ML based analysis?
Yeah, that's a good question. Actually, our data set was of around I think 2 GB. Since our computational resources were not that much, we had to do this on commodity hardware. So we used, I think it was of 2 GBs, and it actually the Spark has this ability to bring all the data from hard disk to the memory. So since we know that RAM is the fastest memory than the hard disk drive. It brings all the data into the memory, and then then the processor fetches from the RAM. So that means that the searching is quite fast. It might take only milliseconds of time. So yeah, that's it. And about what was the next part of your question? I'm sorry.
Yeah, was there any considerable change in the performance of like when we want to search the data set? Hello?
Yes. As I said, we are using Apache Parquet as a technology for the first time for single-cell RNA-seq data. Since Apache Parquet is quite faster than the CSV storage type of file, it uses a columnar type of storage, and for that columnar type of storage is quite faster than how we actually store the data generally. Like we have .csv files or H5ADs. So yeah, that has actually given us I have actually shown the comparative analysis of my project that our searching was quite it was quite fast.
Yeah, and one last question. And how many, like, what was the number of slave nodes that you implemented in the architecture?
That's we used 3 cores in parallel, three nodes.
3 nodes, and what was the RAM of each node like?
Each node was, we started with, we started with the question that there might be a possibility that sometimes the RAM would be, a constraint for the user. So we gave only 4 GBs of RAM for each core.
Okay. Thanks, Adil.
Thank you so much.
Thank you, Asif Fadil. And there are 2 questions from Shivam Pandit, and I request you to quickly answer these questions. The first question is, what is the standard error given by the suite while prediction? What is the standard error of prediction?
Actually, I haven't measured that. I'm really sorry. I'm afraid I haven't measured yet.
So please, I think you can take it as a nice suggestion. You can
that's a nice suggestion. I would be. Thank you, Shiva.
And another interesting question, which is really I was looking for such a question, is related to Galaxy. So can such type of for tool can be incorporated into the Galaxy platform or Galaxy portal, or any cloud-based application, or we have to use it in local system?
That's really a nice question. Actually, we can take. I'm not sure about Galaxy Portal. I haven't heard about it. I was not in the workshop actually. I'm sorry, but yes, cloud-based portal. Yes, you can take it to the cloud. You have Amazon Web Services. We can actually take it to the cloud. That will be nice for having the person having less amount of resources for that. And yes, for if currently it's for the local system, we are using the local system.
Thank you, Asif. And Asif, you can contact us after this conference. We are looking into Galaxy also, so we can help you to a possibility of letting you know that whether we can incorporate your tool in the Galaxy community platform or not. Yeah.
Yeah. Thank you so much. Thank you so much.
Do you want to have any question, Prash?
Yeah. Want to ask any questions? Just a quick comment, Asif. Thank you so much. So Asif, it's a great talk. What I was saying is, Dask and other parallelization modes could be embedded. So since this single cell technologies are just are in place, again it's also a good idea to benchmark certain data sets and come up with a simplest wrapper where you could give it for the public domain or to the community. This is what I was just I've just given a comment. Thank you.
Thank you so much, Prash. Yes, that's a nice suggestion. Thank you so much.
Thank you, Prash. Thank you, Asif, and thank you all the participants who were asked the wonderful questions, and also to those who have slept, I think. So we come to end of this session, and I request Professor Jayaraman to summarize the session with his words.
Thank you, Harpreet. Before summarizing, I would like to talk about Harpreet. Harpreet is, I forgot to introduce him to you. Harpreet is a very active faculty member of the PG Department of Bioinformatics at Ansharaj Mahila Vidyalaya, Jalandhar. Apart from being a very active core member of BioClues, he has also been elected as a secretary of the AP Bionet very recently, and is a very active researcher with multiple interests. Yeah. With this, I will go for summarizing today's first session. Yeah. In the first invited talk, Professor Antunes enthralled us with different exciting aspects of microbial ecology under. I'm sorry. Yeah. The first invited talk, Professor Niaz Ahmed, gave an excellent overview of the pathways of antimicrobial resistance, the evolutionary dynamics of antimicrobial resistance of bacteria and their functional repertoire, abundance and diversity of the antimicrobial genes and classes, and he gave several other interesting aspects of antimicrobial resistance. In the next invited talk, Professor Antunes enthralled us with different exciting aspects of microbial ecology under different environments, their physicochemical fingerprints, and fascinating details on microbial ecology of genomic aspects and analysis of overall biotechnology and education. I request both the invited speakers to please help young researchers to provide additional details of the research through email if any of the youngsters approach you. And in the oral presentations, the first presentation, Danya, in her presentation on in silico approach of blood-based differentially expressed genes and interlinked pathways in neurodegenerative disorders, analyzed GEO data sets and identified 2 common genes in 3 different diseases. And she also identified pathways of differentially expressed genes and potential biomarkers. As if in another exciting oral presentation, he talked on normalization framework for single cell transcriptomic data, and he pointed out the difficulties in the conventional tools and moved on to explain the Spark-based big data analysis for the workflow of single cell transcriptome data analysis, and showed very excellent lots of different visualization tools. Yeah. With this, I would like to thank all the speakers, both invited speakers and the oral presenters, for providing an excellent first session of the InBix-2021 conference. Thanks a lot. Yeah.
And Professor Jayaraman, before we close the session, I think we are heading towards the lunch break, and a quick lunch break rather. So it's a memorable day, it's a memorable session, it's a memorable event in InBix always. So I request all the participants to switch on their cameras. I request the technical team to allow the participant to switch on their camera so we can have a smiling photograph. So Pras, will you help us to take the screenshot? Because you have patented the technology of taking. You have patented the technology of taking screenshot.
Yeah, I don't know how to take that. Subbu helped me last time. I do not know how to put all of them in one screen. Maybe.
Unfortunately, I also don't know. Sugu, can you help us?
Subbu is not here. Subbu is not here. Maybe others. Yeah. I don't know how he did that.
Only host can do that one. There is an option called Together More.
Let me try.
Together More. You need to click on that one, so everything will come in the same pane.
Together More.
Together More. Yes. Let me try. I'm trying it.
Yeah, adjacent to the share screen, three dots right.
Yeah, I've clicked there.
Yeah. Okay.
Together mode. I have.
Only host can do that one, not everyone.
Sugu, you can do that. Sugu has come. Yeah.
Yeah.
Sugu, please.
Sugu, please do that.
Yeah, Sugu, please need a quick photograph from you.
Yeah, we are actually doing that.
Yes, we're doing that.
Thank you.
Yeah, already done. Yeah,
Oh, you
thank you.
already done? We should smile, Please take again.
Yeah.
Go for one more.
Yeah, I'll take one more, please.
Okay.
One with smiling faces and one with thumbs up, yo. So there should be two, photographs, I think.
Yeah. Yeah, now one with the thumbs up, everyone. Only one, I'm seeing only one thumbs up. I cannot do it because I have to work. Yeah, thank you. Thank you so much.
Thank you.
The next will be the hungry faces.
So this is the InBix standard time. We are always on time. Absolutely great. Thank you so much, revered chairs, for organizing this. Thank you so much. Yeah.
Thank you very much. Thanks to all the organizers, the NEAST, the organizing committee, InBix organizing committee, Prash, Sugunkar, Professor Larhari Shastri, and Professor Jayaraman for giving this opportunity. Thank you very much.
Yeah, thank you, Professor Jayaraman sir and Harpreet sir for chairing this session 1 entitled Advances in Biotechnology. We had three invited talks and 2 oral talks. Now we'll meet again at 2 o'clock with session 2. Thank you, everyone. Go for quick lunch.
Thank you, sir. Thank you so much.
Thank you.
Yeah, thank you.
Hello. Hello.
Yes, sir. Hello.
Am I audible? I just wanted to check because I have a presentation in session 2. I just wanted to confirm that I'm audible.
Yes, sir. You are audible.
Okay. Thank you. Thank you.
Yeah. I logged in. Anytime you can.
Yeah. Yes. We can start. Exact. I think by 2 o'clock we can start.
Okay.
Dear all, welcome back. It's 2 p.m. IST. Am I audible?
Yeah, yes, Anil, you're audible, please.
Yeah, fine. Thank you. Yeah, hope all of you have completed a quick lunch. Now we'll move to the session 2, and the session entitled as Advances in Structural Biology. And this session will be chaired by Dr. S. Nagamani and Dr. Renuka Surojala. Nagamani and Renuka are available, right?
Yeah, we are available.
Yes, sir.
Yes.
Please.
Yes.
Yeah. Please take care of the session 2. Thank you.
Yeah. Thank you, Dr. Anil Kumar. And in this session 2, we have 3 invited talks, 2 oral presentation and 2 poster presentation. So the first invited talk will be delivered by Dr. R. Parthasarthi. So Dr. R. Parthasarthi, he is a principal scientist in CSIR IITR, Indian Institute of Toxicological Research. And he is leading BIRAC Bionist and also Environmental Monitoring and Intervention Hub in IITR. He has earned his PhD from CLRI, and after that he moved to US and he did a couple of postdoctoral studies, and he has more than 20 years of research experience, especially in applied biochemistry, to unravel important issues in human health and the environment. He has authored and he has published more than 100 publications, and with the high citations, and his index is around 45. And currently his lab is mainly working in the development of artificial intelligence and machine learning based computational toxicity models, and also computer-aided drug design, database development. And he has received several awards. And very recently, in the year of 2019, he received the Young Associate of Indian Academy of Science Award. And with this small introduction, I would request Dr. Parthasarthi to deliver his lecture on toxico-informatics, data-driven platform for chemical risk assessment. Sir, please go ahead.
Yeah. Thank you, Dr. Nagamani and Chairman and organizers and the participants. Very good afternoon. And thank you for the introduction. And first of all, I thank the organizers, particularly Dr. Sastry sir, for creating such a event, which is mainly focusing on informatics and bioinformatics approaches or computational driven methods utilized for R&D purpose, particularly in India. So this is a very occasion and event and timely it needs to be conducted. So it's like a great thought process and to initiating this kind of a event. And So today I'm going to talk more about our efforts. Currently, we are working in R&D directions in our laboratory in CSIR, Indian Institute of Toxicology Research. So as the name reflects, we are working mainly focusing on toxicology. Toxicology, it's like art of identifying the adverse impact. That's what it is, like how we can understand the adverse effect or impact of the lethalness using various tools and techniques. So particularly current this era is tremendous changes has been made, particularly this data-driven and informatics-based approaches. The toxicology is the core component is involved on predictive toxicology. That predictive toxicology driven by our computational approaches currently going to be help or aid in overcoming this time horizon related and research intensive process of in vitro and in vivo experiments. So that's to alternate to those things, we are currently working on developing a platform driven by informatics-based chemical risk assessment. So I would like to highlight here, like currently we are, why this toxicology is very, very important to understand this particularly chemical toxicology. We are surrounded by various chemicals, day-to-day activities of so many things. Without chemicals, we cannot think about it, our all day-to-day activities. So we are surrounded by chemicals, or we are using chemicals for every actions. So in this is pretty old data, but still that is what it's available. More than 60 million compounds were known to exist in CAS database of various pharmaceutical, agrochemical, household, and personal care products. As per the National Toxicological Program, they started collecting because of the concerns created on various toxicological issues, like including cancer and other diseases. There is a huge attention given to develop such an understanding of chemical toxicity, particularly long-term and short-term toxicity. For that, like there are 80,000 chemicals are registered, only 350 long-term and 70 short-term testing results, like testing endpoints are available. That's also took for one chemical, probably it may take 2 to 4 million over 3 years. It may take to conduct the entire toxicological suites of tests. So if you consider that scenario, if you want to do for the entire chemicals, it requires really, really huge amount of money investment, as well as resource, as well as the time. So in that scenario, like we definitely need an alternative approaches to elucidate this long-term and short-term testing models, particularly replacing this animal experiments. So UPA, US OPA is like made a resolution recently of eradicating this animal-based testing by 2035. And European Union also like currently working on to particularly how to replace this animal model, but they are very, very less success. Like UPA still struggles to replace animal model, particularly for pesticide toxicity. There is a lot of pressure on European Union also. They are also working towards how we are going to replace these animal models, not only for like I'll come back to that later. So particularly how we can improve upon our toxicological testings of various chemicals and products, process, or drugs, or and so on and so forth. So India also currently we are pushing towards developing alternative to animals model in various biomedical research. So all our countries, particularly all over the world, this huge concern is going on how we are going to replace this in vivo methods using these animal models. The challenges under these replacing animal models are like two major reasons, like animal model falter and other technologies are not ready yet. That I'm going to cover it in a couple of slides. So this is the first one. So the need for alternate for better health decisions, we are particularly using this in vivo models and in vitro models using animals as well as the experimental methods like non-animal models. So animal models, you have several concerns like pain, ethics, or ethical behavior, and all those things also required highly skilled manpower, time-consuming protocols, high cost. So these are the kind of a data available in 2015, not non-animal alternatives, like there are how many tests as conducted, but the main reason is how much they are accurate. The accuracy level for these in vivo methods are only 5 to 25% if you consider these rodent models. And for based on this understanding what they developed the in vitro models, that accuracy is still further higher. But these are all very, very particular concerns. Like we cannot develop completely, we are not success at how that's human relevant. So the Richard Klusner, he was a former director of US National Cancer Institute. He once told that the history of cancer research has been the history of curing cancer in the mouse several times. And we have cured mice of cancer for decades. And it's simple, didn't work in human beings. So that's his viewpoint on what is the status of comparing this test of toxicology testing of extrapolating from in vivo in vitro to human system. There are 6 pack tests of animal tests currently has been replaced, and currently there is no animal models are currently used for fragrance and some kind of flavors. US protecting agencies working to replace in vivo models for non-animal alternative for particular some of the cosmetic items mainly. There are 6 tests kind of acute dermal, acute oral, acute inhalation, eye irritation, skin sensitization, and has been periodically is getting replaced in 2016, 12, and 2018. So if you look at those 6 pack tests, there are several endpoints need to be analyzed. It's really, really huge, humongous task. There are so many endpoints, so many mechanisms, and really, really a tough problem. So how we are going to handle this of replacing these animal models? So there are initiatives started going on like 3R, like refine and replace and reduce this animal model. There are some systematic and integration of various platform. The idea has been emerged. So in that scenario, it's a kind of a cycle process, like how we can going to reduce this cost, time, efforts, as well as how to increase upon the precision, accuracy, and reproducibility. So that's a major thing currently working with how we are going to develop these models. So if you consider this in vitro methods, some of the methods are like lab on chip or organ on chip, organoid models, or like other in vitro models are combined with the in silico approaches, particularly data-driven informatics-based approaches, genomics, transcriptomics, everything now it's considered as a big, the informatics play a huge role in those elucidating those test methods. So we are integrating with in silico methods with in vitro methods. And finally, that going to refine and reduce these animal models. This is the way the 3R concept, they started working on it. So there is some matrices behind it, what kind of benefits we are going to get reducing the animal models as well as increasing upon accuracy. This data shows from EPA, they clearly indicate like keep on reducing these animals used for the some of the test items, some of the testing methods, particularly acute dermal and some eye irritation, skin sensitivity studies. It's showing a clear trend of reducing the animals, reducing the cost associated with the testing. So when this cost associated with the testing is reduced by using this in silico approaches, we call them as a non-testing methods. These are low cost, high throughput process. This also provide mechanistic understanding. Some of the experimental methods sometimes can't provide the molecular level understanding how this mechanism is working on. So in silico method have additional advantage of providing the using quantum mechanics, molecular mechanics, or system biology approaches, whatever it is, they also going to provide you much more mechanistic understanding, which cannot be come with your in vivo or in vitro methods. So that has another advantage. Another one is like within the shorter time, if you have a precise chemistry understanding of the system, you can redesign or re-engineer some of the substances present in the chemicals or some of the moieties. You can clearly replace them because those are all having the alert for the higher toxicity potential. So that can be also replaced using computational approaches. So this is the overall kind of summary what are the advantage of using in silico models. And there are a very interesting study. It's published in I think a couple of years ago. They made a huge compilation of 10,000 chemicals and associated with the around 8 lakhs studies. So they made a analysis using a machine learning approaches and animal repeat test. That test indicate that the machine learning approaches predictive methods are highly accurate compared to animal repeat test, and also it also having a better accuracy than in vitro models. Though this showing clear a more positive sign. We can take it forward using in silico or computational approaches to beat this animal testing. At CSIR IITR, we are mainly working on 3 major verticals. First one is developing this alternative platform using clear understanding of the molecular level phenomena of how these toxic substances, I cannot classify them toxic substances of any molecule or compounds, how it's go and interact with the biological system. And that is also reflect on a very, very specific study. So we are also developing a model in which it can robust way it can predict. So we are working towards developing the platform system for toxicological predicting, and also we are providing services to the industries. And currently there is a few programs we established at our toxicoinformatics research, as well as CSIR IITR on translating our research into technology development and supporting entrepreneurship, startup, and industries. So this is what I was talking about, the alternate toxicoinformatics based alternative to animal models, like how we are going to understand the interaction and predicting toxicity of the biological system using various approaches like multi-resolution approaches. And we are integrating with bioinformatics and genomics and to connect with the adverse outcome pathways and identifying early biomarkers in case how we can do some predictive biology or predictive toxicology in that cases. For developing a robust prediction, we are utilizing this chemoinformatics like QSAR based approach, QSTR based approaches that we are right now integrating with various artificial intelligence machine learning tools to have a very robust predictions. So that's what it is. Overall, we are working towards generating the knowledge and applying the knowledge and how we are going to utilize them in relevant applications for support the hazard and safety assessment, prioritization for chemicals and risk assessment, and replacing this in vitro model, as well as integrating with them to make it from prediction to protection. So now I'll show you a few examples of the ongoing work, what currently we are working on. So this is one of the work published in special issue of Theoretical and Computational Chemistry. So in that work, first we thought of developing a genotoxicity models for some specialty chemicals. We collected some 100 around specialty chemicals, and we looking for some of the predictive tools, like it's a Vega platform. There are 4 carcinogenicity models are available, 4 mutagenicity available. So this is any participant, they can easily do from your end. Like, okay, take the molecules and take the QSAR models. If you apply this using this Vega or some of the tools, you will get some. If you give any input, you will get some output. So when we started, okay, what are the best models available for genotoxicity predictions? That's why like genotoxicity is like the chemical have the tendency to damage their genetic materials. That's called genotoxicity. And carcinogenicity and mutagenicity are the part of this genotoxicity. So using experimental methods, there are few like bacterial mutagenicity test or comet assay people are using it. So we want to compare them with some of the reliable experimental tools. So that's where we started this work. So when we looked at the literature, there are 4 models available of Vega and Vega platform of carcinogenicity and mutagenicity. So we made an predictive, started applying those 4 methods, but we started getting some haphazard results. We don't know which method, which model are working good or having high specificity or sensitivity or accuracy. And that's why we made a benchmarking first to understand which model we can take it forward. So for that, we have selected around 4,000 compound from cancer some databases. It's already existing in CPDB and ISS scan. And those 4,000 chemicals we made into 2,000 of refinement, clean them up, and then we have taken these 2,000 chemicals and applied to these selected 4 models. Based on various classification, we went ahead with 5-fold or 10-fold analysis. So that reflected help us to identify the better models. Hello. Can someone mute this?
Yes, sir.
Yeah. Yeah. Okay.
Sir, actually, right now we are able to hear the echo from your side, I think.
Yes, not from me. I think it's some others.
Oh, okay. Now it's fine, sir.
Unmuted. Okay, perfect. You can see my slide. Okay. So using that ROC, which model is better? So based on the ROC curve, the model which is performing better, we have selected that and started predicting. And similar to that, we made a comparison with the experimental data available. And that also we made another overlapping analysis that provided a much better risk characterization of those chemicals. So that's what the summary of the work, like we made what generally available model. We not developed any model here, but we made a systematic benchmarking of validating those models and validating models, how we applied for the some of the known training set and test models. Then we can made a clear-cut prediction of those chemicals and comparison with the experimental data available. So this provide a good understanding for us to apply this informatics-based approaches or QSAR-based QSAR-based approaches for predicting genotoxicity of the specialty chemicals. Then there is another problem we applied for an additive manufacturing. Recently, it's a huge market and things are really getting really bigger in the sense like many of the materials currently getting used in this additive manufacturing process, particularly 3D printing methods. And biological components also used for various other purposes, but still there are a lot of materials like plastics, metals, ceramics, powders, liquids, even living cells are integrated together using in this 3D printing. And whenever the new technology, new product, new comings, as well as there is a risk assessment also evolving, and that needs to be evaluated with more critically. So in that sense, we started comparing a couple of additive manufacturing the material, particularly the acetonitrile butadiene styrene. It's a polymer used for 3D printing, ABS filaments. There are other PLA elements, some of the other elements also we have compared. But this study, we are mainly focusing on ABS filament. So one of my students, she made a systematic analysis of collecting the chemicals released of the heating of this ABS filaments. And they collected around 40 chemicals which are released from this ABS. And we made that characterization of those chemicals of various toxicity endpoints or toxicokinetics and pharmacokinetics or pharmacodynamics based studies. And then finally, we compared with various adverse outcome pathways. So this appeared in Chemical Research Toxicology. So if you see this graph showing here on the right-hand side, this PLA and ABS, there is a higher emission rate. That's where it's big concern for us. So we have taken this ABS model, ABS filament, and we collected the list of chemicals released upon from this ABS. And we predicted various toxicity endpoints like using QSAR, inhalation toxicity, oral toxicity, carcinogenicity, and using expert bit method, other endpoints has been predicted. And that predicted, not only the prediction gives us a better characterization of which chemicals or what are the things are going to induce this particular toxicity endpoints, and then we went back and carried out a pathway analysis of identifying the signaling as well as the protein associated with that, how they are going to interact and change upon these proteins to induce this end effect of which pathway is going to get overrepresented, then that can induce several other diseases or disorders. So there are very few studies available experimentally. People, I think, expose the workers exposed to these chemicals, like working in this 3D printing, 3D manufacturing factory, and they found out few acute infections like throat infection or lung infection, throat irritation. But finally, if it's a long-term exposure created various other key unexpected long-term disease associated with that, that's also we identified using this study. So further, as Dr. Nagamani mentioned during the introduction, we are developing a couple of databases, and these are the couple of databases published. We are working towards developing various toxicological databases, compiling them and making it using very robust for anyone can use to do risk assessment and hazard characterization. These are the two databases currently available in our group. Like FocusDB is like we compile like around 2885 food additives of various classes currently used in food manufacturing, and among which we also group them what are all the additives can be natural products, and others are like how we can avoid or replace them using those additives. And that we have collected around 4,000 data points and of experimentally available, and predicted around 90,000 data points, and with a very link to diseases and disorders or like physiochemical properties, like kind of entire data sets in one place we compile for food additives. So another one database is about polyphenol toxicity, which are used in dietary substances. That's also it's available, like entire range of toxicity endpoints and physical chemical properties are compiled, and recently it's appeared in food chemistry. So during COVID time, as part of CSIR consortia, we started working on repurposed drugs using computational approaches. So this one of the approach we have adopted during that time. So that time the HCQ, hydroxychloroquine and chloroquine gaining popular as a preventive prophylaxis drugs. So that also so we thought of, okay, when we looked at it, there's also having some kind of adverse effect of kidney or heart damages, patient with some kind of critical illnesses. It's causing some adverse effect. So we thought of developing an analog to the hydroxychloroquine and chloroquine, and using developing like structural similarities from a database from, I think it's not mentioned here. Okay, so from the databases as from like chemical libraries. So we find out what are...
Sir, actually, you have another 5 more minutes.
Okay, I'm done with a couple of more minutes. Yeah, that's it. So we, with structural similarity, we identified similar compounds, which resembles this hydroxychloroquine and chloroquine, and selected them, and we studied with various host as well as the COVID, SARS-CoV-2 such proteins. And we find out which are the chemicals having higher binding affinity. Further, we have taken up molecular dynamic simulation. Here we combine both docking and screening, as well as here we also did some acidity and basicity calculation of potential of how these molecules behave similar to that, not only structural similarity, how these properties also behave in a similar manner. That's what we have carried out in this study. And during the time, we started compiling various plant-based bioactive components, and which can be used in various stages of COVID-19 treatment. And in that, we identified several chemicals. That chemicals also we carried out. There are host protein as well as the viral proteins. We found out these phytochemicals of some of the selected chemicals having a potential bind with both the host as well as viral targets. So these are the few aspects on COVID-19 studies, and we also work another biotransformation of various chemicals currently used in, like this is the BPA, predominantly used in various cases. So this we made this biotransformation, particularly the quinone metabolites derived from various biotransformative process, how it's go and interact with our biological system, and what are the mechanism involved. That we have integrated with various system biology based approaches and quantum mechanics as well as molecular dynamic simulation. This is also going to appear in soon. So finally, our idea is to develop a next generation risk assessment platform by integrating our various robust computational models and with in vivo and few in vitro methods, which it can possible to proceed with. So finally, I'd like to summarize here. There are greater advantage using our computational to predict toxicity and ensure the safe use and minimize the toxicity for human and health of environment as well as environmental species. So currently we are making an integrative study in which in silico play a dominant role, which can come in a parallel line. It can replace this in vitro and in vivo methods. And we also working with various developing a risk assessment platform, which can further help in regulatory framework of various agencies in our country as well as the world. And I would like to thank the team as well as the funding agencies. And thank you so much. Once again, I thank the organizers for this wonderful platform. Thank you.
Oh, thank you, sir. Thank you for your wonderful speech on toxicological research. So, I mean, due to time restriction, that I would like to take one question from the participant. So there is one student, she asked if there is only a 5 to 20% of success rate in the in vivo models. So why does that FDA have not replaced the in vivo models in the basic process of validation of the drugs?
This is like 5 to 20% of the clinical at the clinical stage. They are only that success. So you need to repeatedly using various drugs and models to do this testing again and again. That's what it is. So the 25 success or failure rates are huge when based on developing this animal models. This is a wonderful question, actually. Still, we don't have better alternative to animal models. That's the first reason still FDA couldn't replace the complete testing. At least some level of quantitative understanding or qualitative understanding we are getting using animal models. And particularly relevant to one of the toxicity endpoints of liver or like kidney, and definitely animal models you can use them, but it's not completely replace entire toxicity testing. And that if you compare that, the repeat dose of toxicity of the one of the paper I've shown you, that has repeat testing also provide lesser than the model created using in silico approaches. Yeah, there is long way to go to replace these models.
Yeah, thank you, sir. So one more participant, Ganguly, you would like to ask any question? You have raised your hand. If you want to ask, you can directly ask to the speaker. Yeah.
Hello, Ricky.
Good afternoon, sir. I am from Northeastern University, Shillong. Sir, first of all, I want to congratulate you, sir. It was really a very enlightening session. Sir, I have one query. I have seen the data you're showing some MD simulation data where, sir, you have represented like very nicely those graphs and all. What software did you use for the visualization of the data, sir?
Oh, I have to ask my student. Actually, this VMD, GROMAX.
GROMAX, okay, sir.
Okay, No other further question on behalf of InBix organizing committee and.
Can I make an entry? I'm Vibha Tandon.
Yes, ma'am. Yes, ma'am.
Yeah. So I have a. It was a wonderful talk, and I also learned a lot. And I have a comment rather than say there. See, this toxicogenomics is an area which is an alien still to a lot of people who are working drug discovery, because I would suggest that IITR should take some lead and conduct a lot of workshops on this area, because people do QSR, we do a lot of other things, but doing toxicogenomics using computational methods, it is still not very well understood by the people. And it's not that it is not through computational method, it is actually not understood by the because IITR is one institution probably who is working exhaustively on the toxicology area. So we keep in our say courses also toxicology courses and all those things, but we teach only basic things over there. And we are not able to even teach also properly, because you can say the trained manpower is not available there. IITR should work in this area and try to train more and more people, maybe train people like us by conducting more and more conferences and more workshops, which can be attended by other.
So well taken, ma'am. Yeah, we are doing it, but yeah, it's a kind of certain limitations are there. Definitely, point well taken. Definitely, will push more forward. Most of still in India, many of in other lab like kind of industry and all, they are trained by IITR of some of the animal experimental and toxicity studies. But still, there is a lot of things we can do. Definitely, we'll push it forward, ma'am. The study. Thanks for your wonderful advice suggestion. Thank you.
So thank you, ma'am. So on behalf of InBix organizing committee and CSA NIS, I would like to thank Dr. Parthasarthi for his wonderful presentation. Thank you, sir.
Thank you.
So hello, without...
Sir, there is a question from Rick Ganguly. I'm sorry to disturb you.
Yes, sir. We already taken. I mean, sir has already addressed.
Okay, thank you. Sorry.
No, you can give it back. If you have any other question, ask them to write, and we can offline we can discuss. Thanks.
Choose. See you.
Yeah, thank you.
So without any more further delay, we will move on to the next speaker. So our next speaker is Professor Kunal Rai. So he is a professor from Jadavpur University in Kolkata. So he is a recipient of various prestigious fellowships, including Commonwealth Academic Staff Fellowship and Marie Curie International Incoming Fellowship. And his main research area is QSAR and molecular modeling, with a specific application in drug design, property modeling, and predictive ecotoxicology. Dr. Kunal Rai has published more than 300 research articles with a Hutch index of 45, and he has also co-authored 2 QSAR related books, and he is the co-editor-in-chief for various journals, including Molecular Diversity, and he is the editor-in-chief for International Journal of Quantitative Structure Property Relationship, and also that Professor Rai has recently been placed in the list of top 2% science-wide author database of the world. So with the small introduction, now I would like to invite Professor Kunal Rai to deliver his lecture on a new similarity-based read across algorithm for the prediction of small data sets, case studies with nanotoxicity data. Sir, over to you, sir.
Good afternoon, everyone. Dr. Nagamani, can you hear me?
Yes, sir. We are able to hear and we are able to see your slides. Thank you.
Fine. So at the outset, I would thank Dr. Shastri for inviting me to speak on this platform. And my today's presentation will be on a new similarity-based read across algorithm for the prediction of small data sets, case studies with nanotoxicity data. Nanoparticles are very interesting group of materials because of their interesting properties, which is due to high surface area and volume ratio, and they have high reactivity. And because of these interesting properties, nanomaterials are used in different applications, in different industries, like pharmaceutical industries, in cosmetics, in solar cells, in ceramic industries, textile industries, food processing industries, and so on. But due to their small size, they can cross cell membrane, and they can be accumulated inside cell, and they can cause toxicity, including membrane damage, releasing of toxic ions, impairing enzyme function, and many of their toxic action are believed to be mediated through free radicals. And as already mentioned by Dr. Parthasarathy in the previous talk, in silico toxicity prediction is very important nowadays because we do not have sufficient resources to evaluate toxicity of thousands of chemicals against thousands of endpoints. So we must adopt some in silico models and alternative tools, avoiding animal experiments, so that we can fill the data gaps. And all these data gap filling techniques by in silico methods are also supported by OECD, breach regulation, and US EPA. Now, among nanoparticles, there are two major classes. One is carbon-based nanoparticles, and second one is metal oxide-based nanoparticles. Metal oxide-based nanoparticles, due to their inherent physiochemical properties, are applied in different areas, including cosmetics, textiles, paints, water treatment agents, solar batteries, and in catalytic converters. But it has been seen that these nanoparticles exhibit different kind of toxicity, including respiratory, cardiovascular, inflammatory responses in humans. But we do not have sufficient data on the toxicity and ecotoxicity of metal oxide nanoparticles. So it is important for us to understand the toxicity pattern and also mechanism of toxicity of nanoparticles, because they are being used in several industries and we are exposed to nanoparticles. For example, we use metal oxide nanoparticles in cosmetics, say like in UV filters. So we are being exposed with the nanoparticles and their toxicity. So this slide shows the mechanistic aspect of toxicity, cytotoxicity of metal oxide to E. coli, which involves electron transport chain, disruption of electron transport chain, membrane damage, radical mediated damage affecting nucleic acid and proteins, phototoxicity, etc. Metal oxide nanoparticles venture human system through breathing, gastric tract, skin, and they circulate via blood. But as I've already mentioned, that reactive oxygen species play an important role in mediation of toxicity of metal oxide nanoparticles. So we must have some tool to evaluate the risk of metal oxide nanoparticles, and this should be done more economically and more easily. As it takes a lot of time to do experiment, and it is also there are also ethical concerns for animal experiments. So alternative methods, basically no animal methods are preferred. So the in vitro techniques and in silico techniques are more suitable nowadays for risk assessment of chemicals in general and also for nanoparticles. In silico methods are time and cost effective. They support 3R principle: replacement, reduction, and refinement of animal experiments. They can be applied for virtual compounds even before we have the compounds physically in our hand. And these kind of methods are supported by different regulations and organizations like European Center for Validation of Alternative Methods, International Organization of Medical Sciences, breach regulation, US EPA, OECD, and so on. Now today I'll talk on read across, which is one of the in silico methods, but it is different from QACR, quantitative structure activity relationship. Read across is a prediction method of unknown chemicals from chemical analogs with known toxicity from the same chemical category. Read across methodology is accepted by REACH and US EPA, and it can be used for data gap filling. So it uses strategies like toxicity prediction from similar chemicals, which may be from 1 to 1, or it may be from 1 to many, many to 1, or many to many. So broad classification will be analog approach in cases when we have limited number of chemicals. So it may be so that from 1 to 1 toxicity prediction can come under this class. And when we have a sizable number of chemicals, then we can talk about category approach, where we can use a number of chemicals as the source chemical for prediction of a target chemical. So here this figure shows both analog approach, where there is 1-to-1 prediction, or there may be a category approach also, and it can be used for interpolation and for extrapolation. Now, what we mean by similarity? We represent chemical compounds in various forms, in various graphical representation, but the chemical information encoded within the chemical structure should be converted into some numbers so that we can do the numerical manipulation. These numbers are called descriptors. So we can give examples of simple descriptors which can be obtained from some experiments, like say log P value, partition coefficient, or pK value, which are obtained from some experiment, or we can derive these numerical values from the structure representation using different levels of theory. So descriptors can be obtained at different levels, 0D, 1D, 2D, 3D, 4D, and so on. So 0D is that is actually constitutional descriptor, the count, different kind of counts. 1D based on substructures or fingerprints, 2D based on graph theoretical representation, 3D considers three-dimensional representation of compounds. In case of 4D, we also consider the conformational aspects. And when we have the numerical values of the compounds available, which encode information about chemical structure, then we can apply appropriate feature selection and learning algorithm to derive some kind of model which can be used for prediction of new set of data. So it may be like this. If we consider a two-dimensional representation of a chemical compound, so these are the two-dimensional representation of compounds, and from these we can encode information in various forms. Say, for example, say number of carbon atoms, number of nitrogen atoms in at very low level. We can give this kind of information, number of oxygen atoms, or molecular weight, or some say 2D descriptor like say E-state index and so on. So we can have a large matrix of descriptors having information about molecular structures. Then we can apply different techniques like normalization, standardization, feature selection, and we can remove outliers, etc. And then we can finalize our table. Then we can apply appropriate feature selection and modeling algorithm, which may be either regression-based or classification-based. So this we do in case of our QACR analysis, what is called a quantitative structure-activity relationship. This similarity concept is at the core of read across, where we can consider that similar type of compound or compounds with similar structures will have similar kind of property or toxicity. So when we have only one determinant feature, we can use this kind of graphical technique and we can derive the slope from say two compounds or at least two compounds, and we can interpolate toxicity of some data point, or we can also extrapolate toxicity sometimes. But when you have a number of features, determinant features, then we have to go for techniques like say principal component analysis, which is a data reduction tool, and which can compress the initial number of descriptors or features into lower number of dimensions or principal components. And we can have a plot like this, PC1 by PC2, which can help us to show the compounds in different clusters, and from that we can have some idea about the singularity. Or we can use say self-organizing map, which is an unsupervised method, which unlike ANN, which does not use back propagation, and which uses neighborhood kernel function, and which can make clusters of similar compounds. And from that we can have a similar compounds in at a particular region map. Now, as it is important for us to understand the difference between read across and QSR. QSR is basically a statistical model building process where we have the target property that is being modeled, which may be either toxicity or activity or any other property. And we can correlate this response property using different descriptors or features using different kind of statistical tools. So the basic notion of QSR is the biological activity or property that we are going to model, or toxicity here in case of quantitative structure toxicity relationship, using different descriptors. And the simplest example will be a multiple linear regression model if we are talking about regression model. And here in this particular equation, x1, x2, x3, these are actually descriptors like log p or pK value or number of carbon atom or so on. And A1, A2, A3 are the corresponding regression coefficient. So this equation is based on statistical method of development of the model. And as we know, for any statistical model, there will be some statistical tests to know the reliability of the model developed. So we can find out the reliability and statistical soundness of a model from different metrics like the determination coefficient and explain variance. Now, determination coefficient does not take into account the number of compounds used in developing the model. So we can use adjusted R-square where we can use this information, the number of compounds used in developing the model and number of descriptors used. So that gives us an idea about the degree of freedom of the model. So we should have more number of data points to develop the model and one number of descriptors so that we can have a statistically reliable model. And we have other measures of checking the statistical quality of the model like variance ratio and standard error of estimate, where also we can see the use of these terms, that is number of data points and number of descriptors. So to have a reliable and statistically acceptable model, we should have number of data points high and number of descriptors low. But there are some endpoints where we may not have sufficient number of data points. In those cases, it is challenging to develop statistically reliable models because, as I mentioned, QSR is based on statistical model building, so we must have a good number of data points, at least 50, to have a reliable model. In addition to that, we have to validate that model using different techniques like internal validation, external validation. In case of internal validation, also we have this metric Q-square, which looks like the R-square or determination coefficient. Only difference is that here we are using either leave one out or leave many out predicted value instead of the fitted values or calculated values. And in case of external validation, also we are using our model for prediction of tested compounds which have not been used for model development, and we are using the similar kind of metric R-square Fred, where the expression is similar to R-square. Only difference is that here we are considering the predicted values of tested compounds with their corresponding experimental values, and also we are comparing the tested compounds predicted values with the mean of the training set. Now, when we have a data set having a very low number of data points, so as I mentioned that the statistical model building may be unreliable. So in those cases, we can use read across or similarity-based method, because read across is not a hardcore statistical process. So QSR is not suitable for small data sets. As I've already mentioned, that there are several endpoints for which we do not have sufficient number of data points. And nanotoxicity is a field where we see that the toxicity of nanoparticles, experimental derived toxicity values are limited. So if we want to see, say, the toxicity of nanoparticles against E. coli or some other cell line, we'll see that the number of available data points will be very limited, maybe in the range of 20 or less than 20. So it is difficult to develop statistically valid QSR models. And also we are unable to use a higher number of descriptors in those cases to allow sufficient degree of freedom. So read across is not a statistical fitting process. Its calculation is comparatively easier than QSR. Alternative tool for hazard assessment aimed at filling data gaps. And for nanotoxicity, as data sets are small, we can apply read across. Objective of the present work includes to develop an easier and efficient method for quantitative read across prediction for nanotoxicity and for toxicity in general. Quantitative toxicity prediction of very small data set using new method, comparison of the results with previously developed method, and development of an application, web-based application for read across predictions. So in this particular case study, we have used 3 data sets which were previously reported by some other group using their own method. And here we have used PLC50 value of metal oxide against human keratinocyte cells, PEC50 of metal nanoparticles against E. coli, PLC50 of metal NPs against E. coli under dark condition. And we have used the same features as they have used. I'm not going into details about those features because that may not be important here. Now the scheme, we start with our data set. In all cases, the number of data points was less than 20. Now, as we have to develop the read across, and we have also to show the quality of prediction, so we are divided into 2 parts. One is training set or source compound, and another is test set or target compounds. And here we have used 3 kind of similarity: Gaussian kernel function, Laplacian kernel function, and Euclidean-based similarity. And we have computed toxicity of the target compounds based on weighted average predictions. Now, weighted average predictions has been done based on the sorted similarity of the training compounds. For each query compound, we have found out similarity of the training set compounds, and then we have sorted, and the similarity we have measured based on one of the 3 methods that are listed here. And based on the sorted training set compounds, we have taken their toxicity value with the proper weightage, and we have predicted toxicity of the target compound. So this is the basic workflow. Now about Gaussian kernel function, this is the expression of Gaussian kernel function, where x and y are 2 vectors of length n, and this one is actually the L2 norm or Euclidean distance between the 2 vectors, and sigma is a hyperparameter which has to be optimized, and we have used different values of sigma like 0.25, 0.5, 0.751, in this way up to 2. Then Laplacian kernel function, which is given by this expression, where you can find this is the L1 norm, our Manhattan distance between 2 vectors, and gamma is a hyperparameter which is to be optimized. We have used different values like 0.25, 0.575, 1, 1.5, and 2. And third one is Euclidean distance-based similarity. In case of training compound, we have done prior scaling using standardization approach, that is value minus mean divided by standard deviation, and then we have computed we have computed Euclidean distance of individual compound to training compounds, and then also we have again scaled the Euclidean distance values between 0 to 1. and 1 minus scale Euclidean distance has been used as similarity. Now, using our reductors prediction, we'll get some values, but again, we have to compare these predicted values with the experimental ones to know whether the predicted values are good or bad. As I've already mentioned previously, we have shown the expression for R square played or Q square F1. This is a similar expression. This is the sum of difference between the observed values of test set compound from their predicted values, and this is compared with the sum of individual observed value of tested compounds to mean of the training set toxicity. So here training set mean toxicity is used as the reference, whereas in case of Q square F2, the mean of test set experimental value is used as the reference. And also we can compute RMSE using this expression. Obviously Q square F1 and Q square F2 should be higher value, should be close to 1, and RMSEP should be a lower value for a good quality of predictions. And one can also go for classification based prediction, though we are actually getting quantitative values, but we can classify the target compounds as toxic or non-toxic, taking the mean of the training set compounds, mean toxicity of training set compounds at the threshold, and we can classify them as toxic and non-toxic. And then we can also cross verify it from the prediction. And we can compute different kind of metrics like sensitivity, specificity, precision, accuracy, efficiency, and so on. Now, this software has been developed in Java, and this has been made available in this page. And it needs training and tested data as input in XLS format. And the user has to provide sigma and gamma values and number of similar training compounds, distance threshold, and similarity threshold. And the program generates two output files. One is the predicted response, and there is the sorted experimental values with respect to distance and similarity. So these are the snapshots of these input files. This is a snapshot of different windows while running the program. And the program output looks like this. This is the sorted series of compounds, and this is the biological activity prediction. You can see this is the prediction from Euclidean distance values, this prediction from the two kernel functions, and from the predictions one can see the Q square F1, Q square F2, and RMSEP values, and then compare different kind of predictions. Now we'll go to results. First is toxicity prediction by Euclidean distance based similarity estimation. As you can see, among the three data sets, with the 9, 8, and 8 compounds in the training set, using all training compound values as the source, the Q square F1 and Q square F2 values are like this. We see in case of data set 2, the predictions are not that much good, and in case of data set 1 and 3, it is just passing the required threshold of 0.6. Then we have gone for sigma optimization. So the previous one was done without with some constant value of sigma. In case of here, we have varied the sigma values separately for 3 data set. In case of Gaussian kernel, we see that Q square F1 is maximum when sigma is 0.75. In case of data set 3 also, Q square F1 and Q square F2 are maximum when sigma is 0.75. In case of data 2, the sigma, that is Q square F1 and F2 are maximum when sigma is 0.5. Then we have gone for gamma optimization in case of Laplacian kernel. And we see that in case of dataset 1, gamma equal to 1 shows the best results. In case of dataset 2, gamma equal to 2 shows the best result. In case of dataset 3, gamma being 1.5 shows the best result. So one has to optimize the values of these hyperparameters. Then we have gone for number of closed training compounds. Initially, we actually worked with all available training compounds, 9, 8, and 8 in 3 data sets. But now we have gone for whether all training compounds are really required for toxicity predictions. In these cases, we have taken number of closed training compounds as 2, 5, 7, and 9. And we see that in case of dataset 1, we find that the best results are obtained when the number of closed training compounds are 5. In case of dataset 2, the best results are obtained when the number of closed training compounds is 2. In case of dataset 3, the best results are obtained from when the number of closed training compounds is 4. This is also represented graphically, the effect of closed training compounds on quality of prediction. Okay, now we have gone for optimization of threshold. So previously we have worked without any threshold. Now we have gone for optimization of threshold, distance and similarity threshold. Now here we have not used a number of closed training compounds because as we are using threshold with respect to distance and similarity, so that is already taken care of. Now when in case of dataset 1, we see the best value is obtained when the threshold is set to distance being 0.4 and similarity being 0.05. In case of dataset 2, the best results are obtained from distance being 0.5 and similarity threshold being 0. And in case of dataset 3, the best results are obtained from equivalent distance 0.4 and similarity being 0. So these are also represented here with these graphs. Now using the best combination of setting, like for dataset 1, in case of Euclidean distance, the threshold being 0.4. In case of dataset 2, when you are using the kernel functions, similarity threshold being 0. And in case of dataset 3, Euclidean distance threshold being 0.4. So these are the setting that shows the best results. We have also seen the corresponding predictions quality in case of classification for the target compounds, and we see that the results are very good. In case of dataset 2 and 3, the all sensitivity, specificity, accuracy, precision, all values are 100%. And in case of dataset 1 is also close to 100%. Now we have compared the performance of our method with the previous method. This is for dataset 1. As I've mentioned, in case of Euclidean distance, we get the best results. Now, when you compare with the previous method, there were two previous methods. One was the quantitative read across here, and other was nano QS here. But as you can see, the Q-square 82 value is superseding with the previous values, 0.96 versus 0.74 and 0.83. In case of dataset 2, our the kernel function-based method, the Gaussian kernel and Laplacian kernel, the values are 0.91 in both cases, but that is superseding the values obtained previously by the quantitative read across being 0.80 and nano Q is at 0.83. So in case of dataset 3 also, we get a better result. We are with our method with Euclidean distance based similarity 0.95. In case of quantitative read across is 0.91 and nano QSR minus 0.20. As you can see, actually read across is performing better than QSR method here. You see the for QSR method it is negative value, and in case of our read across method the value is very good. So these are the graphical representation of the comparison of different validation metrics with the previous studies. Now I'll come to conclusion of this work. So here we have shown a new quantitative read across algorithm based on various similarity estimation techniques, where we have used Euclidean distance, Gaussian kernel function, and Laplacian kernel function with optimization of sigma and gamma values. We have seen that the best quality of predictions are obtained from 2 to 5 closed training compounds. So it is not required to use all training compounds. We can use 2 to 5 closed training compounds, because if we use more number of training compounds, more noise are introduced in the calculation of or prediction of the target compounds. So it is better to use less number of closed training compounds. And the distance threshold and similarity thresholds are required to be used. Suitable distance threshold will be 0.425 based on the case studies that we have done, and suitable similarity threshold being within 0.05, 0 to 0.05. And this tool is available in this particular web page. And this new similarity-based read-across algorithm easy to use, efficient, and no prior expert knowledge is required. And one can use input files, XLS input files, and the output will also be XLS file. So this is the graphical user interface in the website for this software. We acknowledge the financial help received from ACRB for this particular work. And thank you very much.
Thank you, sir. Thank you for your wonderful presentation, and you have completed on your time. So there are some questions, and I would like to take one or two questions. So one student she asked, with respect to the read across method, what is the concept behind the prediction? Is it structure based or ligand based?
It is obviously ligand based. It is based on similarity. As I've already mentioned, I have calculated 3 similarity based functions: Gaussian based kernel, Laplacian based kernel, and also Euclidean based distance based similarity. All these similarity functions are calculated based on chemical descriptors. Descriptors are opting from chemical structures, so these are ligand based approach. And this is purely similarity based approach, where we are not using in any statistical fitting process while developing the model.
Thank you, sir. So there is yes, sir.
The quality of predictions we are evaluating using statistical based approach, but for deriving the prediction we are not using any statistics. But to check the quality of prediction, we are using statistics using different metrics like U-square, F1, F2, RMSEP, and so on.
So there is one more quick question. So what is the optimum number of compounds one can use for developing QSAR model?
I have previously mentioned that for QSR we should have a considerable number of compounds because it is a statistical model building process. So it should be at least 50, and in some cases where if you have some endpoint for which you do not have a sufficient number of data points, but the endpoint is very interesting and important. For example, say you are developing model of some catalysis or maybe some nanotoxicity modeling. So in those cases, you can use say 30 number of compounds also. But if it is below 30, then one should go for read across instead of QSAR.
Thank you, sir. Thank you very much. And on behalf of this organizing committee of InBix-2021, I would like to thank you for your informative talk. Thank you, sir.
Thank you very much.
So next, I would like to hand over the session to Dr. Renuka to take over for the next speaker. Ma'am, please.
Yes, thank you very much, sir. And it's a great pleasure inviting Dr. Vibha Tandon for this invited talk. She is a professor at Special Center for Molecular Medicine in Jawaharlal Nehru University. She did her PhD from University of Allahabad under the guidance of Professor Krishna Mishra. She worked as research associate at University of Allahabad and postdoc at IIT Kanpur, Ambedkar Center for Biomedical Research, University Delhi as research scientist. Later she moved to Department of Chemistry, University of Delhi as associate professor. Later she joined JNU in 2014 as professor. She heads multidisciplinary labs at SCMM and JNU, where her group actively works upon synthesis of chemical compounds, biological activities, animal testing, computational modeling, and her field of research includes chemical biology, radiation biology, cell signaling, drug development, and study of their action mechanism based on genomics and proteomics.
development of antibiotics for virus-resistant strains. Yeah, she is also a member of Indian Science Congress Association, Indian Chemical Society, Indian Association of Cancer Research, National Academy of Science India, along with other scientific societies. She has published over 70 articles in reputed scientific journals with a H-index of more than 31. She is currently serving as a chairperson for WSOE Chemical Science Kiran program and DST, and also a member of DST Curie program. Thank you, ma'am. It's a great pleasure inviting you for this talk.
Is that my
Please.
slide visible?
Yeah, it is.
Okay. Fine. So is it fine now? You can see the full screen.
Yes, it is.
Okay. Fine. So very good afternoon to everybody, and I would like to thank the organizing committee of CSIR NEAST Zorhat for including me in this program, and and I would also like to thank Professor G.N. Shastri, who always remember me and include all his endeavors. So thank you very much for including me in this conference, because after becoming a PI or a teacher, you are more or less doing your work and you don't get time to hear to others. But when you become the part of such conferences, you learn a lot, because you're sitting there and listening some very good talks, and you come to know some very recent work by by a wonderful scientist of our country. So with all these things, today what I'm going to talk about is, and I would like to thank Renuka also for giving such a generous introduction. So first of all, I would like to tell you that what I'm going to present in next 25 minutes. I will be presenting about the one of the molecule which we have synthesized in our lab, and we will be trying to tell you that how this particular molecule we have taken from the we have developed it as a antibiotic. And first, although it doesn't involve much of the computational data, I have not involved it here because I thought a lot of people are already presenting the computational work, but obviously we have reached to this molecule through computational work. But I wanted to take it to the further, so I have not included the initial work, rather I have included more or less more advanced work along with this molecule. So I will show you the journey from a small molecule to up to the preclinical estimation, its target identification, and its mechanism identification. So I'll be showing you all those kind of a data. And more or less, we have been working on one of the bread and butter molecule of my laboratory is bis-benzimidazole. And the molecule which I'm going to present you today is also about the is also a bis-benzimidazole. So we are say showing, we are working on two major projects in our laboratory, and one is the development of antimicrobial agent, and the another is the development of a say radio protector to protect the normal cells while you're giving the radiation therapy to the cancer patients. So in both the projects, we have reached to a stage where we have to go to for the clinical work. We have already done the preclinical work, and we are trying to do, along with certain CROs, to do the clinical, to accumulate the clinical data. So now I'm coming back to my project, the presentation, the title for which I have submitted to you people. So all of us know that the more or less a lot of antibiotics which are available today, they are not working on the bacteria, and a lot of resistance is acquired by the bacteria. And that resistance, if you look into this resistant map, you can see over here that this is the condition of 2019, and I have taken only aminoglycosides resistance here, because aminoglycosides is supposed to be one of the molecule which is still working against the resistant bacteria. So you can see here that aminoglycosides are still again, we have the resistance even against the aminoglycosides, and you can see that almost the whole India is looking light gray, not a darker gray, but still the resistance is here. So if you look into that, what are the typical pathogens which are today known as a resistance pathogen? So Acinetobacter, Enterobacter, Enterococcus. So number of Klebsiella, Pseudomonas, Salmonella, E. coli, Streptococcus, Salmonella, and Staph aureus. So there are a number of say resistant bacteria available all over the world, and these bacteria is almost become, we are not able to handle them, because there are not much cure is available, not much molecules are available, and people say that the antibiotic pipeline has dried up. So this is another antimicrobial resistant surveillance system map. And here again you can see, our own country is in really the bad stage in terms of the AMR, antimicrobial resistance. So like this, if you go on, you will find that there are a number of antibiotics available, and every day one or two novel analogs of these antibiotics are being enrolled. But what has been found, actually, largely it has been found by WHO, and WHO has given a report in 2018, 19, 19, 20, and 2021, and they are giving 4 recommendations continuously there, that the reason for the quick resistance to the novel molecule is also there, that most of the molecules which are being which are being launched as an antibiotic, they are the analog of the older antibiotic. So bacteria already recognize that conformation, and it is easier for them to develop the certain the site specific mutations against those bacteria. So number one reason is that they want that we should have a completely novel structure if we want to bring a antibiotic. The second point which they have been talking about, that if we want to get a novel molecule, that particular molecule should be against a new target. So we have to find out novel drug targets in the bacteria, in the pathogens, and leaving whatever are the existing pathogens, we should look for the novel pathogens which are there. Another third thing they are trying to say, that if we are trying to give a molecule, develop a molecule, that molecule should be if we give it into the say multiple doses or continuous dosing of these molecules are being given, so that particular molecule should not develop resistance faster. It can develop a persistent bacteria, but it should not develop a resistant bacteria. So and there should be a journey between the persistence to resistance. So if you are trying to do develop any molecule against these pathogens, these are the few things which were suggested by the WHO, that you should take in consideration such things before moving ahead with any molecule. And I think the most of the audience which will be listening this particular say talk, more or less I'm very sure that you people, most of the listeners or maybe the audience are from the chem informatics or bioinformatics. They have no dearth of molecules. You have lots and lots of molecules, but it is really unfortunate that the journey from computational study to the wet lab study is not very easy, and more or less there is no proper say collaboration between the computational scientist and the wet lab scientist have been found. And in India, this is one of the most unfortunate thing, that we miss collaboration, and that is how we miss the train. And you can say that we are not able to do take the molecules up from the bench to bedside. So I would also request that more and more computational scientists should try to talk to the more wet lab people, and wet lab, and it should be vice versa, is so that the existing whatever molecules are being proposed by the computational methods should be we should be able to take it to the for the clinical utility. Now, when WHO released a priority pathogen list, they said that they have they have divided into 3 priority pathogens: critical priority, high priority, medium priority. So they said that Acinetobacter baumannii, Pseudomonas aeruginosa, Enterobacterius species, which are resistant to the carbapenem, and third generation cephalosporin antibiotic, they are the critical priority, and we need and the whole world need molecules against them. Similarly, they need high priority, they need molecule against the Enterococcus fecum, Staphylococcus aureus, and Salmonella species. That is vancomycin resistant, methicillin resistant, Staphylococcus aureus, and fluoroquinolone resistance. Similarly, the medium priority, Streptococcus species and Shigella species. You need to give the, like this, they have given, actually they have given 11 say strains. I have mentioned here the 8 strains. So the another one were Enterobacteriaceae, Klebsiella pneumonia, and E. coli. So they have given a few more pathogens which were included in these 3 categories only. So when we had the class of molecules with us, and we tested these molecules, our molecules, against these pathogens, and when we test, and we found that out of those 11 pathogens, our molecules were active against these 8 pathogens. So we started working further with these molecules, and which I'll be talking to you in further few slides. So that is why I showed you this list. And the particular target which we have chosen was a topoisomerase 1A. And more or less, everybody knows that the fluoroquinolones are one of the magical antibiotics, which were actually launched in 1962 as an nalidixic acid, which was later on, which was quinolone actually, and later on it was converted to the fluoroquinolone, and a whole class of fluoroquinolones were used, and fluoroquinolones actually target the topoisomerase, but they target topoisomerase 2, or topoisomerase 2 is called as the gyrases. And it has been found that there are a lot of mutations have occurred in the gyrases. So because of the mutations in the gyrases, fluoroquinolones are no longer effective in these pathogens. And so people thought that if we find out there are 4 kind of a topoisomerases are available inside the pathogens. Some of them have 2 topoisomerase, some have 3 topoisomerase. So those are called as the topoisomerase 1, topoisomerase 2, topoisomerase 3, and topoisomerase 4. So we thought that rather than looking at gyrase, if we look at the topo 1, and we should try to see that because topoisomerase 1 is also responsible for the replication of DNA. So we thought that if we target topoisomerase 1 rather than topoisomerase 2, it will be there will be no say mutation will be found there, and it will be easy. And then if this topoisomerase will be targeted, and we will find a molecule targeting selectively topoisomerase 1, not acting on 2 or 3, then we can fight with the resistant bacteria. So for that reason, what we did, that we have chosen E. coli topoisomerase 1, and we have cloned E. coli topoisomerase 1 and expressed in our laboratory. And as you can see, the E. coli topoisomerase 1 is made up of domain 1, domain 2, domain 3, and domain 4. And this is an active site of the topoisomerase. And here the drugs can come and bind over there. So, and there were some crystal structures were known, and there were some preliminary studies were reported by a group of US. And so we have taken lead from there, and then we started trying with our own molecule. I've shown you below the human topoisomerase 1 also, because I wanted to tell you that there was like not a similarity in the active site of E. coli topo 1 and human topo 1. So if you because the infection will occur to human, and if you target the E. coli topo 1, if you're targeting the bacterial topo 1, it should not kill the human topo 1. It should not affect the human topo 1. So that is why it is also always a challenge to make a molecule which should only affect the bacteria inside the human, and it should not affect the human. So that is why I have given you here human topoisomerase structure, just to show you that it has a little different structure, and the molecule which we have chosen did not show any activity against the human topoisomerase. So we want to claim it here that the molecule is safer for the humans. So there are as I told you, that lot of molecules are known against the gyrases, but against topoisomerase 1, only molecules are there which are up to the clinical stage, but they have never reached to, they are at the preclinical stage, but they have not reached up to the clinical stage. So there were a lot of molecules were proposed by different researchers, and there were another class of molecule was proposed by few researchers which have reached to certain stage, and GSK was working exhaustively on this particular molecule, which was known as gyrase, and which was working on the dual inhibitor. Like it was working on the two topoisomerases together. One is gyrase and another was topoisomerase 4. It has reached up to the certain level, and then later on it was found that it was not working on the biofilm making working bacteria, and it was also showing some kind of a toxicity which was causing a lot of concern to the people. So that is why it is still have the emergency use approval. It is not completely in the market. Some of its say derivatives are in the market. So now I'm going to on my own molecule. My molecule of interest is here, and its short name I've given PPEF. And what is the criteria of this particular molecule? I would like to explain it here. That you have a say double-stranded say DNA, and when the topoisomerase comes and sit here, it causes a single-strand cleavage, and then when there is a single-strand cleavage occurs, then there is a DNA strand turnover occurs. Means both the strands, they get exchanged to each other, so that there is a mixing of DNA occurs. And after the mixing, then what happened, that there is another term occurs that is called as the religation. So what is the function of a topoisomerase? First cleavage and then religation. So both the things are being done by the topoisomerase itself. So we thought that if we introduce an inhibitor which binds to the topoisomerase, and after the cleavage occurs, it doesn't allow the religation of the DNA, then we will have small strands of the DNA, and that is how the DNA will be decomposed, and those kind of molecules are called as the DNA poison inhibitor. So we have taken this mechanism, and we have done experiments with the PPF, and we have found that our molecule was actually a poison inhibitor. It was not stopping at the first stage, it was allowing the cleavage, but it was not allowing the religation. So there were a series of molecules we have prepared. I'm only showing you 4 of the molecule here. And basically, I'm showing you this molecule because this molecule is nothing else but an analog of one of the DNA binding dye, which is very well known to the biologist as a hex 33342. So it is a dye which is known for the DNA staining. So we have taken the lead from that particular molecule and created a library of 150 molecules. And out of those 150 molecules, we have chosen our molecule, which has shown the very less toxicity, but it was highly efficacious in the animal models. And we have till now we have worked almost with the 455 strains, and you can see here that those are E. coli, S. aureus, and S. typhimurium, S. flexneri, S. pneumoniae, E. fecum, and all of them are multi-drug resistant bacteria. We have acquired these multi-drug resistant strains from different hospitals, Safdarjung mainly, different pathogens from different, CDC USA, PHC England. So we have been getting the pathogens from all over the world. And we have got a collection of pathogens with us, and through that we have done this work in our laboratory, and we have collaborated with one of the CRO, that is the Anthem Biosciences. So now, how do you look at the topoisomerase inhibition? So topoisomerase inhibition is looked through one of the biochemical assay, which is known as the relaxation inhibition assay. So the basic function of topoisomerase is to relax the DNA. So as you can see that when you have this is a supercoiled DNA, and when you add the topoisomerase, it gets relaxed. So when you add our molecule, so when I add the molecule of interest in increasing concentration, you can see that the relaxation is reduced, and slowly only the supercoiled form of the DNA is visible over there. So this suggests that this molecule is acting against the topoisomerase, and it is inhibiting the relaxation activity of the topoisomerase. So here I have used the S. aureus topoisomerase and the Acinetobacter topoisomerases, and we have shown that our molecule is having an ability to inhibit the topoisomerase 1A of bacteria. Here we have done some work on the computational, we have done docking and MD simulations, and after that I'm showing you the poses here, that how the molecule is binding between the say E. coli topo and DNA as well as the molecule. So this is the double helix of the DNA, and this is the topoisomerase, and our molecule is binding at in this stage to the topoisomerase DNA complex, and tyrosine 319, which is involved in the religation, it doesn't allow tyrosine 319 to religate the say DNA, and that is how that is the reason that the DNA remains in the cleaved form, and slowly we get single stranded DNA and the small double strand DNA fractions, which are inhibiting the so that causes the death of a bacteria. So similarly, we also did, I have included this slide because I wanted to show you that because the title of my talk is about the unique gate dynamics. So as I showed you here that the topoisomerase have a typical structure. So it has a loop 1, loop 2, loop 3, domain 1, domain 2, domain 3, and domain 4. And people call it so it has been shown earlier by certain people. They have not shown it in terms of the drug discovery, but in terms of to understand the topoisomerase conformation and its function. People have shown that there are certain kind of gate get opened when the DNA comes and binds to there. I would like to show you here that if there is a E. coli topo 1A is like going on here. So you can see that it is in its closed form, and slowly you would see that it comes into its open form. But if you looked into the it will take time actually. So we have to wait over here. But the whole logic in this particular slide, which I want to tell you that the E. coli topo 1A has an say remains into the closed form and its open form. So in its open form, it binds to the DNA. But when PPEF, our molecule of interest, when it goes and binds to the topoisomerase, it doesn't allow the opening of the E. coli topo 1A. And as it doesn't allow the opening of the E. coli topo 1A, DNA itself is not able to bind there. So you can see it here, what is happening here. Now, if I show you here, here only DNA is there, so you can see what is happening here. And what we have also found that there is a certain energy of the closed form of the DNA, and there is certain energy of the open form of the DNA. And it has been found that from so here again you can see that how it is changing its conformation. But if you look at here, you will find please look at here. So you will again find that it is I have only shown you here single stranded DNA over there. So you can see over there that there is the opening is occur, but opening is not very large. But if you look at here, first, please look at here that you would see that whenever PPF is binding here, it will not allow the opening of E. coli topo 1A. So this was the technique which we observed, and we can go it. So this I would like to show you from here. So we have done the molecular dynamic simulation with this molecule, but it was not a simple molecular dynamic simulation. It was a little bit of a different way of doing it, and it was done by Dr. Naresh Patwari from IT Bombay, and he has used the well-tempered metadynamic simulations and using two reaction coordinate over there. And so if you look at here, I don't know whether it is visible to you or not. So you can see here that this is the closed state, the energy is this. And when slowly it comes to the open state, its energy is minus 15. So you can see here that energy, there is an energy barrier from the closed state to the open state. And when the DNA, when the DNA is bind, that this energy barrier is reduced. But when the PPF comes and binds over there, then this energy barrier is further increased a lot, and which can be explained over here from this particular say graph also. You can see here that topo 1A PPF is a complex which is showing the different energy, and when there is a topo 1A ssDNA, then the energy the protein residual energy is different, and this is a kilocalorie per mole which is over there. So you can again, if you like to see over here, that loop 1, loop 2. So there are two loops which are getting separated. So this is one loop here, and this is a loop 2. So loop 2 is getting separated out from the loop 1. So if you look at there, you can say that the topo 1A have this energy, and when you add the PPF, then the energy is further increased, and when you are having the topo 1 dsDNA, this energy is further reduced, and when you are having topo 1 dsDNA PPF, then what is happening? So you can see here that how beautifully we could find out that by the metadynamic simulations, that the gate open close configurational dynamics of topo 1 have three prominent minima. One is like there, which is completely a closed form. Then the second is there when there is a partial closed form or partially open form. And the third is which is the completely open form. So if there is a molecule which doesn't allow this energy barrier to be crossed, that particular molecule can be utilized as a topoisomerase inhibitor. So we have developed this model, and this is the first of its kind of a model where we can use other drugs also to screen them as a the topoisomerase inhibitor. In the pursuance of this, I think I don't have much time. I will directly move to the say animal data over here. So then what we did actually that we have done certain animal work with this particular molecule. So what we have done that after knowing that this has a good MIC against the bacteria, it is working. We have done the target identification in terms of the topoisomerase 1A. Now we wanted to see that how it is working inside the animal model. So we have chosen two infection models here. The first infection model was systemic infection model, where what we do that we do the infection in the bacteria in the animal in the thigh only, and then we have treated them with the particular say molecule PPF. And you can see over here that the colony forming unit which decides the infection in the thigh was in the control, it was like this. When we have added with 3 mg per kg, it was reduced further, and when we have used the 5 mg per kg body weight, and I have not shown here, and when we use the 7 mg per kg body weight, you can clearly see that the percentage survival of the animal has also increased. All the animals, untreated animal, died in 4 days, whereas the treated animal survived up to the 7 days and 8 days. Similarly, then we have created another model which is called as the neutropenic thigh model. What is neutropenic thigh model? Neutropenic thigh model is a model where what we do that we create a systemic infection. We create an infection in the blood of the animal, and in this kind of a model, what happened that the animal survive only few hours after the infection. So you can see here that when we have added PPF, the colony forming unit have reduced. We have added one efflux pump inhibitor that is also reducing the infection. But when we added the PPF along with the efflux pump inhibitor, you can see that the infection has reduced up to certain, say, several logs over here, which you can also understand from here that 1 log reduction, 3 log reduction, and even the 6 log reduction in the infection occurs. Please understand that these are the infections which we have done in the animals were again the multi-drug resistant bacteria of E. coli. The similar work we did with the S. aureus. We have chosen the methicillin resistant S. aureus, and we have developed the models again, and you can see that the levofloxacin was used as a standard antibiotic, and you can see that the levofloxacin at 4 mg per kg, the data with the levofloxacin was not that good, was better than the PPF, but when we have used the PPF 8 mg per kg, all the animals survived during this. This is called as the Kaplan-Meier graph. But in the levofloxacin treated animals, some of them were have not survived. Similarly, you can see that we have again done this in the neutropenic thigh mice model also. So we have done a number of, we have created number of say infection model, and we have tried to see and compare our molecule with the standard antibiotics in the multi-drug resistant bacteria. Then we have done the pharmacokinetics of this molecule. I don't have time to explain this. So in summary, you can say that we could develop a molecule which was having a new drug target, and that molecule was working on the gram-positive and gram-negative bacteria both, and it was active against the multi-drug resistant bacteria. And we have done some mechanistic studies also, which we have not which I have not shown you here, that how it actually inhibits the topoisomerase. I didn't have time to tell you about that. So with all these things, I would like to thank you. And we are now collaborating with Anthem Biosciences for for the all toxicity studies and IND enabling studies. We are Dr. Padma Devarajan is doing formulation of this molecule. Ruchi Anand is trying to do the structure activity. She is doing the crystallization studies because she is a crystallographer. And our clinical say advisor is Professor Dr. Rajni Gan, who is the head of department of Safdarjung Hospital microbiology department. So with all these things, we have I would like to thank you for giving me time, and I work here. This is a place, Special Center for Molecular Medicine. This is a small center in the JNU, and these are the few of my collaborators and funding agency. Thank you very much. I'm open for the questions.
Yeah, thank you very much for this informative talk. And yes, we have some few questions like how did you arrive at this compound? Any in silico screening was performed?
Yeah, so as I told you in beginning itself that yeah, we have done in silico screening, but my main observation was this, that we knew that this particular molecule, one of the analogs of this molecule was human topo inhibitor. But please realize that we wanted to develop a molecule which should not act on the human topoisomerase, rather it should act on the bacterial topoisomerase. And then our priority was, another priority was there, that the molecule, the parent analog of this molecule was highly toxic and highly mutagenic and clastogenic in its nature. So the another priority of our molecule was that we can create a molecule which should be less toxic, less clastogenic and mutagenic. So we have done certain QSR studies and SAR study, but I did only on the analog of this molecule only. I did not find this molecule from any library. I took this molecule from the wet lab data and tried to develop my own molecule.
Yeah, that's wonderful. I would like to thank for this wonderful talk, and it's very knowledge gained talk with respect to various approaches you have used in this developing new novel drug. And yes, I would like to take the opportunity for Dr. Nagamani would like to take over the session with the oral talks. Thank you.
Okay, so thank you very much, and thanks once again. If anyone have a question, can send me by email. Thank you very much. Okay.
Thank you. And so next we will start with oral and poster presentation. So in this particular schedule, we have 2 oral talk and 2 poster talk. So the first oral talk will be delivered by, I think, Rohitas Yadav and his team. And the title of the oral presentation is E-pharmacophore based screening, docking, dynamic simulation, and DFT analysis of novel proteasome inhibitors for multiple myeloma.
Thank you.
And Yadav, you are available, Rokite?
Yeah, yes, sir. Thank you, sir. Thank you, sir.
And please stick to the timings. You have 3 minutes to present.
Please
Sure,
share
sir.
the screen as well.
Is it visible, sir? My screen.
Yeah,
Hello?
you please make it full screen.
Okay. Okay. So, so good afternoon, everyone. And I would like to thanks to the organizing team for giving me this opportunity to present the research work on this platform. and myself, Rohitas Yadav, research scholar in Department of Pharmacology, AIIMS Rishikesh. So my research title is e-pharmacophore-based screening, molecular docking, dynamic simulation, and DFT analysis of novel proteasome inhibitors for multiple myeloma. And so next I'm coming on the introduction and rationale of this study. So as we know, multiple myeloma is the second most common hematological malignancy of plasma cells, and the 20S proteasome, which is a very most important target for designing the drug against the multiple myeloma, which is a ubiquitin dependent proteolytic system, degrades the misfolded proteins and maintain the cellular homeostasis. So so the bortezomib is the first FDA approved drug against the proteasome, and the bortezomib treatment often achieve brief response, and it is frequently associated with drug resistance. So after a long time efforts to develop the proteasome inhibitors, effective and safe drugs are still missing. So thus, therefore, this present study deals with in-depth pharmacophore-based screening, drug repurposing of FDA-approved drugs that could be target the PSM beta 5 for multiple myeloma therapy. So next, coming on the workflow of this study. So this is the complete methodology part of this study. So in this study, we have retrieved the 1,379 FDA approved drug molecules from Zinc database, and these compounds prepared by ligand preparation module of Schrodinger software, and then these molecules are further used for phage-based virtual screening and molecular docking. So for the molecular docking, the 20S proteasome, the crystal structure of 20S proteasomes are downloaded from the protein data bank, and then protein structure were prepared using the protein preparation wizard, and the PSM beta 5 chain were selected for molecular docking. So in the docking procedure, two-phase docking procedure were applied in this study. One is the standard precision docking, and another is the extra precision docking. So we have reduced 138 final drug molecules in through the molecular docking, and among 138 molecules, best two drug molecules were selected on the basis of docking score as well as the ADMET profile of these drug molecules, and then these molecules were used for molecular dynamic simulation. So next, coming on the results of this study. So these are the two final selected molecules, risedronate and zoledronate, and these are the structure ligand protein complex structures, and so these are the ribbon structures. So ligand are complex with the target proteins, and this structure is showing the ligand protein in physiochemical interactions of ligand protein. So same, these figures are showing the ligand protein interactions. So next, coming on the results of the MD simulations. So these graphs showing the RMST, RMSF, radius of gyration, ASA. So the MD simulation results reveal that these ligand protein complex were stable till 100 nanosecond simulation time. And so next, coming on the proposed mechanism of these potential ligand compounds. So so basically the proteasome, this is the structure of proteasome. Proteasome are basically responsible for degradation of misfolded proteins, misfolded and unfolded proteins in the cells. So when the misfolded protein complex with the ubiquitin, and then these misfolded proteins are degraded through 20S proteasome subunits. These proteasome subunits are basically having the catalytic property, and these are consist with 4 chains, 2 alpha and 2 beta. So basically beta chains are further consists with 7 subunits: beta 1, beta 2, beta 3, beta 4, 5, 6, 7, and so beta 1, beta 2, and beta 5 are basically responsible for caspase-like activity, trypsin-like activity, and chymotrypsin-like activity. So in this study, we selected the PSM beta 5 target proteins, and this subunit is basically responsible for chymotrypsin-like activity. So these two molecules were selected against these target proteins. One is risedronate and another is zoledronate. So when these molecules will bind with these target proteins and block the activity of protein degradation, misfolded protein degradation. So when these misfolded proteins are accumulated in the cells, and further they are responsible for increase the endoplasmic reticulum stress, and when endoplasmic reticulum stress increase, then these are responsible for cell cycle arrest and apoptosis. So this is the proposed mechanism of this study. Next, coming on the conclusion of this study. So the molecular docking based investigation revealed that risedronate and zoledronate demonstrate the high binding affinity, minus 9.51 and minus 8.67 kilocalorie per mole docking score respectively with PSM beta 5 target protein. And the MD simulation analysis for 100 nanosecond also showed that risedronate and zoledronate exhibit the better binding free energies and stable interactions with PSM beta 5, hence can be promising proteasome inhibitors for multiple myeloma. So this in silico docking study would be helpful to further in vitro validation and development of new pharmacotherapeutic drugs for relapse and refractory multiple myeloma. So I'll acknowledgement research facility available at Department of Pharmacology in Rishikesh, and I would like to thanks to Dr. Prajwal Nandekar and Sarojini team for their cooperation and software. Thank you everyone. Hello.
Oh,
Hello.
sorry. Yeah. My
Yes,
mic
sir.
was mute. Yeah, thank
See,
you for your
I have
presentation.
finished. Any questions?
Yeah. So I have some few doubts. Actually, you have mentioned 2 drugs, and in the graph it was showing 3 graph. So what is the another one?
Simulation graph, sir.
Yeah.
Actually, one is the APO form of the single protein simulation graph, and two is the ligand protein complex graph for comparison. So this is the only protein simulation graph, and two other is the ligand protein complex.
So which one is the upper one?
APO one is the blue one, sir.
Okay. And what is the difference between this pharmacophore and e-pharmacophore?
Actually, if pharmacophore is the basically the compound or molecules that is responsible for pharmacological activity, but in the e-pharmacophore is the energy-based pharmacophore model. Developed the model on the basis of energy or chemical characterization of the molecule, like similar structure based. Like
Okay.
hydrogen bond donor, hydrogen bond acceptor basis on that.
Okay, and one more student, she was asking, and she wants to know, I mean, which software you have used for this pharmacophore modeling? She just wants to
Sir,
know.
Schrodinger's phage-based pharmacophore model development, sir. Schrodinger software, Maestro software, sir.
Okay. Okay, thank you.
Thank you, sir. Thank you so
Thank
much.
you, Rohit.
Okay, sir.
So we will move on to the next oral presentation. So I would like to call upon Rick Ganguly and Atanu Bhattachi. So the title
Am I
of
audible?
their, yeah, you are audible. And the title of their oral presentation is Enrichment of Pharmacokinetic Characteristics of UK122, a potential inhibitor for urokinase type plasminogen activator.
So my
Yeah,
slides are
please.
visible.
Yeah, you just go to the first slide. Yeah, fine. You please go on.
Good evening, everyone. I am Rick Ganguly from Department of Biotechnology and Bioinformatics from Nehu, Shillong. And I'm very fortunate that I got the opportunity to talk about my topic today, that is enrichment of pharmacokinetic characteristics of UK122, which is a potential inhibitor for urokinase type plasminogen activator. So urokinase plasminogen activator is a plasminogen protein, and when it binds to its receptor, which is UPAR, it basically initiates a series of proteolytic cascade that degrades the extracellular matrix component of atherosclerotic plaque, which gradually makes the atherosclerotic plaque unstable. And UK122 is a reported standard compound for this UPA. As an inhibitor, it is known. So UK122 has also some serious solubility, toxicity, as well as pharmacokinetic issues. So we tried to do some series of modification or changes with the help of some pharmacokinetic descriptors that resulted in a novel compound, C15, that has higher solubility, less toxicity, and does not interfere with some, certain cytochromes, hence has less likely have a chance of any physiological effects. So one of the greatest statisticians of 20th century have said, essentially all the models are wrong, but some are very useful. So many of the lead compounds that get discarded in the process of drug discovery are unremarkably, they failed in the process of pharmacokinetic procedures and pharmacodynamic characteristics. This has hindered several drug discovery processes. From 1964 till 2000, the majority of the clinical trials failed due to these reasons. And due to the computational use, this failure has reduced down to 5% from 40%. That is a remarkable change. So the objective of this small part of the research that we have conducted was to enrich the property of UK122 against urokinase type plasminogen activator as an inhibitor. So these are the methodology that we have used. We have initially checked the quality of the target molecule, that is UPA, and we have resolved the structure at a resolution of 1.6 Angstrom from PDB. We have used several servers, chemoinformatics servers such as SwissAdme, which I have majorly used, which is a free software, and we can use so many different parameters we can use in that and consider those parameters. We have used mol inspiration server. So we have generated several structures out of the taking the template UK122, and we have screened those compounds based on their physiological properties, lipophilicity, solubility, pharmacokinetic properties, and drug likeliness and medicinal chemistry. Then I have used a particular chemical search space in SwissAdme, which is boiled egg. So to detect the different positioning of those compounds, whether it is present in the gastrointestinal tract or whether it has a probability of crossing blood-brain barrier from the preset model that they already have in their database. And there are chances of any compound to become a PGP substrate or a P-glycoprotein substrate, which is a part of the efflux system that throws out any compound outside the cell. Osiris Data Warrior I have used to check the toxicity of those compounds. I have used GOLD algorithm. So GOLD algorithm have a preset of genetic algorithm, and it gives a scoring function in terms of GOLD score to show the molecular interaction between a receptor and a small molecule. So UK122 was used as a receptor, and we have checked the molecular interaction with C15 and our standard with the receptor. With UPA, we have checked the interaction with UK122 and C15, and those protein ligand complexes were visualized using Python Molecular Viewer or PyMol. So this is the structure of UK122, and this is the search page of SwissADME. It shows a bioavailability radar with so many different parameters. And here we can see that the pink region is the acceptable region or the ideal state of the parameter that it should be. And in saturation, it is having a saturation which is not fitting inside the favorable region for UK122. It has slightly or moderately solubility issues, and this pharmacokinetic properties under which there are so many different parameters are listed, and it binds to one of the cytochrome, that is CYP1A2. It is throwing certain pain and brink alerts also. So pain alerts are basically pan-assay interface compounds, so which shows that any compound can have multiple targets, so the specificity is less, and there are certain structural alert based on the structural component of the ligand. So this is the overall chemical evolution that we have performed with the help of SwissADME, where we have started with initially the SMILE format of UK122, and the structure from the structure we have done certain modification, and the successive changes were named as C1, C2, C3, and so on till C15. And below we can see the bioavailability radar, where from the starting point where the saturation was out of the accepted region, it is coming into the fitting into the bioavailability radar. So this is the goal result for molecular interaction between UPA and UK122, which is giving a goal score of 57. As compared to that, the goal result of UPA with our idea, like the new compound C15, is giving a goal score of 54. And these are the list of hydrophobic and hydrogen bond interaction, which is showed in a 2D format, where all the green dotted lines are the hydrogen bond and the asterisks are the hydrophobic interactions listed here. So this is the chemical search space of SwissADME server, where the starting point where we have started with UK122 fits in this white region. So the yellow part of this search space, this is called a boiled egg search space, where the yellow part represents the yolk, is the part where the compounds which are within this part have a higher probability to cross the blood-brain barrier. And the white space is basically the probability of these compounds are higher to be absorbed in the gastrointestinal tract. So for an orally available drug, we actually prefer the compounds to be residing within the white zone. And from the starting point till the final compound, it is fitting within the white zone, which means that these compounds have higher probability to be absorbed in gastrointestinal tracts. So these are the conclusion, where initially the UK122 had an affinity or of binding to cytochrome P451A. So P451A is a component of cytochrome P450, which has a mixed function oxidase system that plays a major role in the metabolism of xenobiotic entering the body. As compared to UK122C15, the new molecule have showed no involvement with that cytochrome. The saturation level has shown that UK122 was found beyond the favorable range of, as compared to C15, was found within the acceptable range. While studying the in silico toxicology studies with the help of Osiris Data Warrior software, we have seen that UK122 has certain mutagenic properties. As compared to that, C15 have overcome that adverse effects. Molecular interaction studies with the help of GOLD, as I have already mentioned, have a minimal change in the overall interaction scores. And UK122 and C15, both the compounds were found to be absorbed in GI tract. And neither of the compounds are P-glycoprotein substrate, hence they have less likelihood of getting efflux out of the cell. So this is a very basic attempt to develop a novel compound based on pharmacokinetic evolution from a parent molecule with solubility and toxicity issues without changing the specificity of the molecule to a particular enzyme target to be a more suitable drug molecule. I'm thankful to the Department of Biotechnology and Bioinformatics, NEHU, Shillong, for providing me the necessary infrastructure for carrying out the computational work. These are few of the references that I have mentioned.
Thank you.
I'm sorry, did I just... Yes, I'm sorry, did I just... Sir, I think my slide just... Hello?
Yeah, we are able
Am I
to
audible,
see.
sir?
Yeah, you are audible.
But sir, okay, I don't know. I cannot see my slide. Sorry, I think maybe some technical.
No, we
error.
have seen all this. Yeah, we have seen all your slides. So,
okay, sir. Thank you, sir.
Thank you for your presentation. And there is a question: are there any other bioactivities of this compound, like for other diseases other than what you have
Yes,
presented?
sir. Yes, sir. This compound is like actually have a specificity to other targets as well. Some it has certain interaction with MMP9 as well, sir.
Okay.
Like there are papers which have quoted it. Yes.
Yeah, thank you, Rick Ganguly.
Thank you, sir. Thank you so much.
Yeah, now I request Dr. Renuka to chair the two poster sessions.
Thank you very much, sir. Yeah, the next poster talks will be like by Saptadeep Pal and Mala Mujumdar. The topic of the title will be like is evolution of the anti-diabetic activity of Madhurakshaka active, providing new insights with inverse virtual screening. I would like to request to take 3 minutes for this poster session. Thank you.
Ma'am, my slides are visible? My poster, I mean?
No. Please share it.
Oh, 1 minute. Is it visible now? Hello?
Yeah, it is. Yes, it is.
Good afternoon, everyone. I am Saptadeepa Paul, working under the guidance of Dr. Mala Majumdar from Jain deemed to be University. And my topic of presentation, poster presentation is evaluation of anti-diabetic activity of Madhurakshak active, providing insight with inverse virtual screening. In my present study, I have selected a polyherbal formulation, Madhurakshak active, which is being used for management.
Sorry to interrupt you in between. Can you make Zoom full screen?
Is it fine?
Yes, it is fine.
Yeah. So in my present study, I have taken a polyherbal formulation, which is Madhurakshak active, and it is widely used for management of diabetes mellitus, which is a multifactorial disorder. And it is claimed that this polyherbal formulation has synergistic mechanism of action, but we do not have any scientific data regarding the same. So what I have done is I have taken the polyherbal formulation and I have subjected it to LC-MS analysis, from which I have got 150 compounds, and I have taken those compounds for literature search. From literature search, I came down to 20 compounds which were having lower data regarding diabetes. And those compounds were further subjected to ADMET properties, Lipinski rule, and physicochemical properties, among which I have finally came down to 6 compounds, which is 6-hydroxylutenol, glyceraldehyde, and the glucose arylamine, dentronil, grandonil, and 3-O-methyl gallate. These compounds had shown proper drug likeliness, and hence they were taken. They were further taken for DIB web server, where these compounds were docked against 17 anti-diabetic targets.
Okay, now?
From the data that I have obtained from the server, showed that these compounds were having anti-diabetic activity. Compounds that are 6-hydroxylutenol, glyceraldehyde, and the glucose arylamine, dentronil, exhibited maximum binding value against 8 or more anti-diabetic drugs. Thus, it can be further considered as a potential phytopharmacological agent. And also, 6-hydroxylutenol has displayed better binding potential against all the targeted protein considered for the study. The conclusion that I have arrived from the study is that multi-compound, multi-target regulation of anti-diabetic polyherbal formulation Madhurakshak active showed potential inhibition of all the protein target. The result of this study, taken together with the known scientific literature, indicates that anti-diabetic potential of the Madhurakshak active and provides data for synergistic mechanism of action of the polyherbal formulation. Thank you.
Thank you very much for this poster presentation. Have you done some computational studies, or only the LC MS studies were screening was done?
No, ma'am. First, I have done the screening from LCMS. After that, I have done physicochemical analysis, ADMET, Protox, I have used for toxicology.
Okay, good work.
Thank you.
Yeah, the next poster presentation will be by Vinay Kumar and his team. They will be presenting on in silico analysis of the inhibition of butyryl cholecystyrase enzyme in Alzheimer's disease. A D2 QSR approach they are having. Yeah.
Hello, am I audible? Hello. Hello.
Yes.
Yes, you are audible.
Can you see my poster?
Yeah. You can present. We can see.
Okay. Good afternoon, everyone. Myself Vinay Kumar from Jadavpur University, and today I'm going to talk about my recent investigation entitled "In silico analysis for the inhibition of butyrylcholinesterase enzyme in Alzheimer's disease, a 2D QSR approach." Before I start to talk about my investigation, I wish to enlighten on basic about Alzheimer's disease. Actually, Alzheimer's disease is the most common form of dementia in elderly individuals. It is chronic, irreversible, progressive, and neurodegenerative disorder with both genetic and non-genetic causes. And the word Alzheimer was first coined by German neurologist Dr. Alois Alzheimer in 1906. According to the World Alzheimer Report 2020, there were around 55 million people worldwide living with dementia and are expected to exceed 80 million in 2030 and 152 million in 2050 worldwide. So there are only 5 FDA-approved drugs reported, which have only symptomatic relief, that are Acrin, Donepezil, Galantamine, Rivastigmine, belonging to the class of acetylcholinesterase inhibitors, and last Memantine belong to the class of NMDA receptor antagonists. So the key point is there is no cure for the disease till now. So there is urgent need to identify a novel treatment strategy which can cure the Alzheimer's disease properly. To keep all these points in mind, in this investigation, we have attempted to develop 2D QSR model using 1130 butyrylcholinesterase enzyme inhibitors to identify important structural features responsible for this enzyme. This is my methodology which we have adopted in our study. First, we have collected 1130 diverse chemical compound with defined inhibitory activity from previous published literature after multiple steps. And one thing I want to mention here, in this study we have used only 2D descriptor have been used to develop QSR model. Unlike 3D descriptor, the 2D descriptor allow development of simpler and more reproducible model because there is no requirement for the optimization of 3D structure. And after multiple steps, finally we have developed the PLS based regression model, and the developed model was validated by using various internal and external validation parameter, and we also performed the applicability domain assessment of tested compound, and finally we have performed the mechanistic interpretation of the model descriptor. This is the equation which we have derived in this study. This equation, this model qualifying all the acceptable internal and external validation parameters, and the descriptor in this equation are arranged according to their contributions. And this descriptor which are contributing positively, that means when the descriptor value increases, the interactivity also increases, and while the descriptor contributed negatively, that means when the descriptor value increases, the interactivity gradually decreases. This is the scatter plot. In this plot, we have established the proximity between the observed and predicted value of training and test set. Additionally, we have also performed the molecular docking study to identify the interaction pattern inside the active site of the enzyme. And for the conclusion, multi-layer variable selections help in better data handling and remove chance of intercorrelation. Our developed model can efficiently predict the inhibitory activity of new analogs, and the structure obtained from this model and information obtained from the docking studies may helpful for the rational design of novel inhibitors. These are my references, and I'm thankful to ICMR for the providing SRF fellowship. Thank you so much.
Thank you very much for this excellent poster presentation. And yes, I think I would like to give ask Dr. Nagamani to summarize the session. Thank you.
Okay, thank you. Shall I stop?
Yeah, you can stop.
Yes.
Thank you, ma'am. So in this session 2, we had 3 different invited talks and 2 oral section and 2 poster session. So in the first invited talk, Dr. R. Parthasarathi from CSIR IITR. So he has given a brief introduction about this toxicoinformatics, and also by applying the machine learning, how we can develop the data-driven model for toxicology. And in the second talk was delivered by Professor Kunal Rai. So he has shown very briefly about the QSAR and how we can apply the QSAR method for nanotoxicity. And the third invited talk was delivered by Professor Vibha Tandon, and she elaborately explained about the work which has been done through computational as well as the experimental studies in her group. So after that, we had a 2 oral session and 2 poster session. So with this, we have end to this particular session 2. So now I think I would like to hand over this session to InBix committee for the further proceedings.
Yeah, thank you, Dr. Nagamani.
Yeah, we would like to please open your video so that we can have a group.
Yeah, we can.
Thank you.
Yeah, please, everyone, request you to please open your video.
I think Vinay Kumar, he has to unshare his screen. You please unshare your screen.
Please, everyone. I hardly
You
see
just
very few people are here.
actually you have, yeah, Vinay Kumar, you have to answer that cross mark you have to press.
I see only 5 people switching on the camera. There are more than 40 plus. Please don't feel shy. There's nothing to really feel shy here. Hardly 7. Yeah, all the participants. Lipsa, there are a lot of participants. Yeah, please feel free to open a video. Yeah, please 30 seconds more. Yeah, please take your time. Please let me know you have any issues. Yeah, please give a smile. Yeah, thank you so much. Yeah. Yeah, now we have a buffer time with networking. We are here, like you can ask any questions that you get. We'll start sharp at 4:40. Okay, with the elevator pitch. Okay, so we'll meet sharp at 4:40. Yeah, thank you one and all. Thank you. Yeah, welcome back, one and all. Now we have come to a final session. It's based on elevator pitch. Describe yourself and your work. You pitch an idea and explain in 30 seconds. And the rapporteurs includes Santosh Kumari Duppala and Mohar Kumar Megha. So Santoshi and Megha, please take over the session. And after that, you can conclude the, yeah, I'll get back to you by the time. Yeah, thank you.
Yeah, thank you, sir. Thank you.
Thank you, Sugu.
Yeah, good evening, everyone. Very warm good evening. And thank you for giving me this opportunity. And I think like since morning, we all have a very good sessions, like wonderful sessions with keynote speakers and invited talks and oral talks and poster talks. And now we are having a elevator pitch. So till now we have all the scintillating talks. So let's start with our elevated pitch also. So first, let me introduce myself. Like, I'm Santosh Kumari Duppala, research scholar from a lovely professional university and a core member of BioClues. So now I would like to tell about even job opportunities. Like here, a few students are also there, like students and research scholars and postgraduates who are all there. Like we are just thinking like a few companies are associated with our BioClues. So the even students will be helpful. For students, it will be helpful who are interested in like job opportunities. Those who are interested can give their names to us so that we'll get back to you on this. So and Mohan, can you introduce yourself?
Yeah. Yes, my name is Mohan Kumar Megha. I am working as a senior specialist in Eli Lilly Private Limited in Bangalore, and I am a core member of BioClues. So yeah, that's about me. And let's continue, Santosh Kumari.
Yeah, okay. So from the participant list, like I would like to call by first names. So we'll be giving the 30 seconds. Within 30 seconds, you will be introducing yourself and you'll be telling about your work and everything. So first name is Ayushi Wellinger. I would like to call Ayushi to introduce herself. Are you there, Ayushi? We'll go next one. Alfredo Benso. I think few.
Hi.
Being late. Yeah. Hello.
Yes. Hi.
Hi.
I was not thinking to be called right now because I have a keynote tomorrow. I was just
okay.
Following the session to know you guys.
Okay. So next, Anjumaro Patel.
Don't know why they are not responding.
I think Sugu sir, everybody went for a break. They haven't returned back yet.
Yeah. Is everyone back from the break?
Yeah. Everybody is silent. They'll just call the names like Ayushi, are you there?
Or we can continue with who all are there. All those who are there, can you please raise their hand and so that we can continue from yours.
It's like anyone can give, and I would like to tell that the best elevator pitch is going to have a gift from BioClues, BioClues coffee mug. So just prepare yourself to tell what you want to tell? CSIR. Sir, here I'm getting the name that CSIR. Who? Dr. Sandeep. Yes, sir. You can say, sir.
So can I continue?
Sir, Jairam sir, please go ahead, sir. You raise your hand.
Okay. Sorry, you talked about. She talked about Dr. Sandeep. Okay, I will talk. Yeah.
Yeah, you can.
Yeah. I am Jayaraman.
Yes, sir.
Bioinformatics
Please
Bioinformatics and machine learning are my lifeline. Not necessarily in that order. Yeah.
Thank you, sir. Thank you. Next, Dr. Sandeep, would you like to say?
Okay, so I'm working as an assistant professor in JNU University currently. So I want to create my career in next generation sequencing and how I can use bioinformatics in the cardiovascular biology to solve the issues of cardiovascular biology. That is one of my aim with this entire thing.
Okay.
That's all from my end.
Wonderful, sir. Yes, sir. That's wonderful. And next, Anpreet Chaudhary, would you like to introduce yourself?
Hello.
Yeah, Anpreet, we can hear you.
Hi. Good evening, everyone. I'm Anpreet Chaudhary. I'm a MTech student. I'm doing my MTech from Lovely Professional University, and I'm working on potential regulatory role of long non-coding RNA and miRNA with the protein-protein interactions of multiligand age-range axis. So I'm working on this. Thank you.
Okay, that's very nice. And
Good, Anpreet.
yeah, next Anpreet, Antra.
Call Antra, yeah.
If Antra is not there, like Saptadeepa Paul raised her hand.
Yeah, I'm Saptadeepa Paul. I am a PhD scholar working in Jain University under Dr. Mala Majumdar, and I'm working on unveiling the mechanism of various anti-diabetic polyherbal formulation using various in vitro and in silico approaches.
Okay.
That's all. Thank you.
Thank you. That's wonderful.
Thank you.
Anvesh Pandey.
Anvesh Pandey, can you go next? Yes, Anvesh.
No, I think he's not there. He's there.
He has unmuted his
Okay.
mic.
So next we'll go with Arvind Preet Kaur.
Hi everyone. I'm Arvind Preet Kaur. I recently finished my master's in bioinformatics from HMV College Jalandhar, and recently I joined Birla Institute of Scientific Research Jaipur as a Design and Innovation Fellow. Thank you.
Thank you, Arvind.
Thank you, Arvind.
Thank you.
Next.
Bhanushri.
Yeah.
Hello, everyone. It's my pleasure to meet you all over here. Myself, Anushya Vaisa. I'm doing my research in nanoparticle toxicity from the Department of Geology, Mizoram University, and I research from the beautiful part of India, that is Northeast India, Assam, from the Brahmaputra Valley. That's all. Thank you.
Thank you. Bhargav.
Hello all. I'm Bhargav Kumar. I'm a PhD student with Dr. G. Narahari Shastri in CSIR NEAST. I am a computational chemist and I mainly focus my work on non-covalent interactions. Thank you.
Next, Kikru.
Kyuho.
Hello. This is Kikru. Yeah, hello, can you hear me?
Yeah.
Okay,
Yes.
hi. Okay, so I'm Kikru and I'm a research scholar under the supervision of Dr. G.N.R. Sastry at CSIR NEAST, and my research area mainly focuses on the database and knowledge relevance creation and the analysis of this data for their pharmacological properties, and also incorporating the various parameters of artificial intelligence and machine learning. Thank you.
Thank you, Kikru. That's wonderful. Is that?
Yes, sir. Hello, sir. Hello, am I audible?
Yes, you are.
Okay. Hi, this is Esther Jamir. I'm a PhD student working under the supervision of Dr. G. Narahari Sastry at CSIR NEAST. My area of research includes identifying therapeutic agents for communicable diseases, and with a main focus on viral and bacterial diseases. And I aim to focus on open source tools and programs for lead optimization and designing inhibitors against trackable targets. Thank you.
Thank you, Esther. Gayatri, would you like to go next? Gayatri Gogoi. You are not audible, Gayatri. You may want to unmute your mic. She's not audible. Can we move on to
Yeah, we can
Ghazala?
move on to next. Please let us know who you are and just introduce yourselves. Ghazala. Two hand raise like Rekha. Rekha, would you like to introduce yourself? Please go on.
Good evening, everyone. My name is Rekha Sharma, and I'm pursuing integrated MTech in biotechnology from Lovely Professional University. And I am working on the potential regulatory role of non-known coding RNA, miRNA, and protein-protein interaction of multi-ligand range axis due to proximity of receptor in cancer type. And I'm working with the Anprit Chaudhary. Yeah, that's all. Thank you so much.
Thank you, Rekha.
Thank you, Rekha.
Next, Leisure John.
Yeah, good evening, everyone. This is Lijo John, and I'm a research scholar working under Dr. Narahari Shastri. And my basic area of interest is application of AI and ML methods in trying to understand the diversity and the multidimensionality in the chemical space. And also, as I demonstrated yesterday, and you might have listened to our MPDS portal of chemical space. So that is also my another work that I am currently pursuing. And additionally, I'm also the life member of the BioClues.
Yes.
So
thank
that's
you.
all about me. Thank you.
That's nice. So wonderful.
Thank you, Lijo.
Yeah, thanks.
Next, Shalini Rajgopal.
Good evening, everyone. I'm Shalini Rajgopal, research scholar working under the guidance of Dr. Prashant Sravachala from Amrita School of Biotechnology. My work is about dissecting vitamin K pathways in humans using next generation sequencing analysis. Thank you.
Thank you, Shalini. Next, Nidhi Shukla.
Good evening, everyone. I'm Nidhi Shukla. I'm a research scholar with Dr. Prashant Suravajala. I'm using next generation sequencing approach to study mutations, which is inherent to prostate cancer, is specific to Indian population. This is all about me. Thank you so much.
Thank you, Nidhi. Next, Rick Ganguly.
Hello, am I audible?
Yeah.
Hello. Okay, I'm Rick Ganguly. So I'm working in the Department of Biotechnology and Bioinformatics from Northeastern Hill University, Shillong. And my research topic is screening and identification of inhibitors for enzyme involved in plaque instability in atherosclerotic conditions. So I'm working under Dr. Atanu Bhattacharya, who is also specialized in computational biology. Thank you.
Thank you.
Thank you so much. Next, Lipsa Priyadarshini.
Hello. Good evening, everyone. I am Lipsa Priyadarshini, working under Dr. G. Narahari Sastry at CSIR NIST. So my current area focuses on integration of machine learning with molecular modeling tools, and I am also involved in developing open source computational drug discovery tool named as molecular property diagnostics. Thank you.
Thank you. Santoshi, we have left a few people after G. So we'll go back to Gautam.
Okay. Yeah.
Gautam Moirangthem. Hari Prasad B.
Yes. So my name is
Gautam, please.
And I work under Dr. Wahen Mumoni in the Center for Infectious Disease, CSIR. And my area is mainly studying gut microbiology in English cardiovascular disease.
Thank you, Gautam.
Thank you, Gautam. Hari Prasad. Okay. Amit Nargotra.
Yeah, hi. Good evening, everyone. I don't know whether I am actually for this session or not. It was just on and I heard my name. I'm sorry. But anyway, I'm Amit Nargotra and I'm working as a principal scientist in IIM Jammu. And I dived in this interesting journey of computational chemistry and bioinformatics around 20 years ago. And by training, I am an engineer and that too in electronics. Then I did my computers and did PhD in bioinformatics, and I think it's really have been a very fascinating journey in bioinformatics, being surrounded by all the wet lab people over here. And the best part is that I can do whatever I am interested in, like whether it is Alzheimer's, whether it is tuberculosis, whether it is cancer, or all the studies. And I think if I, as an engineer, can do it, you all can do much better than me. Yeah. Thank you.
Sorry to intervene. Nargotra is a invited speaker for tomorrow's event. Hello, Dr. Amit Nargotra.
Hi, Dr. Nagamani. Good evening.
Yeah, it is Sugunakar. Yeah.
It's very inspiring, Amit Nargota.
Yeah.
Yes, sir. Very motivational. Next, going to Munmi Bora. Munmi Bora, are you there? Yeah, next, Romi Wahangbam. Mr. Romi. Okay.
Can we call Lukapriya Datta? Lukapriya Datta.
Is everyone present over here? And still not answering. Oh.
Muhammad Hussain. Some people are joining in now. I mean,
Okay.
Muhammad Hussain. Munni Bora.
Okay, we can go next, like Palak. Palak. Then Pradeep R. Okay.
We are done with Rekha and Rick. Next we can go to
Sir.
Shalini. Oh, sorry, she is also done.
Veena Rawat. Veena, could you like to introduce? Okay. Javed Hazarika.
Hello, am I audible?
Yeah.
Hello, myself Javed Hazarika. I'm working as a postdoctoral fellow with Dr. G. Narahari Sastry sir at CSIR NIST. My area of interest is large scale and multi-scale simulation in the context of protein function paradigm. Thank you.
Thank you.
Thank you.
I think Shivam Pandit has joined.
Yes, ma'am. Good
Yes.
evening, ma'am.
Yeah. Would you like to tell about yourself and your work? And
Yes, ma'am. Sure, ma'am. Okay. A very good afternoon to good evening to everyone. Myself Shivam Pandit. I currently pursued my BTech in biotechnology. I have completed my BTech in biotechnology this year, and I am actually working as an intern in CSI NIST for the Drug Discovery Hackathon 2020 and mentorship program. And my area of interest, I would love to explore more in the field of cheminformatics and understand the computational drug designing aspect more. And that's what in a nutshell, ma'am. Thank you.
that's wonderful. So, is there anybody? Sundar sir. Yes. Okay. So this elevator pitch is not only like you can speak about your work. Everybody can speak actually, even students and researchers, even like professors, anyone about their work. So I think we finished. Everyone else?
We can give 1 minute if anyone wants or has been left. 30 seconds. If anyone wants to, please raise your hands.
Yeah, hello everyone. Am I audible?
Yeah.
Yeah.
Yes.
Myself Nandan Kumar, working under the supervision of Dr. Jinarari Shastri at CSIR NIST, Jorhat. And actually, I work in the field of protein and membrane modeling, where I try to like analyze exactly how the different components affect the stability as well as the complexity of the large system, especially the membrane, biomembrane. Thank you.
Thank you.
If everyone we have finished, is there anyone who want to speak?
I think we are done, Mari.
Yeah, then we'll just hand over and just last end notes. And here, as we told like before, is there anyone who wants like any job opportunities, who are searching for any job opportunities? Then you can just mail us, or you can just now tell, you can tell right now itself who are interested to do any job, or who are in search of that.
No, I would like to just add to Santoshi what she's saying. Santoshi, with your permission, yeah.
Yes, sir. Yes, sir.
It's more like a job assistance, like someone who's wanted to explore, and that's where we are trying to pitch in. As being a BioInclusive not-to-profit organization, we are trying to give some kind of job assistance, and we are associated with some companies. So students who want to do job, like they wanted to explore it that at this once they finish their MTech or BTech. So if you're looking for any job opportunities, yeah, please do let us know. You can always feel free to just send a mail with your CV and let us know your interest. Then accordingly we can get back to you. It's more like an assistance. Okay, you wanted to develop some kind of area. You wanted to know. There are some mentor-mentee projects is also being handled. It's not chargeable. Let this be very open. We have an excellent scientific fraternity across more than 30 to 40 countries, where people will pitch in and then try to help in building up a mentor-mentee projects. So please feel free to contact us. You can always reach out to bioclues.org to fill your details. Yeah, thank you.
Thank you, sir.
Thank you, Subha.
Would like to end the session.
That's nice to know. So, Saptadeepa Paul. Yeah,
yes.
We look forward to listen to you, Saptadeepa. Yeah, you can always contact Santosh Kumar Dupala here. She's a finance chair for BioClose, and she's joint secretary, and she'll do the needful. Okay.
Okay,
yeah,
sir. Thank you.
Yeah. And if there are no further points, Santoshi, we can end up the day. But before that, I'd just like to brief regarding day 2. And we have some scintillation talks tomorrow, November 12th, starting at 9 a.m. With the invited speakers, Gyaneshwar Chobe, Anshuman Dikshit in the morning session, And the theme would be on frontiers in biosciences and machine learning. And thereafter, we have 11 to 12, we have a panel discussion, Galaxy India timelines and challenges. We have panelists, Dr. Narhar Shastri sir, Pardasarathi, Jayaraman Valadi, Kiran Telukunta, Harpreet, Gareth Price, Nagamani, and then Anshu Bharadwaj. Then followed by, we have session 4, advances in translational omics. Innovative talk from Josna Batra, a large-scale association analysis of MIR SNPs with prostate cancer. Followed by innovative talk from Amit Nargotra. Just we happen to listen to sir's points, like he clearly mentioned that. Then followed by Rajesh Sani. Then we have next session on bioinformatics for school children. The young minds who are young researchers who are here, who are like BTech and MTechs, and then after you have PhDs, you have postdocs, and then you have extremely talented expert people over here, professors from different government and central government institutions and research institutions in India, even from abroad. They'll happen to listen and interact and listen to the school children. And BioClues takes extreme pride in introducing those school children working on different projects with a allied group of scientists in bioclues.org. So I request like everyone to come get some kind of motivation. I feel that is very important. Please come and encourage the young minds as well, so that will really very rewarding for them. So I believe like you will give your active participation tomorrow. And thereafter we have a cultural event trying to show the focal music and then the Northeast region, the flavor. As we know, we are India with a lot of cultural diversity. So let us witness that tomorrow in the cultural event from 4:15 to 6 o'clock. I hope you will have a very peaceful and relaxing day from 4 to 6 p.m. tomorrow. And that's all from my end. And I hope we look forward. Sharp 9 a.m. without any further delays from our side. Sharp 9 a.m. we're going to start. Like I thought, like there will be more participants will be there, but the moment they heard it's elevated pitch, so I think they were a bit hesitant. Yeah, so active participation is expected. That's what BioClues believe in.
Dr.
Yeah. Prashant, would like to say something, please?
Yeah. Thank you so much, Sugu, for taking this along. Santoshi and Mohan, for organizing this. So I'm sorry I was not there, but I'm sure there were many students, or participants who participated in this elevator pitch. Yeah. So probably we'll connect them to different stakeholders, especially the company CEOs, during the next, probably week or so. Maybe we can even connect them to the persons, directly, by email. Yeah. But as you rightly point out, I just want to echo that, the participation should be strengthened in such a way that every participant, should speak, should ask, some kind of questions. Yeah. So, Sugu, did everyone, participate in the elevator pitch?
Yeah, mostly, Prash. Mostly they have participated. Some like they're still shying out. So
yeah,
we expect
we agreed. Sugu, we agreed that they have to pay a fine of chocolate, right?
Yeah, absolutely. Absolutely.
Okay. Just I'm sure Santoshi has noted down those names.
Yes, sir. Yes, sir.
Yeah, good. So friends, don't worry. Those who have not participated in elevator pitch, we are absolutely chocolate lovers. We usually take chocolates. Okay, from that. Thank you.
What about giving mugs to us? Those who participated.
Thank you.
Yeah, thank you so much. And that's all from our side today. For today, yeah, take care and see you tomorrow sharp 9 a.m. Yeah, thank you so much.
Thank you. Thank you all. Thank you, sir.
Thank you, everyone. We'll see you tomorrow.
Yes.
Thank you all.
Subbu sir, once wait. A few people are just interested. They're giving their names. So
yeah, please, I'm here. Not at all, not an issue at all. I'm here itself.
Yes, sir.
Yeah.
Saptadeepa Paul and Rekha.
Yeah, we are there. We are there. You have one group photo if like everyone is here?
Yes, sir.
Yeah.
We'll have a group photo.
Yeah, everyone. It's a
Please,
group
photo.
Yeah, please. Could you please switch on your cameras?
Yes.
And I'll take extreme pride in taking a photo. I see only 6. Please.
Sundar, sir.
So more than 30 are here. Yeah, thank you, Nidhi. Yeah. Yeah, Lizzo is here. Yeah. Where is Mohan?
Sir, my
Mohan?
camera is not working.
Oh
So
God,
sorry for that.
You missed it. Okay. Prash, Hariprasad, Lipsa Priyadarshini. Okay. That's all. Okay. Yeah, thank you and all. It just like it's like the last event for the day. You can just give a smile. It doesn't cost long. Okay. Thank you. Thank you so much and look forward to see you tomorrow morning 9 a.m. Thank you. Bye bye. Take care.